Do we have both the speaker for this session?
Yes, we have. I think the speakers still available. Yeah.
Namaskar, Sushil sir. Good morning, Shakti ma'am.
Good morning.
Neha ma'am, do we see Dr. Neeraj? I think he's one of the speakers in this.
Not yet, Devanshi.
Just a second, I'm getting a call. That's about the speaker.
हाँ हो गया. Hello, good morning everyone.
Good morning.
Good morning,
Good
ma'am.
morning, Shipra ma'am. Good morning.
Good morning, ma'am.
Good morning. Dr. सुशील.
Yes, ma'am.
वो एक्चुअली एक ही स्पीकर है इसमें, एक को सडनली कुछ मिसहैप हो गया.
हाँ 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 हाँ.
डॉक्टर नीरज टंडन ओनली शी विल स्पीक.
Okay.
एंड हाँ, नूपुर बोस इज नॉट एबल टू.
Okay.
तो
Then we can give the sufficient
वैसे
time
भी
to
थोड़ा
her.
लेट, कोई बात नहीं, थोड़ा टाइम दे सकते हैं उनको, मतलब कोई ऐसी बात. मैं उनको कॉल करके देखती हूँ. शी इज नॉट देयर.
Namaskar, Shipra madam. Maulin here.
Oh, Molin, how are you? Good morning.
I'm fine, like you.
Yeah, nice to see you here.
Thank you so
कल
much.
आप थे कि नहीं थे कल?
कल मैं था ना, लेकिन कनेक्शन में प्रॉब्लम हो रहा था, पता नहीं ज्वाइंट ही नहीं हो
मॉर्निंग
पा रहा.
में देयर वाज सम प्रॉब्लम. कोई बात नहीं, आज रहिएगा.
हाँ.
आप शायद ओरल में नहीं रह पाएंगे ना?
नहीं, ओरल में मैं हूँ.
आप रहेंगे? अच्छा.
हाँ हाँ.
इनवाइटेड में शायद वो शंखा जस्ट कॉल्ड मी एंड सेड कि शायद यू वोंट बी एबल टू.
नहीं,
नहीं, मैं
मॉलिन
present
सर तो रहेंगे मैम, वो दूसरे जो चेयरपर्सन है ना.
नहीं, इवन आप
नहीं,
रहोगे
मॉलिन
फिर
सर
ठीक है.
नहीं, मॉलिन सर शायद ओरल में नहीं रहेंगे, मतलब इन करके रहेंगे, लेकिन सर का कुछ मीटिंग है, अर्जेंट काम है, इसीलिए सर चेयर नहीं कर पाएंगे. इसीलिए डॉक्टर जयतो नायक विल बी द चेयर.
ठीक है, तो शंकर डॉक्टर जैतो का आप डिटेल्स भेज दीजिए उसमें ग्रुप
यस
में.
यस ऑलरेडी हैज दिस
अच्छा,
डॉक्टर जयतो.
Yes, good morning. Am I visible? Shipra madam?
Yeah.
Dr. Jayathan Nayak here.
Nice
Yes,
to
hi.
see you. Good morning.
Yes. So anyway, I have received your mail from Dr.
Okay.
Shanko, and yeah, I have accepted it, so it's okay, no problem. I'll be
ठीक
there.
है, then it's okay. Then डॉक्टर मॉलिन आप अपना लिबर्टी ले सकते हैं, कोई प्रॉब्लम नहीं है.
श्योर, थैंक यू. नहीं, मैं प्रेजेंट तो रहूंगा.
हाँ, डेफिनेटली. मतलब आप इनवाइटेड में तो आप रहेंगे ही ना? इनवाइटेड में. ठीक है.
ठीक है.
आई एम जस्ट कॉलिंग, ट्राइंग टू कॉल नीरज जी.
नहीं बहुत कुछ. हाँ.
दिनेश जी, वो उनको एंटर एक्चुअली नीरज टंडन इज नॉट एबल टू एंटर, कोई उनको एंटर कर दीजिए।
मैम सेकंड एंटर
डॉक्टर नीरज टंडन।
मैम वो दिख तो नहीं रहे हैं, वेटिंग लिस्ट में भी नहीं दिख रहे हैं उनको। मैम
वो
बोलिए,
बोल
शायद
रही
वो
है
अभी
हम मे बी अगेन ट्राई कर रही हूँ।
मैम
ट्राई करें तो हम।
वेटिंग रूम इज डिसेबल मैम, एनीबॉडी कैन जॉइन विद द लिंक।
अच्छा, पता नहीं क्यों, क्या फिर से ट्राई कर लीजिए। नीरज जी, आप फिर से ट्राई करेंगे एक बार? ओके, थैंक यू।
Ma'am, if sir is not able to join, sir can use Zoom ID of some other person, then sir can join.
Hello. Madam. Yes, madam. हाँ मैडम, ऐसा शायद एक लेटर तो आया था मैडम। ये वो वर्मा
आ
जी के
गई
पास।
क्या नीरज जी? शी एंटर?
No, ma'am.
नेम से शायद नाम
नहीं
भी
है मैम अभी। आप आई थिंक एक बार
नाम नहीं आया
दोबारा
था, है ना?
पता कर सकते हैं। हमको प्रॉब्लम क्या आ रही है, देन वील ट्राई टू सॉर्ट आउट।
ओके। She will come in 5 minutes.
Okay, ma'am.
She says that she will take 5 minutes. देवांशी, just for everybody's information, actually उनका थोड़ा ICMR में the net problem is there. She's saying that it is going to solve. थोड़ा सा टाइम लगेगा. हमने कहा ठीक.
अच्छा अच्छा, कोई बात नहीं, ठीक है मैम, ठीक है.
I mean everybody should not feel perturbed.
Host, I think we can have some instrumental music playing in the meanwhile
some
then,
video or
Yes, ma'am.
something.
or any videos, a tattva
हाँ.
video or anything, any fillers videos that we have, we can just put it on till the speaker arrives.
Right.
Okay.
Thank you. Thank you.
Okay.
Good morning, ma'am. Ma'am, you are already co-host, so just it's from your ending. Just check it. Yeah, ma'am, that's fine.
मेरे को शेयर करो मेरा वो प्रेजेंटेशन शेयर करना स्क्रीन शेयर कर दो पावर पॉइंट है वो खोल दो पहले पावर पॉइंट खोल दो इसको बंद कर दो इसको बंद कर स्लाइड मोड में कर दे तो हो जाएगा क्लिक स्क्रीन
Neha ma'am, I think we have our speaker here. So we can begin with the sessions. In the meantime, they are doing the settings and we can introduce our chairpersons.
Okay. So should I start?
Yes, I think we can go ahead now.
Okay.
Yes, Shipra ma'am, should we start?
And Dr.
Yes,
Manisha
yes, I
and
think so.
Dr. Hemant is also there. Yeah. Okay. So good morning, everyone. I, Dr. Neha, along with my fellow colleagues, Dr. Hemant Verma and Ms. Chawla, welcome you all to the keynote session. It gives me an immense pleasure to introduce Dr. Sushil Singh. Dr. Singh is presently working as a scientist F in a carbide division at Solid State Physics Laboratory, DRDO. He obtained his bachelor's and master's degree in physics from MVM College, Bhopal, and doctorate degree in the superconducting at Barkatullah University, Bhopal in 1993. I think, please mute.
मैम आप म्यूट हो गई हैं नेहा मैम
नेहा आप ना म्यूट हो गए हो
Okay. Should I start from starting or by the time I just...
No, I think only the last line you missed.
Okay. He obtained his bachelor's and master's degree in physics from MVM College, Bhopal, and doctorate degree from the superconducting from the Barkatullah University, Bhopal in 1993. In 1994, he received his postdoctoral fellowship work at Warwick University on oxide thin films. He joined as a faculty position in GGD University, Bilaspur in 1997. He has been awarded Commonwealth Fellowship in 2001, Tokyo University of Technology, and invited him as a JSPS invited fellow in 2004. He has been engaged in the research of ferroelectric thin films for non-volatile memory application. He worked as a senior researcher in the next generation FERM project jointly run by the Tokyo Institute of Technology and Fusey Toshu, Japan. Since 2008, he is working as a SSPL, and he has been engaged in the R&D projects such as silicon carbide bulk single crystal growth, MEMS-based adaptive mirrors, multifunctional thin films, and cesium oxide nanoparticles for the defense application, and so on. He is a life member of Material Research Society of India and National Environment Science Academy, NESA. He is the author of around 100 research publications in a peer-reviewed international journal and the written 2 books chapters. So welcome you, sir. And now I would like to invite Dr. Hemant Verma to introduce Professor Sachi Shah. Thank you.
Thank you, Dr. Neha. It gives me an immense pleasure to introduce our second chairperson of the session, Professor Sachi Shah. Professor Shah is an environmentalist with 2 decades of teaching and research experience at various reputed universities and institutes. She is presently director at School of Interdisciplinary and Transdisciplinary Studies, IGNOU, New Delhi. She attained her PhD in Environmental Science from G.V. Pant University of Agriculture and Technology, Pantnagar. She was awarded National Environmental Science Academic Eminent Scientist of the Year Award 2019 Commonwealth Education Media Center Fellowship 2013, and ESDF Fellowship Award 2021 at World Environment Summit held at India Habitat Center. Recently, she was nominated by Honorable Chairman UGC as expert committee member to prepare guidelines and curriculum framework for environmental education. She is a member of University Advisory Board 2022-23, Mahakoshal University, Jabalpur. Under her directorship, in line with New Education Policy 2022, various programs like MSc Environmental Science, MA in Sustainability Science, MA in Environmental and Occupational Health, MA in the Folklore and Culture Studies, and PG Diploma in Migration and the Diaspora were launched. She is a member of Steering Committee and Program Coordinator Certificate Program in Skill Acquisition and Knowledge Awareness for Livelihood Promotion launched by IGNO for District Skill Committee members. She is a member of Board of Management, Academic Council, and several other committees at IGNO. She has more than 60 national and international publications to her credit. She has authored, edited many books. She has successfully organized many national and international seminars, especially during the pandemic. She has significantly contributed in the field of environmental monitoring, bioremediation, waste management, bioprocessing, bioconversion of waste, bioenergy, and climate change. Now, over to you, ma'am. Hello.
Yes, we can conduct.
Yes. Okay. Thank you, Dr. Hemant, for such a kind introduction. First of all, Dr. Shipra Misra, convener, EWASH, Dr. Sushil Kumar Singh, additional director and scientist in DRDO and my chairperson of this session, Dr. Devanshi Mongo and coordinator of this session, Dr. Neha, Dr. Hemant, and Dr. Manisha Chawla. First of all, I would like to extend a very hearty appreciation to the organizer of this EWASH for a very successful conduction of the international conference, and also for inviting me to this highly regarded event. And it gives me a great pleasure to extend a very heartiest welcome on behalf of this EWASH committee. I also wish to welcome our keynote speakers, Dr. Neeraj Tandon, who is former scientist G and head Medicinal Plant Division, Indian Council of Medical Research. And as the organizer has informed that our second speaker, Dr. Neeraj Tandon, Dr. Nupur Bose, who is University Professor and Head PG Department of Geography, due to certain pre-engagement, is not able to join today. I welcome you all, and I really hope that today's event will serve as a catalyst for strengthening cooperation on the transfer of knowledge. Conferences like previously organized conferences from STE and Hindu College, this conference will also provide an opportunity to talk about various common knowledge, shared best practices, and also discuss the groundbreaking scientific research and technology, and which will help India as a whole and all the scientific community, research community to grow. As we have witnessed that this year 2020 has seen that our Honorable Prime Minister has launched a flagship program on environment, that is Lifestyle for Environment. Beside that, in Sharma El-Sheikh COP27 also, the decision was taken for decarbonization, for net zero emission. Also, the cheetah has been introduced in by the Prime Minister. Beside that, the focus of the research work and the community is basically on net zero, decarbonization, bioenergy, and resource efficiency, circular economy, and so on and so forth. We have also witnessed I hope you all have witnessed that dance to decarbonize policies to accelerate clean energy transition in India. With this, I welcome you all once again for this session on behalf of organizer. Today, shall I, am I supposed to introduce the speaker of today's seminar?
Ji,
Okay.
ma'am. Please
So,
introduce Dr. Neeraj.
Okay, so today's the seminar, the first speaker of this seminar or the only speaker of this session is Dr. Neeraj. She is going to talk about the phytopharmaceuticals. So as we all know that phytopharmaceutical is a modern terminology. It is for a range of natural chemicals that compromise herbs and that comprise of herbs and plant-derived substances, and they are used as nutritional as well. If I, first of all, I would like to thank to give me this opportunity to introduce Dr. Neeraj Tandon. She is a very renowned scientist. Dr. Neeraj Tandon is a former scientist G and Senior Deputy Director General at the Indian Council of Medical Research, ICMR, and is presently working as a scientific consultant in the same organization. She is involved in the planning, coordination, monitoring, and implementation of various research programs and activity at the Indian Council of Medical Research, New Delhi. She has vast experience of more than 40 years in the area of biomedical research, policy, planning, publication and information, education, communication, and research management. Her primary research area include medicinal plant and traditional medicine program, employing various through these medicinal program are organized or facilitate the development of safe and effective standard drugs for various diseases where there is no satisfactory cure in allopathic system of medicine. It is very difficult for me to introduce because she has extensively worked into this area, which has been evident from various document, patent. She has been principal investigator of many projects. She has more than 75 publications to her credit. She is also WHO Fellow and Medicinal Plant Information System at the Department of Medical Chemistry College of Pharmacy, University of Illinois, Chicago. She is member of many university advisory panel, like she is in the panel USP Dietary Supplement Botanical Expert Committee Monitors and other things. She is also member of task force of many projects. So we are really privileged to have ma'am over here as a keynote speaker for this session. So I once again welcome Dr. Neeraj and request her to present her give her keynote speaker, as initially the time was mentioned in the program was 20 minutes, but if Dr. Sushil Kumar sir give the permission and Chipra Mishra ma'am, we can give her more time, as we are having only one speaker over here. With this, I hand over the session to ma'am, please. Thank you.
Yes, नीरज जी, आप जितना टाइम लेना चाहें लीजिए, अच्छा लगेगा.
Okay, thank you, ma'am. Can you hear me?
Yeah.
Yes,
Sure.
yes, ma'am. Yes.
अरे इस पे नहीं मेरा ऑडियो तो लगाओ, मेरा ऑडियो नहीं है वहाँ.
ऑडियो तो होगा.
मैम हम सुन पा रहे हैं आपको.
Sorry.
Are you able to hear me, please?
Yes, ma'am. You are very much audible. We can also see your screen.
Okay. So, इधर से नहीं पहले से करना, ये तो थर्ड वाला स्लाइड आ गया. इसको इधर करना है.
इसे आगे जाएगा, इसे पीछे आएगा.
Thank you very much, madam, for a very nice introduction, and I'm very grateful to the STE, Dr. Shipra Mishra, and all the associations of all the patrons and co-patrons for giving me this opportunity and speaking on a topic which is relatively new and many people would not be knowing about it. But before I go to the topic of the phytopharmaceuticals, which is a new drug class regulated in India, I will just briefly speak about various regulations to take the medicinal plants forward as drugs for various refractory diseases in allopathic systems of medicines. which do not have a very good cure. So as you all know that India has a very rich diversity of medicinal plants. And I've been working in this area for the last 40 years at ICMR. And as Madam has already introduced me, and I've been involved in various activities relating to these medicinal plants. So there are 4 ways to take the medicinal plants forward. The first one is you can take it by the allopathic route, that is to the synthetic molecules, and get a drug out of these medicinal plants. The second one is through the Ayush route. The third one is through the FSSI route, that is the nutraceuticals or the OTC drugs or food supplements. While the fourth one, which is recently been implemented, is the phytopharmaceutical route for taking the medicinal plants forward. So before I come to the topic, just let me give a brief global scenario of botanical medicines. And as you all know that these herbal medicines are an important alternative to the modern allopathic medicines. And very few medicine plants have been scientifically evaluated for their potential in medical treatments. They are very poorly regulated and often neither registered or controlled by the health authorities. Now, as we can see that the safety of herbal medicines remains a major concern throughout the world. For example, in the United States, the FDA has estimated that over 50,000 adverse events are caused by botanical and other dietary supplements. Therefore, most herbal drugs, the efficacy is not proved and the quality is not assured. So to address this issue, the US FDA has created a separate guideline document for botanical products for NDAs and IND submission 2004, which was revised in August 2015. Similarly, in India, the CDESCO, formerly the DCGI, has created a separate guideline for botanical products called Phytopharmaceutical Guidelines 2013, the first revision of which took place in November 2015. Now, what is the Indian scenario? The major class of Ayurveda, Siddha, and Unani, which is the ASU drugs, there are 2 types of ASU drugs. That is the classical drugs, which are mentioned in the authoritative books of the ASU systems of drugs. And the second is the patent or the proprietary medicines, which makes use of the ingredients referred in the formulations of the authoritative texts, but with intellectual intervention, innovation, or invention to manufacture requires proof of effectiveness based on the pilot study as per relevant protocols. So in 2010, the Ministry of Ayush introduced Rule 158B, which made the requirement of proof of effectiveness for licensing of a patent or a proprietary ASU medicine, and which was followed by the release of the GSP guidelines for voluntary use of the researchers interested in taking up clinical trials in the ASU medicine. So the Government of India published a draft amendment to the Drugs and Cosmetic Act on 24th of October 2013, and this created regulatory provisions of defining phytopharmaceuticals, that is botanical-based drugs, and a schedule providing requirements of scientific data on quality, safety, and efficacy to evaluate the marketing authorization of a plant-based lead as a drug on similar lines to synthetic chemical moieties. So in India, it is known that the new draft regulation was not present when Guglu tablets for treatment of hypercholesterolemia, that is Comifora and the ginkgo biloba tablets to treat liver disorders, were approved and marketed as drugs by the CDSCO, formerly the DCGI. However, it is known that the approval of Guglu tablets took more than a decade to get this approval as a drug. And it had required efforts to convince the authorities that several requirements applicable to synthetic chemicals were not possible for a botanical-based drug. So in India, ASU drugs have been under the purview of Ayush, while the phytopharmaceuticals are under the purview of the CDSCO. The Gazette notification defines regulatory provisions for phytopharmaceuticals and regulatory submission requirements for scientific data on quality, safety, and efficacy to evaluate and permit marketing for a herbal drug on similar lines to synthetic chemical moieties. So in Schedule Y, the newly added Appendix 1B describes data to be submitted along with the application to conduct a clinical trial or import or manufacture a PPH drug in the country. The regulatory requirements for NDA for the PPH drugs include standard requirements for a new drug, safety, pharmacology, human studies, and confirmatory clinical trials. So this is the Gazette notification which came out in 2015, which has been notified in the Parliament for taking the drugs as phytopharmaceuticals. So what exactly is a phytopharmaceutical drug? It is defined as a purified and standardized fraction with defined 4 bioactive or phytochemical compounds, which can be qualitatively and quantitatively assessed of an extract of a medicinal plant or its part for internal or external use of human beings or animals or diagnosis, treatment, mitigation, or prevention of any disease or disorder, but does not include administration by parenteral route. Now, this new regulation for phytopharmaceutical is in line with regulations in US, China, and other countries involving scientific evaluation and data generation. It does not simply depend on the traditional knowledge alone. So PPH can be obtained from a botanical medicinal plants from any part of the globe. It is not necessary that the plant should be from India, and it should be mentioned in the authoritative books of Ayurveda. So this is advantage. Any researcher who is interested in any plant across the globe can do researches under this regulation on that particular plant and take that forward in this route. So why phytopharmaceutical? Why we should go for phytopharmaceutical route? Because this regulation is expected to develop new drugs from botanicals in a very, very scientific way and would help in the acceptance of the use of these products by modern medical profession. This is very important. Modern practitioners can also prescribe these medicines, along with the Ayurvedic people also. So the phytopharmaceutical proposal would promote innovations and development of new drugs from botanicals in a more scientific way, would give boost to research in drug development for innovators, industry, and national laboratories and pharmaceutical labs in India. Now, the scope and potential of these drugs is that either they can be used as complementary drugs or as an adjuvant use. They can be used as a mainstream therapy, or they can be used as safer alternatives to synthetic drugs. Now, there is a lot of stress of PPH for the available information on the plant. It is a very tedious process. You need to have the information on the plant, the formulation, and the route of administration, the dosage, the therapeutic class for which it is indicated, and the claims to be made for the phytopharmaceuticals, supporting information from published literature on safety and efficacy and human or clinical pharmacology information. Therefore, the data generated should be identification, authentication, and source of the plant used for extraction and fraction, the process for extraction and subsequent fraction and purification, formulation details of the PPH, manufacture process of formulation, and the stability studies. So what are the current GMP requirements for to manufacture this PPH drugs? These are similar as prescribed in the Schedule M of the Drugs and Cosmetic Act. However, there is no specific GMP guidelines finalized for PPH drugs. The GMP required would depend on the ingredients and the stage of the processing. Now, what is the possible advantages in this area? This is a new class of drugs. This would encourage introduction of extensive evaluation through biomedical sciences and help in the acceptance and expand use of herbal products by modern and medical professions. Now, across the world, there is a rising interest and demand for plants as a possible source of therapeutics for unmet medical needs, and this would bring this aspiration one step closer to reality. And the number of research laboratories in India, scientific groups, and industrial research and R&Ds who have been working on botanical leads can look forward to take this route for getting marketing permissions. New entrants and investors will also get encouraged to invest in this route for drug development. Now, what are the possible difficulties in this areas? Of the 282 pre-INDs and INDs submitted, only 36% were multiple plant combinations. reflecting difficulties in working with more complex preparations. Now, out of the 282, 113, that is 40%, were submitted by commercial sponsors. The remaining 169, 60%, were proposed by academic investigators, mostly to conduct small-scale proof-of-concept studies and without intention to commercialize these products. Now, despite the increasing R&D activity, progress in developing new drugs from botanicals has been very slow, with only 2 botanical NDA submissions and approvals till date. Now, the possible reasons for disappointing pace of development includes the difficulty in characterizing ill-defined mixtures to provide assurance of quality, besides entering large phase 3 studies, and the problematic translation of unreliable experience in the traditional alternative medicines into testable hypotheses for clinical research. Now, botanical drugs in India, there are very few, and these were before the PPH came into notification, and these are the Guglip. This is perhaps the first drug cleared in India, and Guglip Commiphora mucul plant is based on the ethyl acetate extract, standardized to glucosterols E and Z. The second one is the ginseng extract capsule, standardized to ginkgo bilobides, and silymarin extracts to treat liver disorders, ginger capsules, standardized to G6 gingerols for treatment of chemotherapy-induced nausea, and piperine along with anti-tubercular drugs. Now, in 2004, FDA in US created a pathway for drug makers to get approval of drugs derived from plants. It had received about 500 applications and it approved only 2 botanical drugs until today. The first one was for the genital warts, and it was developed by Germany's Messrs. Medigene. And the second one was from Croton lechuleri, and it was that is the dragon blood tree. It was developed by Messrs. Salix Pharmaceuticals for treatment of diarrhea in HIV/AIDS. So these are the only 2 drugs which have been developed so far throughout the globe under this regulation. Now, what shall be the pharmacopoeial standards for the phytopharmaceutical? Currently, there are no phytopharmaceutical drug has been approved by the CDSCO as a new drug. And as and when CDSCO will approve a phytopharmaceutical drugs, the aspect of including a monograph in the Indian pharmacopoeia would be considered, keeping in mind various other factors. Now, this I want to show, this was the phytopharmaceutical program, and MOU was signed between the 3 secretaries of the CSIR, ICMR, and the DBT, Dr. Balram Bhargav, Dr. Mande, and Dr. Renu were there, and they signed a MOU on 31st of December 2018 to take some of the leads forward in this route in our country. And the various government institutions promoting research are the CSIR, ICMR, DST, DBT, CCRS, NMPB, and the Ministry of Ayush. So as you all know that Dr. Yu won the Nobel Prize in Medicine for her artemisinin isolated from the Artemisia annua for malaria. Now, with this new pharmaceutical regulation, the Indian scientists would now think of developing novel inventions to discover PPH drugs for unmet medical conditions. So what is the need? The need for the day is assessing current status of research, identifying knowledge gaps, laying down priorities, planning research activities, avoiding unnecessarily duplication, and of course, the most important one is the judicial use of the funds and resources. Now, introduction of phytopharmaceutical as a new drug is important, timely, and progressive step. It would inculcate higher level of scientific inputs in herb-based products. PPH may be especially suitable for multi-target based diseases like metabolic syndromes, and it would be a booster dose for the herb-based drug business. Now, coming to the next aspect on what I have done here at the Indian Council of Medical Research, and it is very important to share with all of you, all the academicians, the students, the researchers who are into this areas, that the Indian Council of Medical Research has brought out 27 volumes on the reviews on Indian medicinal plants. I'm not very sure whether you have seen these volumes. These are the encyclopedic volumes of all the details, the chemistry, the pharmacognosy, the pharmacology, the clinical, the toxicological studies, and have been incorporated in each volumes, which have been arranged alphabetically in order of their botanical names. The total number of species so far is 6,394 that goes into these volumes, with the total number of citations referred in these books. These are all original citations, nothing from the internet or no shortcuts. Original citations have been cited in these volumes, and there are about, now it is more than 1 lakh citations that go into the entire Indian work on each particular plant has been documented into these volumes for this ease of the students, because everything is so scattered and getting all of them together under a particular volume is quite tedious and requires a lot of labor. And me and my team worked very hard, and we have till date have reached the T alphabet, and the remaining 6 volumes are remaining to complete the series. And the total number of pages that these volumes incorporate is around more than 30,000 now. So I would request all the college students, all the academicians who are in research may go into these publications. There are so many fractions which were left as a PhD thesis and not pursued further, could be taken up for further research, and taking up this route of phytopharmaceuticals, many drugs can come out in this country. So basically, one has to look into the volumes and the plants, which alphabet of the plant they are interested to work in, and go into that particular volume. And all these volumes I will share with you a little later where they are available. And this is the next activity that the ICMR was involved. Now, standardization is a major issue of the medicinal plants. And if the material, that is the crude drug, is not standardized, one would not get good results. So basically, there are 17 volumes that I brought out for the developing the quality standards of Indian medicinal plants. These gives the qualitative and the quantitative estimations, the pharmacognosy, the dosages, the adulterants, the substituents, the powder microscopy. Everything is there in this book. So anybody who's interested in some kind of any plant should match their results with the description given into these. These are not legal documents. These are the guiding documents for everybody to standardize their material before they can take up this material for particular kind of research. And these are 17 volumes, and they have about 580 monographs. These are the plants which are used by the industry as single drugs or as part of various formulations. And these are the most important 580 plants. So I'm sure that anybody who wants to do any kind of research first should go through these volumes and able to standardize their material before they take up further research, because unless the material is good and the quality of the material is good and not the adulterants or the substituents or the mandis from where the material is brought, you will not be able to get good results. So
Madam,
the
I
third
have just one inquiry here, if you
yes,
permit.
Yes, please.
Do you have the detail about this is for the high altitude or which region, which weather?
Yeah, so we are giving the habit and habitat of each plant. So it is mentioned in those monographs wherever the plant has been collected. This was a multi-centric task force project, and we had collected the material from 9 different regions of the country. And therefore, a average mean was taken out to develop these values. And if the plant is from the region, the high altitude regions, it has been mentioned in these monographs.
Okay.
Now, the third activity that we came, I was very keen to do this particular activity is the phytochemical reference standards. How anybody can develop a standard marker compounds, because in the phytopharmaceutical route, you have to take out 4 biomedical markers from the particular plant. So you need to have a standard also to match whether your marker is matching with the standard markers or not. So in this 4 volumes, I developed 120 phytochemical reference standards, the entire procedures to evaluate your phytochemical biomarker, which you are isolating from the plant, I mean the fraction, and then matching your results with these, the TLC, HPTLC, GCMS, all profiles are given into these 4 volumes. And the material is now with the Pharmacopoeia Commission of Indian Medicine, and anybody can get the material from these, because getting a few μm of this material is very costly. And when researchers need this, they can take out the biomarker from the plants, follow the procedures that are laid down into these 4 volumes, and you can easily isolate your own phytochemical reference standard to match the bioactive markers that you are taking out from the particular plant. So the next activity I would like to discuss is that there were 3 areas that ICMR was very keen to make monographs, and the first one was on the area of liver disorders, because this is the one that many people in the country are suffering from. And in these volumes, I will suggest there are 3 areas that we've been able to bring out, because it is very difficult to integrate the scientific knowledge with the modern medicine as well as the Ayurvedic physicians' knowledge, getting all the 3 together and bringing out a monograph with potential plants, the scientific data on 10, 15 potential plants that are used in this area. So the medicinal plants which are used in liver disorders and are investigated with modern approach, and the methods and the parameters of hepatoprotective evaluation of medicinal plants has been given in this particular monograph. So anybody who's interested in pursuing liver disorders should go through this monograph and see the potential plants. There are about 380 formulations, Ayurvedic formulations in the market for various liver disorders. But I'm very sorry to say that you need to develop a formulation which has been scientifically evaluated for the acceptance by the industry. And this is the second area, that is the lymphatic filariasis, the coastal region. We had a lot of problems with people facing lymphatic filariasis. This is a second monograph, and herein we have 12 potential plants for lymphatic filariasis, and the details, the prognosis, the dosage, the drugs used in modern systems, the Ayurveda, and backed by the complete scientific research on 12 medicine plants goes into this books. And anybody interested in lymphatic filariasis may read this book for any further information that they desire. And the third one is in the area of diabetes mellitus. These have been written by these were task force projects and written by renowned scientists having expertise in their own field. And here again, there are about 12 plants for diabetes mellitus. Entire literature on these 12 plants has been given in these monographs. And of course, we have also taken into the consideration that the Ayurvedic aspects, who should take these plans, who should not, what are the prakriti of the patient, who should require these plans. All these details are mentioned. It's a detailed monograph for people to pursue the diabetes mellitus, and this is one of the very useful monograph for them too. Now, the most important work that ICMR did was developing a compendium for safety reviews on selected, unless the plant is safe, unless it is mentioned in the pharmacopoeia, you cannot take up the plant for further research. It has to be safe. As I mentioned in my slides, there are so many adverse effects of these scientific, so safety of the plant, the part which you are trying to pursue, should be ensured. And this is a wonderful book of 75 plants, the most important plants for which the safety data has been incorporated. And this safety data you cannot get. It is so damn scattered, and we got it from various pharmacopoeias across the world. And we have, collected this matter from various the APIs, the IPs, the USPs, and all the papers pursuing to these 75 plants, and the safety aspects of these 75 plants has been recorded in this particular monograph. And of course, the ethical guidelines. This is another publication for biomedical research on human participants. What are the guidelines to be followed ethically? So this was another publication which I brought out. And this is for the guidelines for research in herbal medicine. This is another one. So this is our website. So anybody, this is the Medicinal Plant Division website, which is hyperlinked to the ICMR main website. So all these publications, the abridged version of these publications are there on the ICMR's website. So anybody who wants to go and see, have a gist of what is going on in various publications that I mentioned, you can go to this website and have a gist of what is the plant, what are the chemical constituents. Some structures are also there. The entire matter is not there because hard copies are available with us, only a gist. So if you are interested in any particular plant, you can go to the hard copy, and there is an order form also here. You can order, and for whatever books, there is a 40% discount that we are offering for all academicians, researchers, students on all these publications for the ease of them to procure these publications. And so thank you very much. Before I end, I would let me tell all of you that I am currently pursuing a phytopharmaceutical drug in the area of pre-diabetes, and we are now on the human safety studies, that is the phase 1 clinical trials. We roped in industry at the very beginning, and so the industry was able to provide us the entire material because these are huge projects, and we are roped in the major hospitals in the countries for getting patients. But now at the present scenario, we are on the phase 1 safety studies, because unless we prove that the drug is safe, the phase 2 studies cannot be carried out. So therefore, we are now on the verge of this, and I hope that ICMR is able to bring out this PPH, the first one in the country, in the area of pre-diabetes. So thank you very much for this presentation.
Thank you, ma'am, for the excellent talk and information about the plants and this thing. And we'll see if people have some questions. So if you take some questions, we feel very happy. But madam, I have the small inquiry about that one. Do you have any interface between the farmers and industries?
No.
Okay.
That
Because
ICMR is not doing. That the National Medicine Plan Board is doing.
Okay.
National Medicine Plan Board under the Ministry of Ayush, they have a very good system, of connecting farmers to the industry. There is a special program, and you can contact the National Medicine Plan Board for this activity.
Okay.
Ma'am, I just have one question. Sorry.
Yes, please. Yeah.
Have you included mushrooms also in these volumes?
No, these are only higher plants. No fungi.
So suppose, okay. Yeah. So suppose we want to work on mushrooms, then what should be the path and what should be the criteria?
So then again, a bioactive fraction from the mushroom. I have noted, yes, the same procedure. You can do that, and only the 4 bioactive, which should be unique to that particular plant. It should be present in such a quantity that it can be quantified, and the pharmacological activity of that particular plant is due to that particular marker. So mushrooms.
Yeah, scientific validation if we do, and then we can take the certification by producing all that.
You can do that, because this information is, we are doing only the higher plants. We are not including the fungi. Maybe if I get a chance, I'm also very keen to do fungi.
I think some high altitude mushrooms are,
yes, of course,
they are really important. Like Chinese are using you.
Yeah.
So if that can also be included, I will be very happy, because we are working on that. So
that's great.
We have done a lot of work on that.
Yeah, I know. I was in contact with DRD also.
Yeah.
Many years back, I was in contact with Dr. K.K. Srivastava, Dr. Brahm Prakash, and I used to work with those people also, at that time. And they also used to tell me that there are some fungi, with DRD was also working. I was very keen, but the policy of ICMR was as such yet. First, we should finish the plans.
Thank you. It's a real good work.
Yeah.
I mean, I must congratulate you.
Thank
it was
you,
required for a very long time, actually.
Yeah. So this is a kind of an encyclopedic work, for everyone. That's the same.
Very helpful.
Yeah.
Thank you.
Thank you so much.
Madam, in this your work in the, you have the short-term crops also for 3 months, 4 months duration, medicine plant, which can be used
if
for the
that medicinal plants, if there is any that particular medicinal plants, see, we are not touching the agriculture aspects at all. Okay. If that particular plant has some medicinal value, we take up. that particular plant. But we are not into crops because we don't want to mingle up with ICAR and ONN and National Medicine Plant Board and Ministry of Ayush. I am solely responsible for those plants which have some kind of traditional medicinal value. And then we, do, because I am now working on methi, on getting the first PPH from methi. So this is, if it has a medicinal value and it is used in the traditional knowledge, or it is a perspective, even for modern pharmacological studies are there, then we take up those plants.
Ma'am, I have one question.
Yes, please.
Yes, ma'am. Ma'am, can we include the some alga plants? Actually, they have very good
I
medicinal.
know that algae, fungi, yeah, these are our. Let us finish this. I want to reach up to till Z and finish up the higher plants, and then maybe we come to the lower plants also.
Okay, because we can use as well as drug delivery system also. So, and the most important thing, they have very less toxicity, because toxicity is a very big issue with these phyto-based compounds.
So this is Goa is doing, the CSIR Goa, lot of work is being done by the ocean plants, by these people. So,
but for the policy making, can we approach for that which organization for this?
CSIR is working on this, and their Goa Institute, National Institute of Oceanography, is working on this particular these algae, the blue green algae.
Yeah, and the diatoms also actually.
Yeah, of course, you can pick up anything.
Okay, okay. Thank you so much, ma'am. Yes.
Thanks a lot.
Ma'am, what is the, first of all, I would like to congratulate you for a very good presentation and giving an overview of giving global scenario of botanical medicine and giving emphasis on Indian herbal plants, rich diversity. Ma'am, can you please give us an idea about the emerging global opportunity in the
So
Indian...
the Herbal Medicine Compendia, which is brought out by the USP, for which I'm chairing the Southeast Asian regions. So here, anybody, who has some kind of a work, an interested research work, it's all free, You go into the United States Pharmacopoeia website, and there you have a Herbal Medicine Compendia. There you can see, as such, how many monographs have already been published in those herbal medicine compendia. So if you have any kind of a promising plant or any kind of promising research, you can always approach them for further work in that particular area.
We also have one question from, I think, Gaurav Gautam. He can ask the question. Gaurav?
Yeah, good morning, ma'am. Ma'am,
Yes.
I have a question regarding authentication of medicinal plants. Actually, I would like to ask that authentication of medicinal plant, is it necessary to validate a medicinal plant by a botanist, or we can do the validation or authentication by ourselves using the monograph as published in Indian ICMR or Ayurvedic Pharmacopoeia of India?
So anybody can do it. You have to match the results with either IP or the ICMR pharmacopoeias or our quality standards. In a pharmacognosy, the powder microscopy, etc., everything is done by a pharmacognosist only. And then the chemist also comes into place, because we need to have particular standard markers with respect to those standard markers. If the active constituents are not there in the plant, then there is no use of conducting research. You will not get good results at all. See, the authenticity of material is being done by a pharmacognosist, whether it is a genuine drug or whether it's an adulterant or substituent being used as a particular drug. And then the second part comes as a chemist. The chemist needs to make sure that the active constituents of that particular plant are there in that particular material. Otherwise, if you will not get any result, the whole labor goes waste, because the plant does not contain those active materials. Then there are so many things: how you store it, how you dry it, how you harvest it. It's a whole kind of a protocol that has to be followed, for getting good results. And all these, our books, they have a protocol as to where to collect the plant from. What should be the location, pesticides, residue, everything. You take into consideration of so many things before standards are developed. And when you are doing it, you need to have a pharmacognosist, a chemist with you. And then, of course, if you want to pursue it as a drug, then pharmacologist is also required. And then, of course, then you go the clinicians and the hospitals and the patient-based study. Human toxicity, safety studies are also very important. And then, of course, who's going to make the drug for you?
Yeah, actually,
What
ma'am,
area
the
you
drugs. Yeah,
Pardon?
actually, the drugs is not. Actually, I'm a pharmacologist. I've just completed my PhD in pharmacognosy and phytochemistry. But this is my question: that is the botanist is required for validation
Yes,
or
of course.
authentication?
Yes, of course. We do not get pharmacognostics. They've become extinct in the country. I have advertised a post of pharmacognostic for more than 16 times, and I don't get a candidate. Pharmacognosist is the base person for developing standards of any botanical drug.
Exactly. Actually, some of the personal
The
professionals says
macro
that
microscopy study has to be done by
authentication.
a pharmacognosist only.
Exactly, ma'am. That's the point I want to say to other people also, that for authentication or validation of a medicinal plant, it is
Definitely.
not necessary that you will go for the botanist. If a
Mm.
pharmacognosy is there, you can also validate based on the published monographs or Ayurvedic pharmacopoeia ICMR monographs also.
Yes.
But you have to analyze that in a proper way. So
Yes.
that was my last question only, that that was confused, and I was also understand to other people's to that, because they are not able to understand these points as well. So
No, no.
that's why I asked the
Pharmacognosis is
question to you.
absolutely essential for developing the standard setting procedure of any medicinal plant.
Yeah, ma'am. Second question is my that whatever the books you have shown, is this freely available on the ICMR side, or there are
Mm. It
chargeable?
is these are priced publications, and you
Okay.
need to get it from our sales division of the Indian Council of Medical Research. And you can write an inquiry, and we will reply to you as to what is the amount that has to be paid, because we are offering now 40% discount to all the researchers to use it.
Ma'am, is there any institutional concession on these books? As
40%.
I have
40%.
completed my PhD from Jamia Hamdard, from under the guidance of Dr. Saeed Ahmed sir. So, is there any concession on these particular books, or yeah,
If you are
40%?
even if you are a student, we will give you 40%. If you are an institution, we will give you 40%. And Saeed has all books of my of ICMR at Jamia, so they purchase and they keep it in their
Yes, ma'am.
library. Yeah, Vidhu Eri and the Saeed Ahmed, they
Ye-
both purchase these books. These are very valuable books, I'm telling you, because
Yes.
to get the pinpoint references of what exactly has been done. There is no point antioxidant. I was hearing yesterday antioxidant, free radical scavenging activity. Baba, you don't need to do all this. All plants have antioxidant activity. No, you should go now on the target-based drugs. Yesterday, I was going through some of the this thing, antioxidant COX-2, anti-inflammatory OX2. So these are all, all plants would be having all this activity. You search for the unique activities that a plant, the attribution of a plant is to, and then go for those activities. And for the unmet how will you position your drugs in the market? Suppose if you take out a drug and it is
Yes.
costing 50 rupees per tablets. I'm just giving a hypothetical example. And the
Okay.
modern drug is being sold for the same prescription for 20 paisa. Your research is waste.
Yeah, research-based Indian medicine plants.
Everything has to be taken into the consideration when first you develop such kind of drugs. The pricing of the such drugs, your botanicals, whether the market is going to accept it. What area you are positioning this drug for? Anti-inflammatory, antioxidant. Nobody is going to buy a drug for antioxidant. And I have stopped researchers. I now tell them, please stop screening medicinal plants. Stop screening medicinal plants. There is ample screening done by CDRI Lucknow. 3,848 plants have already been screened. Go for those such screenings. And if you don't have basic knowledge as to how to proceed, you should approach first a scientific procedure as to what plant you need to select and what activity and for what condition, where are you going to position, and if you're developing a drug, the pricing, etc.
Okay.
It's not like
Thank you so.
hyperzetted and antioxidant, radical scavenging, antioxidant, anti-inflammatory. This is no way, and you're wasting your funds and research activities also. I think you should stop.
No, please, but the last question is that only. Can I ask that? Actually, as you told that the thrust area of research in not being regarding of this particular research. Actually, I would like to. I just considered that the thrust area of research, that is endometriosis, and the uterine disorders that is mostly occur in the ladies, yeah, females, and nowadays the most abundant in India also.
So search for plants for this area first of all. The plants need to be searched for this area. Number one. So you have to go for our consolidative index, the pharmacological index of these 27 volumes. We are giving activity-wise index. Go to that index and search for your uterine activity or whatever activity you are interested. Shortlist the number of plants. Then go to individual plants and see how much of research data has already been done on those plants. And then you choose a particular plant. And the availability of the plant. See, we have suffered a lot. For Pterocarpus marsupium, the hardwood of the plant is used. Now, since the Biodiversity Act came into position and there was a stoppage of felling these trees in the forest, our most potential drug for diabetes, which was in the phase 2 style, due to the lack of material, we had to stop.
ब्लॉक इनपुट में क्या है हमारे स्कोप में क्या है ये डिजाइन कुछ है क्या स्पेसिफिकेशन है क्या है
If any other no questions, we can thanks, madam, for the excellent talk.
Thank you,
ma'am.
Thank you so much, ma'am, for your answer.
Thanks a lot. Thank you very much.
Thank you. Professor Sachi, you want to say something?
With your permission, sir, I would like to just thank ma'am first, and just to summarize what she has presented today. She has given a global scenario of botanical medicine. She has briefly talked about that how herbal medicine that had been scientifically evaluated, how their potential in medicinal treatment has been evaluated. She has also given an overview of the USA FDA about the separate guiding document for botanicals. She has given about Indian scenario where she has discussed about the classical ASU drugs. She has mentioned about the Ayurveda, Siddha, Unani drugs that include. She has mentioned about the Government of India has published a draft amendment to Drug and Cosmetic Act rule on 24th October 2013, and India is also working on the new draft regulation which were presented in Guglu tablet, etc. She has given about the Schedule Y, the newly added Appendix IV that describe the data to submit along the thing. She has introduced the pharmaceutical drugs and gave the definition of the pharmaceutical drug, how the new regulation of phytopharmaceutical is in line with the regulation in USA, China, and other countries. And she has also given why pharmaceutical, etc. Scope and potential of the pharmaceutical drugs. She has also mentioned about the various GMP requirement, current GMP requirement to manufacture these drugs, and data which is already generated. She has given about the botanical drugs in India like guglic tablet, ginseng, silmarine, etc. And also she has given a brief description about the drugs that has been approved by US FDA. And I would really like to congratulate her and her team for an excellent work on giving the various description about the how Indian Council of Medical Research has given the review on Indian medicinal plant. She has told that they are having more than 1 lakh citations, and the everything related to their pharmacology and other aspect has been mentioned. She has also given a description of quality standards, phytochemical reference standard material, how the quality standard materials can be given. And as now she is working on diabetes mellitus. So, ma'am, I really would like to congratulate you for a very excellent work, for a very excellent presentation, And I can see from the list of participants that more than 217 participants are there, ma'am, student. I am really hopeful that all our students, all our participants, they were immensely benefited by your talk, by your guidelines, and I really agree with you that there are number of drugs, number of medicine has been, number of plants have been isolated and identified. Now the focus is to identify the bioactive compound and how India can take lead in pharmaceutical in global level. So there is a need to commercialization to make a put forward for the market globalization or finding the market for this pharmaceutical company. And I am really hopeful with the these recent scenario where we have seen observed that COVID-19 situation where these all herbal medicine has been played a very good role. With your guidance and with your talk, definitely the scope for this pharmaceutical will be increased. And with this, once again, I thank you for your very good talk and very good presentation, ma'am.
Thank you so much, ma'am, for that very extensive words of appreciation. I am very much obliged. Thank you very much. I thank Dr. Shipra Mishra also for giving me this opportunity to come here and deliver this keynote address. Thank you so much.
Now we hand over to the organizers. Okay.
Okay, sir. Thank you.
Okay.
Thank you so much for chairing the session so nicely, and thank you for enlightening us, enlightening us with the knowledge of your words. And really thankful to Dr. Neeraj ma'am. Actually, she created a very good roadmap for the research of phytochemicals and all. So thank you, ma'am, for your guidance. Now, yes, ma'am. Now I would like to invite Miss Manisha to carry with the ST award ceremony. Thank you, ma'am. Thank you.
Thanks a lot.
Yes, ma'am.
Thank you so much, ma'am. So let us continue our this session with the ST Annual Ceremony Award. I'm just sharing my screen. Screen is visible?
Yes, Manisha.
Okay, ma'am. I'm just starting the... So we are continuing our session with the ST Annual Ceremony Awards, award ceremony, and the first award for today is for the category ST Humanitarian Award for NGO, which is presented to Letters to Strangers, India, Kurla, in recognition of their unparalleled vision in addressing the importance of mental health, especially amongst the youth. Hence, we would like to take this humble opportunity to accredit their sincerest effort as an NGO. We have Ms. Arushi Kataria, Chief Functionary, to receive the award. Ma'am, are you there? Do we have Arushi ma'am with us? Okay. So the next category of award is the ST Innovation Award, which is being presented to Dr. Tapan Kumar Roth, Head Technology Leadership Areas, Tata Steel Limited, Jamshedpur, for his impactful work in the fields of environmental protection and innovation. Congratulations, Tapan sir.
Sorry for interruption, but I think this was given yesterday itself.
Okay, ma'am.
Yeah,
So,
no problem.
The next winner in this category is Professor Suraj.
This also was given.
This one.
ये भी हो गया. I think you have to go down.
Okay.
This we start from this.
So the okay, this one. So the award for the category is for the best ST school ST Best School Principal Award, and the award is presented to Sister Rose Fatima AC, Principal from Principal Caramel Convent High School, MAMC Durgapur, to acknowledge her excellence in inculcating value-based education and inspiring life skills in the young minds of school students. Congratulations, Sister Rose Fatima. Okay. So let's move on to the next category, that is the ST Best Teacher Award, which is being presented to the two highly dynamic teachers. They are playing a very important and stimulating role as science teacher, and they are strengthening our society and future generation. So the award is conferred to Dr. Vaishali Mishra, HOD, Chemistry, ITL Public School, Dwarka, New Delhi. Do we have Vaishali Mishra, ma'am? Okay. So the
Vaishali ma'am is here. Please unmute, ma'am.
Yeah, I'm here.
Vaishali ma'am, congratulations, ma'am.
Yeah, thank you so much. I'm very immensely humbled to receive this award from you. So this certificate sincerely acknowledges my effort, and it is a huge admiration. I'm extremely overjoyed to receive this great honor. I want to give my sincere thanks to Dr. Shipra ma'am, who has supported me and encouraged me to get this award. So thank you so much. And once again, congratulations to other awardees also. Thank you.
Thank you, ma'am. The next winner in the same category, that is ST Best Teacher Award, is presented to Ms. Sumita Chakrabarti, Assistant Teacher Bengali, from Hirendra Leela Patranavis School, Kolkata. Do we have Sumita ma'am with us? Ma'am, are you there?
Actually, she won't be able to come, but she conveyed her thanks, and the message was, I think, shared, which could be shown here. But anyway,
okay,
she is busy with the exam, so she won't be able to.
Ma'am, please convey our congratulations to her.
Okay, thank you.
Okay, thank you, ma'am. So the next category is the ST Young Researcher Award, and it is for the faculty who are working tirelessly in the field of research. So the award is presented to Dr. Rabia Parveen, Assistant Professor, Department of Pharmaceutics, SPER, Jamia Hamdard University, New Delhi. Dr. Rabia, congratulations, ma'am. Ma'am, are you there, Dr. Rabia?
Thank you. Thank you, ma'am. First of all, like of this EWASH and also selecting me for this Young Researcher Award. And thank you, ma'am, and thank you, organizing committee.
Thank you, ma'am. Congratulations. The next winner in the same category, that is ST Young Researcher Award, faculty, is given to Dr. Shushendu Banerjee, Assistant Professor, School of Chemical Technology, KIIT School of Biotechnology, Bhubaneswar. Congratulations, sir.
Thank you very much. It's a great honor for me to receive this special award, and I'm humbly thankful to the team who selected me qualified for this award. Thank you for motivating me to continue having the highest level of dedication to my job. Thank you very much.
Thank you, sir. The next winner is Dr. Gaurav Gautam, Assistant Professor, Department of Pharmacy, IIMT University, Meerut. Congratulations, sir. Can we hear words from you?
Hello. Yeah. Hello, ma'am. Yeah. Thank you so much. Thank you so much, EWASH convener and coordinator, for awarding me this prestigious award for ST. And thank you so much, ST sides. Thank you so much.
Thank you, sir. The next winner is Dr. Ishan Pandya, Assistant Professor, Akash Institute, Ahmedabad, Gujarat. Congratulations, sir. please share some words with us. Dr. Ishan.
Sarun, sir is not here in the list.
Okay. So let's move on to the next winner, and the winner is for the category ST Young Researcher Award, which is being presented to Mr. Satyanand Sinha, Research Scholar, Center for Environment, IIT Guwahati, for his dedicated efforts in contemporary research. Congratulations, Mr. Sinha.
Thank you. Respectable patrons, Professor Madhumjat ji, Professor Sharma ji, Professor Srivastav ji, respected conveners, Dr. Mishra ji, Madam, and Professor Jain, other dignitaries, awardees, and my friends. I'm really overwhelmed and really feel happy to be a part of the ST and EWASH 2022. As we have seen over the past 2 days, we have witnessed the several influential talks and research deliberations. I realized that the excellent role played by the ST in forming a bridge between the academia and society, which is really very appreciable. At this stage, we know that awareness to protect the environment has spread across the country. However, still it is a need to educate the larger section, which is evident as knowingly or unknowingly, we are still polluting the environment every day. So there should be a backup plan, and hence scientists are working hard to develop technologies to protect the environment. However, this gap still exists in translational applications, and I believe this is where the STE has taken the lead role. It is one of that organization which over the years has succeeded to put thoughts into the action and in true sense serving the society. Saying this, I feel very much obliged that STE has found me suitable for this prestigious STE Young Researcher Award. Thank you once again. Jai Hind, Namaste.
Thank you so much. Congratulations to all the winners and best wishes for your future endeavors. So,
Manisha?
Yes, ma'am.
Letters to strangers want to say something. They were not unmuted. So that is why. So now can you go back to the first one if possible?
Wait a minute, ma'am. This one, ma'am? I'm sorry.
The
could
first
you repeat?
one. First one.
First. Okay.
Letters to that NGO, humanitarian.
Okay, ma'am.
Yeah, this one.
Okay. So the award was conferred to the award for the category ST Humanitarian Award for NGO was conferred to Letters to Strangers India, Kurla, West Mumbai. We have we have Ms. Arushi Kataria with us for receiving the accolade.
No, sorry, Arushi is not there. This is
Okay.
her mother receiving it on her behalf because she is.
Congratulations, ma'am.
No, thank you. And I think she definitely wanted to be there. And I definitely take no credit for this because I think it's their passion. There is a team of youngsters from college who set this up almost 3 years ago. And as we speak, their team is almost 70 plus people, and they have helped approximately, I think, 45,000 connections across the country. And I feel humbled to receive it on her behalf, but definitely want to say that they are doing a fantastic job with a very small start, but an area which people speak less about, an area which is still behind the doors, and they are trying to bring it to the front and make it a very, very normal conversation that people discuss about mental well-being, and especially at the young age. And I'm sure we've been reading about the stuff which has been happening in various industries where people need those safe spaces, those comfortable surroundings to battle some of the mental emotions they are going through, and they need that support. So it's a start, And I think I want to congratulate this team, this organization for picking this up and doing a great job there. So thanks once again. It's to Arushi, it's to Naomi, it's to the entire team of L2S, they call themselves, Letter to Strangers in India. So thank you.
Congratulations, ma'am, once again, and special congratulations to Miss Arushi. We wish her all the success, and we wish, since she's working in a on small scale right now, and we wish that she expand her this work and help people throughout the country or throughout the world in a better way. Thank you.
Thank you. Yeah, we will convey that. Thank you so much.
So, before concluding the session, I would like to give a special mention to our sponsors and the Gaur Surgicals. They have given us the unconditional support and a very timely support. But without their effort, the workshop or this conference could not have been successful. So Gaur Surgicals was established in 2014, and since then they have been aiming to offer world-class services and best medical technologies in healthcare. In India, they offer the most recent technology that is helpful to patients and is widely used elsewhere. Because of their dedication, affordable care, and around-the-clock services, Gaur Surgicals have been successful in gaining patients' trust. Their primary areas of interest are the public health and government sectors, and they have also done so many public awareness projects like pneumonia and anemia screening project, cancer and PPH screening in children, CCHD screening in gynae. They, PPH screening in gynae and CCHD screening for children. They have a very hardworking, knowledgeable and experienced team which works very dedicatedly and tirelessly for patient satisfaction. Thank you, Gaur Surgicals, once again. We look forward for more associations with you in future as well. Thank you. Now I hand over the platform to my fellow coordinators, Dr. Richa Tyagi, Dr. Ragvi, and Dr. Neha Chaudhary to continue with the session. Thank you, everyone.
Thank you so much, Manisha. Shipra ma'am, shall we start?
Yes.
Okay.
It was already delayed.
Okay, ma'am.
Was to be.
Okay, ma'am. A very good afternoon to one and all. Now we are going to begin with the session. On behalf of the entire coordinating team, myself, Dr. Ragvi Khattar, Dr. Richa Tyagi, and Dr. Neha Chaudhary, faculty members, Hindu College, welcome Dr. Suraj Kumar Tripathi and Dr. Maulin P. Shah to chair this session. In this particular session, we have great opportunity to listen from distinguished speakers, Dr. Pinaki Dey, Dr. Jayato Nayak, and Dr. Aarti Bhatt. And also we have oral presentations in this particular session. Now, I request Dr. Richa Tyagi from Hindu College to introduce our first chairperson, Dr. Suraj Kumar Tripathi.
Thank you, Dr. Ragvi. Good afternoon, everyone, and a very warm welcome to all. Now, I would like to introduce our first chairperson, Dr. Suraj Kumar Tripathi. Dr. Suraj Kumar Tripathi, Associate Professor and Associate Dean, School of Chemical Technology, holding a joint appointment with School of Biotechnology, Kalinga Institute of Industrial Technology, Bhubaneswar, India. He has done his PhD in Material Science and Engineering from Chonbuk National University, South Korea. His research area includes materials and mineral processing, biomaterials, photocatalytic decontamination of wastewater, heterogeneous catalysis, development of solar photocatalytic process for concurrent decontamination of antibiotics and bacteria from hospital effluents. He has published more than 53 research papers and review articles to his credit and he's also contributed chapters in book titled Core Shell Nanocomposers for Gas Sensing Applications, a handbook of research on diverse applications of nanotechnology in biomedicines, chemistry, and engineering. He is also a member of different societies, namely Powder Metallurgy Association of India, Indian Science Congress Association. He has received several awards and fellowships for his remarkable scientific aptitude, like DST Inspire Faculty Award by Department of Science and CSIR Nehru Fellowship by Government of India, Graduate Research Fellowship by Korea Research Foundation, and many more to his credits. He has 5 Indian and US patents to his credits. He has been consultant and honorary technical advisor for Metis Technologies Private Limited and continues to be a consultant and honorary advisor for Jomali Herbs Private Limited. We welcome you, sir, and thank you for joining us today. Now I request Dr. Raghavi Khattar to introduce our second chairperson, Dr. Maulin P. Shah.
Thank you so much, Richa ma'am. I feel privileged to introduce our second chairperson, Dr. Maulin P. Shah. Dr. Maulin P. Shah is currently working as a Deputy General Manager, Industrial Wastewater Research Lab, Division of Applied and Environmental Microbiology Lab at Enviro Technology Limited, Ankleshwar, Gujarat. He received his PhD in Environmental Microbiology from Sardar Patel University, Vallabh Vidyanagar, Gujarat. He served as an assistant professor at Godhra, Gujarat University in 2001. He is a microbial biotechnologist with diverse research interests. A group of research scholars is working under his guidance on the areas ranging from applied microbiology, environmental biotechnology, bioremediation, and industrial liquid-based management to solid-state fermentation. His primary interest is the environment, the quality of our living resources, and the ways that bacteria can help to manage and degrade toxic waste and restore environmental health. His major work involves isolation, screening, identification, and genetic engineering of high impact of microbes for the degradation of hazardous materials. He has more than 200 research publications in highly reputed national and international journals. He has guided more than 100 postgraduate students in various disciplines of life science. He is an active editorial board member in more than 150 highly reputed journals in the field of environmental and biological sciences. He is also serving as a reviewer in various journals of national and international repute. He was also the founder editor-in-chief of International Journal of Environmental Bioremediation and Biodegradation, as well as Journal of Applied and Environmental Microbiology for the period of 2 years. He is also associated as an associate or editorial board member in 28 journals of Springer, Elsevier, Wiley, IOP, Taylor and Francis. He is acting as editor-in-chief of Biotechnology and Genetic Engineering Reviews journal since last 1 year. He has edited more than 175 books in wastewater microbiology, environmental microbiology, bioremediation, hazardous waste treatment. So I welcome you, sir. Thank you for joining us today.
Thank you so much, madam.
Yeah, now I request our esteemed chairpersons of today's session to kindly introduce the speakers. Over to the chairpersons.
Thank you, ma'am. Thank you for a very kind introduction. So it's the time that we should go ahead with the next speaker, and I think we will get a very great opportunity to listen to Dr. Pinaki De, who is our next speaker. having a huge experience in lignocellulose biorefinery, membrane separation process, process intensification, and microbial fermentation operations. He is a chemical engineer and completed his PhD from National Institute of Technology, Durgapur. And presently, he is a senior scientist in CSIR, National Institute for Interdisciplinary Science and Technology, Tiruvananthapuram. And he has been very active in different areas of environmental chemical engineering, particularly biomass intensification and production of useful products from different waste bioresources. So it's an honor for us to invite Dr. Pinaki De to start his talk. Over to Dr. De. Dr. De, I think you need to unmute yourself. Yeah. I think it's not audible.
Is there?
Yes, ma'am, he's here.
Yes, ma'am, he's there.
Okay.
But we are not able to hear.
Dr. Sankha Chakravarti, please contact him if possible.
Post if we can have him unmuted. Dr. Manoj, if you can have him unmuted. Yeah.
Ma'am, sir is co-host, ma'am. So sir can do that, but sir is not audible. I have also asked him to unmute. It's problem from the sir end.
Maybe some speaker setting is needed from
Yeah.
his side. Yeah. So your speakers are like mic is connected with the.
Sir, you can rejoin, sir, if you want, sir. You are not audible, sir. Maybe there is some problem in the speaker settings. When we join, then there comes the option.
Yes, there is some permission needed for connecting the speakers.
Hi, yes, Dr. Pinaki, if you can check like unplugging it once, your this audio device, and again plug in. Just give a trial. No, it's not audible.
No.
Dr. Pinaki, I suggest you please try rejoining. Maybe it will ask you for some audio settings.
Surajji, in case there is a problem with him, then we can take up the next speaker, I think.
Okay, madam. I think Dr. Joyta Naik is here. Maybe we can move to the next speaker.
Yes.
Okay.
Thank you.
Thank you. So, ladies and gentlemen, sorry for the inconvenience. Maybe that's some technical glitch. So let's move on to our next speaker, Dr. Joyta Naik. Joyta Naik is presently Assistant Professor, Center for Life Sciences in Mahindra University, Hyderabad. He is a chemical engineer by training and got his PhD in chemical engineering from NIT Durgapur, and is a prominent researcher, more than 10 years of experience in the field of design and application for the process intensification. His core expertise includes process intensification, bioprocess engineering, water and wastewater treatment, graphene integrated photocatalyst development, and biosynthesis of valuable products. Currently, he is running multiple research collaborations with various institutes in India and abroad. And he has published over 25 in very good journal papers and more than 20 book chapters, and has 3 patents to his credit. And he is very active in the area of biomass valorization for industrial application. And I believe we will be getting a very new insight to his research and how it can be used for industrial application. So over to you, Dr. Joyta.
Yes, Dr. Tripathi, it's a pleasure to hear such good words about me from your mouth. So anyway, am I audible and visible?
Yeah, you are audible.
Okay. So I would like to thank Dr. Tripathi, Dr. Maulin Shah, Dr. Shipra Mishra for giving me this opportunity to present near you regarding this EWASH conference. And as Tripathi had already mentioned regarding my expertise, so I would like to directly share my screen and go for the presentation. So is it visible to everyone? I'll put in the full screen mode.
Yes, it is visible.
Yes, sir.
Okay, thank you. So today my presentation, just a minute. Yeah, today my presentation, this invited talk will be on the graphene integrated photocatalyst development for the transformation of anthropogenic CO2 to biomethanol. And as our chairperson has already mentioned, that I am working now in the Center for Life Sciences in Mahindra University, Hyderabad. So now, before starting off any project topic or anything, we need to know the basic demand of any material or any chemical that we are we are proceeding for the production of it. So primarily the traditional uses of this methanol used to be for the production of different chemicals like acetic acid. And I should mention that more than 90% of acetic acid, though it's organic acid, more than 90% of it is produced till now based in the chemical routes, which is called as methanol carbonylation. And for that thing, methanol is a specific chemical that is extremely required in a high volume. And also formaldehyde, silicon, methanol, some kind of niche application also. This methanol is highly recommended and required chemical. So it's basically a platform chemical also, we can say. And as the trend of the energy that is shifting from highly it is shifting from the fossil fuel demand to different biodiesel production, fuel blending, and all these things. So this alternative fuel production is also now getting highly dependent on this methanol. So in case of biodiesel, which is basically now also it is obtained from different chemical resources, which is basically a methyl ethyl ester. Though it's named as biodiesel, the main component, the main constituent of it is methanol, through which it is being produced. And now the fuel blending is also going all throughout the world, because the fossil fuel demand, and it's having the fixed stock of fossil fuel all throughout the world, and it's exhaustible. It's not going to perpetually favor us throughout the centuries or throughout the next few millenniums. So now the fuel blending is one process that is also going throughout the world, and till now we have received about 10% of fuel blending with methanol, with our conventional fossil fuel, that is petroleum. So in case of fuel blending, it's taking an important part: dimethyl ether production, marine fuel production, and it's not completely dependent on methanol. But what happens that this fuel blending is going on so that the demand of the fossil fuel could be reduced, the exploitation of the fossil fuel could be reduced, and different countries can get more profit, because methanol is still a cheap resource of fuel. If we see the overall demand and the use of this kind of methanol, we can see that by region, China is the biggest consumer of methanol, and Asia Pacific region also. Like if we mix up this full Asia region, so it will come about 64%. And by derivative, formaldehyde, acetic acid production that we have already mentioned, fuel blending is going like 12% all over the world. As per the data of market data of 2022, about 111 million metric ton of methanol was produced. And also what happens that, and we need to just know that what is the requirement of it. I mean, why this is so much produced, I mean, so much used as fuel blending or biodiesel, because this methanol is having high calorific value, not as much high as hydrogen fuel or like the petroleum fuel, but it's having moderate level of calorific value, which stands about 22 megajoule per kg. So if 1 kg of methanol is being burnt, that creates about 22 megajoule of energy. So this is why this methanol has caught the attraction of the researchers all throughout the world as an alternative fuel. And due to the growing exponential demand of fuels, because every time we need to charge our mobile phone, we need to charge, we need to do any kind of household commodity, we need to use the electricity. Now, from where the electricity will come? So that's the biggest question now the whole world is facing. And the researchers, they are now, maybe it's scientific or technical or engineering researchers, all are now much more concerned about the production of alternative fuels. So methanol can work as an excellent alternative fuel in case of all energy demands. Maybe not all energy demands, but maybe it's meeting up the demand of energy in all the cases. So generally, if we see a conventional or typical production process of methanol, it starts from the natural gas. So natural gas, if we do, I mean, this is the the schematic diagram of methanol production from natural gas. What happens that we need to go for a steam reforming process, which is which requires a temperature of about 900 degrees centigrade temperature. So natural gas, and that is basically CNG, and steam and oxygen. If we mix it and we go for a steam reforming process, it gives us the syngas. Now, syngas is basically a mixture of carbon monoxide, carbon dioxide, and hydrogen. Now, after that, by vapor compression and cooling of it, and if we go for the zeolite-based catalyst, that is Cu, ZNO, or Al2O3, this kind of catalyst, if we use, we can produce methanol, and along with that, some amount of hydrogen, water molecules or water vapor is also stays in a mixed condition, and through the distillation we can get the chemical grade of pure methanol. Now, if we see this overall schematic diagram, we first, the attention of the researchers come to the steam reforming unit, which requires a temperature of 900 degrees centigrade, and the compression and the cooling, these kind of operations, they also consume a lot of energy. Because what happens that whenever there is a phase changing phenomena, and also in case of distillation is a separation process, but it needs high amount of temperature and high amount of pressure, because it's not easy, it's not that much of easy to separate methanol from water, because it starts to form azeotrophs. So that's why what happens that these are the typical scenario where a lot of rate processes, that means a lot of temperature driven processes and pressure driven processes used to be involved in case of conventional production. So now we want to shift from this like the typical conventional production to some kind of a production process which is eco-friendly, which is not using that much of high temperature or pressure, and it's being done in a kind of mesophilic atmosphere, not very maybe in some cases high temperature is required, but not that much of high, like 900 degree or more than 500 degree. We do not need that much of temperature. We will not involve that much of temperature. So we need to go for some kind of biomimicking processes. Like what happens if we see the overall nature, what happens, there are a lot of biomimic processes that is going on, which is helping the human effort to make it more simpler. Like if we see a helicopter, so there are like biomimic of a grasshopper, we may think. Or if we see an aeroplane which is flying, that is having a biomimic technology of a bird. So we need to find some kind of biomimic processes, like generally what happens in case of carbon dioxide production. Now, year by year, the carbon dioxide production is going high till now. So what is the natural consuming source of it? It used to be generally photosynthesis. So what happens in case of photosynthesis? The green plants, they're having some kind of green catalyst, which is basically chlorophyll. And the green plants, they're converting that carbon dioxide into their useful resources, which is basically glucose, based on the catalytic activity, photocatalytic activity. So if we can develop some kind of photocatalyst which is converting that carbon dioxide to some kind of value-added resources, that we need to go for, and that is being obtained if some kind of like photocatalytic activity could be reformed, which is converting carbon dioxide and some amount of hydrogen, which is naturally available, that is required in that, and we are forming methanol. Now, so our scheme of the process, we have designed in such a way so that in a process, one photocatalyst, it will be developed and it will be having some kind of dopant, which is basically a doping agent, which will make the process ease for the transformation of electrons, and we may need to have some kind of catalytic support, so that inside the solution, that catalyst is working with a higher stability. So this is the overall mechanism of CO2 reduction through the photocatalyst. So what is happening that inside the photocatalyst, so now if I tell you, like we have used TiO2 there, that is the titanium oxide. What happens in case of titanium oxide or any kind of photocatalyst? Titanium oxide, zinc oxide, these are photocatalytic, these are having photocatalytic activity. photocatalyst. So what happens there? There are the two types of bands are there. One is called valence band, another is called conduction band. Now, when the sunlight or the visible light or the UV light that is falling over the conduction band electrons, they used to get shifted towards the valence band electrons, they are getting shifted to the conduction band. Now, in case of TiO2, what happens? Titanium dioxide, this difference between the valence band and the conduction band, that is a bit high, which is about 3.2 millivolt. And in case of dopant, so what we have to do to reduce that amount of energy and to make the electrons more activated, we are using some kind of doping agent. This kind of doping agent could be like gold or copper. Now, gold is of course costly. Silver could be there, silver nanoparticles. So what we can do, we can use some kind of lower cost doping agent, which could be like copper. Now, copper is not easily available, so we can use copper sulfate. And there are some kind of catalyst support is also required, which we have formed using reduced graphene oxide, which can also be used with like activated charcoal could be also one very good catalyst support. But here we have taken our consideration towards the production of RGO, that is reduced graphene oxide. Now, what happens in case of reduced graphene oxide? That is actually a material which is having very high stability, and it's not that much brittle like disactivated carbons. So we have done a lot of literature reviews, and what we have seen that if the TiO2 loaded copper ferrite is used, then the conversion of CO2 to methanol used to be very much high, which is like 651 mu mole per gram of catalyst. Now, in case of TiO2, in every cases we can see that it's giving the highest output, it's giving the highest yield. So what we have to develop, our development goal is to find some kind of catalytic activity agent which can convert methanol, which can convert carbon dioxide to methanol with a high productivity, high yield. And also we need to find some material like what I have mentioned regarding RGO, that is reduced graphene oxide. It is giving us high activity and high surface area. Now we need to know that what is graphene. Now, graphene is basically a 2D structure of carbon. It's an allotrope of carbon. It's coming from graphite, and for the development of it in 2010, two scientists from Russia, they have received Nobel Prize also for the 2D structure development. And it could be folded and it could be molded to any kind of shape. And it's basically having some kind of 2D structure. It's not like a very stubborn structure. It could be molded, it could be folded to any kind of form. We can form the nanotubes, we can form Fullerene structure and everything. And it's having the high mechanical strength. So you can see in the slide I have given it, like one single sheet of graphene can cover the whole football field with weighting under 1 gram. So it's having that much of high surface area, and the tensile strength and the the structural strength used to be, compressive strength used to be very high for the graphene oxide. So nowadays graphene oxide is being used for different device manufacturing, mobile screen manufacturing, and all these things. So now we have designed our experimental investigations based on this scheme. First, we had to synthesize the GO, that is the graphene oxide. Then we have to form the reduced graphene oxide, and third one is the doping of titanium and copper inside that RGO, that is reduced graphene oxide. Now, what is the difference between this graphene oxide and reduced graphene oxide? Why we are preparing it? Because GO, that is the graphene oxide, that is having that is basically oxide of graphene. Now when it is forming RGO, that means we are reducing it with some kind of hydrogen related material, hydrogen assisted material. Previously, what used to happen that generally for this formation of RGO, that is the reducing graphene oxide, N2H2, that is hydrogen, that used to be generally exploited, but this hydrogen is carcinogenic. So what we have done, we have directly passed hydrogen gas over the hot surface of graphene oxide. So what happened that this RGO is getting formed. This RGO is basically graphene, that is the reduced graphene oxide is basically graphene. Now, in case of the chemicals used in the process, different types of sol gel treatments are being used, different solutions are being given there, conditions are given, like the 500 rpm we have to heat it, we have to mix it, stir it, and after that we need to keep the temperature at 200 degrees. That means we are reducing the temperature. And lastly, at the time of synthesis of RGO TiO to Cu, that is the whole catalyst, that time we have to provide the sonication time for 2 hours, settling time of 24 hours, and we have to dry it, and lastly we have to do the heat treatment. Now this last one, that is the synthesis of RGO-TiCu, that catalyst development, that means the graphene based titanium oxide and copper synthesis, it's having the heat treatment, because what happens that in that case the heat treatment is being necessary. Now, why this heat treatment is being necessary? Because it used to give the amount of potential energy for carrying out the reaction. To carry out the reaction, the catalyst that needs a high amount of energy so that it can dissipate the energy in form of reaction. So now this is the overall process and the scheme of this hydrogen exfoliated graphene synthesis. What happened that there is a quartz type of reactor, and there are 2 gas chambers. One is argon gas and the hydrogen gas chamber. Now, after the production of graphene oxide, what we are doing it, we are keeping it inside the muffle furnace, which is basically a quartz type of muffle furnace reactor. So now, first we need to take out all the gases which is present inside, and we need to fill it up with the inert gas, which is basically the argon gas that we are keeping. And then we are passing the hydrogen gas over the graphene oxide, the graphene oxide volume. So now what is happening that the temperature, we need to keep it at 200 degrees, and time it is very much less, 2 to 3 minutes. And we are seeing that the volume of the graphene oxide after the reaction with hydrogen, that is increasing. It's about 200%. That means 2-fold it is increasing. So now the thing is, for any kind of catalyst development, we need to go for this kind of processing where we are getting higher surface area, because specifically for the catalyst development, the surface area development is of maximum of importance, because the more surface area, the more used to be the catalytic activity. Now, during the production of this RGO-TiCu synthesis process, what we are doing, that first that section I have already told, that in the quartz reactor muffle furnace, we're keeping it, and lastly, and we are getting the hydrogen exfoliated graphene, that is HEG, that is the this kind of like blackish. And after that, we are going for the synthesis of catalyst, where we are mixing the Cu salt, where we have used, in this case, we have used the Cu salt, that is copper sulfate we have used, and TiO2 we are mixing, and there is a sol gel process that we are going through. And there we are having the sonication time, and heating time is there for 2 hours at 500 degrees centigrade, and after cooling we are getting this catalyst, which is at the rightmost corner. So then we have used one Pyrex reactor, that is a specific type of borosilicate glass with which we have made this Pyrex reactor, which was fabricated by the company and ours. So in that Pyrex reactor, what we have done, we made some kind of engineering innovations, and the engineering innovation involves the baffling and the starting process, where inside the reactor, if we are using the UV light, there is equal amount of irradiation of the UV light throughout the reactor, throughout the volume of the reactor, so that wherever there is a catalyst, the catalyst is still being active. And generally what happens in case of this kind of reactors, generally the light source that we generally use, that used to be kept from the bottom to top. But what happens, catalyst, due to its own weight, it starts to fall down. But we have managed to keep this light source from top, generally the light source used to be from top to bottom, but we have kept the light source from bottom to top. So that what is happening is, so if the catalyst is also falling, it's still in the active phase, and also the starter is also there, that's also working. So inside the solution, we are getting a homogeneous solution. So the reaction mixture or the reaction broth, that is completely homogeneous, that we are having. And also what is happening, that inside the reactor, we are getting the high catalytic activity for the transformation of carbon dioxide. And these are the two cylinders of carbon dioxide that is being supplied. And inside the reactor, we are having completely carbon dioxide filled atmosphere, which is getting converted to methanol. So this is the overall mechanism that I already told you previously, and this is the thing which is happening for exactly in this case of graphene loaded CU and TiO2 loaded catalyst. So in case of that, what is happening, the light source is coming, the valence band electrons of the TiO2, that is titanium dioxide, that is getting transferred to conduction band, and also what is happening, the electrons are getting released. Now there is one also specific thing, that we are adding the dopant, that is the copper source. So what is happening, that when the copper source is getting loaded on the TiO2, the surface area of TiO2, that is decreasing. So this is one disadvantage. So graphene oxide is taking care in that part also, that if also the TiO2 load is, sorry, Cu loaded TiO2 is having reduced area of activation energy, but the graphene sheet which is present there, 2D material, so that is giving more surface area, and through the graphene sheet, what is happening, the electron transfer is becoming much more higher. So this is the overall thing, due to which we are getting higher production of carbon dioxide to methanol transformation. So if we see the results, the first one is the exertive pattern of the HEG, and the next one is the HEG TiO2 CuSO4, that combined catalyst. So we can see that the peaks which is running from 20 degree to 30 degree, that is actually giving the incorporation of HEG inside the catalyst, and also the 2 theta values which we have obtained, that is at 25.3, 38.6 and 48.7, that is corresponding to TiO2 crystal planes. And also at 42 degree, 2 theta value of 42 degree at 1B, that is giving the copper, copper as copper sulfate, that is present as the catalyst, that is the presence of copper that is denoting. And the next one is the Raman spectra, and here what is happening, that at 2 peaks D and G, that is 1598 centimeter inverse. So that is giving us the sp2 hybridization, it is giving us the notion towards the sp2 hybridization of the carbon lattice and the distorted hexagonal carbon array. Next, it is coming as a structural analysis, which is basically first one is the same, and the next one is the EDX pattern. So if we see the same image, first one that is only for HEG, that is hydrogen exfoliated graphene, and the second one is for the overall photocatalyst. So first one, if we see there, we can see that there are, the structure is not having that much of volume. And figure 3b, if we see that there we can get a lot of wrinkles inside the surface of it. So that means when the catalyst is getting formed over the surface of HEG, that time it is giving us the more higher amount of area, first thing, and the more amount of wrinkles over the surface, which is basically required for a catalyst synthesis. So the AFM analysis that was also done, and this gives us the the overall energy and the force areas over the surface of it, over the surface of the catalyst. And if we see the second picture, that gives us that it's more having a lot of protruding surfaces that is coming up. So it's not it's not that much even, it's more uneven, it's more like a carpet type of region, which is giving us more amount of surface, active surface area. So lastly, what we have done, we have gone for a small optimization process based on RSM, that is response surface methodology. We have taken 5 parameters. First one is the graphene dose, second one is the catalyst dose, third one is the CO2 flow rate, fourth one is the time, and last one is the pH. Now, time we have also taken because residence time, that is the RTD, that is the residence time determination, that is also one big factor for this kind of catalysis. That means how much time the catalyst should be in contact with the substrate. Of course, the substrate is CO2, which is being converted to methanol production. And lastly, of course, the response of this overall system is the carbon dioxide, conversion of carbon dioxide to methanol in gram per catalyst. So this is the CCD, that is the central composite design, which we have obtained from RSM, that is the response surface methodology. We have done it in the design expert software. And these are the graphs. So if we see the overall graph, one fixed pattern, we can see that if the catalyst dose or the graphene dose is on a lesser side, what will happen that the production is also becoming hampered. That means the observed methanol yield, which is at the y-axis, we can see that in the y-axis we are having observed methanol yield that is becoming lesser. So it is going up with the increase of all the parameters, and lastly it is again coming down. So at the middle of it, it's reaching some point where we are having the maximum amount of product yield or the productivity of the system. So lastly, through RSM, with model validation and lower p value, that is the lower probability value, and lower f value, what we are getting, that is the graphene dose if we are using 30%, catalyst dose if we are using 10 gram per liter, CO2 flow rate if we are using 3 liter per minute, and time if we are providing 90 minutes at a pH of 3. Maximum yield that we are getting from the system is 40 milligram per gram, and maximum concentration that we are getting is 400 mg per liter. So based on that, our maximum productivity, which is milligram per liter per hour. So this term is also a bit of high importance as the productivity, because at the last what required is the output from the reactor. So productivity actually gives us the idea regarding the output from the reactor, how much fast we are getting the output from the reactor. So that is coming as 134. So so as a overall, if we see, let us come to the conclusion section, that based on the results, what we have obtained, that is the TiO2 that is copper doped TiO2 particles that we have doped inside the graphene, that increase the photocatalytic activity to a very high manner. And TiO2 is a successful photocatalyst which could be used for the production of photocatalytic conversion of carbon dioxide to methanol. And pH, temperature, starting time, these are all required. But one thing I must show at the last, that the optimum condition pH is 3. That means this process is utilized in a very significant manner if we are using the acidic pH. So, and the last one is the high amount of HEG, of course, provide the high surface area so that the full study has become successful. And based on that, the methanol yield is also getting high up. So this is the thing, these are the references that we have used, and thanks for your kind attention. So I hope
Yeah, Dr. Naik, it was a very very nice presentation and a very elaborate one. So
yes,
maybe
I hope I have not crossed the time that much.
No, it's okay. So maybe we can take one or two questions. Anybody from the audience side would like to ask any question to Dr. Naik?
Dr. Nayak?
Yes.
Okay, so this is really a very good presentation.
Oh, nice. Your sound system is working now properly.
Yeah, yeah. So
Yes,
whether
yes.
camera is working properly?
No, camera is not working, man.
Camera is not
Camera
working.
is turned off, but your sound is coming properly and we can hear you.
Okay. Again. Okay. Anyhow, Dr. Nayak, just like to ask you one question. Like, what is the commercial status of that particular process? Can you please highlight on that?
Yes, actually what happened that commercial process that is still now using the syngas reforming, still now this kind of process is in developmental phase only, and we have also worked in a small reactor. It's not yet scaled up, and one disadvantage I may say, or demerit, this process is new. So what happened that till now scale up confidence is less. Also, we have done some kind of economic evaluation, though it is fitting, but the concentration, lastly, what we are getting, that is lesser. I must say that it's not that much high. Though the yield and the productivity that we have received from the previous studies, that is much better. But still, the concentration, what we are getting, like if you now tell me that, can you give me 1 kg of methanol to me? I will say no. Now itself. So it needs a lot of developments in that process, and slowly maybe in the next few years we may reach towards it, but not yet. This kind of process has not been yet been commercialized, but yes, we have received the patent of it, and we are on the on the path of, on the avenue of making it a successful maybe commercialized afterwards. But first, for that we need to have a lot of confidence for the repeatability of the study also. So repeatability, scale up confidence, and the economic perspective, these are the 3 things we need to tally a lot, and after that we may think for the commercialization of it. So I hope that summarizes.
I think your answer has satisfied Dr. De. So since we are running late, maybe we can go ahead. We can go ahead with the next presenter, and maybe at the end we can keep the floor open for discussion.
Open
the
discussion.
last number of participants.
Okay, thank you, Dr. Tripathi. Thanks a lot.
Thank you. So maybe, ma'am, we can go ahead with Dr. Pinaki now, because I think his sound system is perfectly all right.
Yes, sure.
Yeah. So
Thank you, Dr. Tripathi. I'm just again, there is a little bit problem in terms of screen, so I'm just sharing my screen. Please let me know whether the screen is visible to all or not.
Sure.
Is it visible?
It's visible to me. I
Yes,
think
it's visible to all.
Yeah.
So, good afternoon to all of you. First of all, thank you very much to the organizing committee member of EWASH 2022, specifically Hindu College University Delhi, then Save the Environment, co-organizing committee members, CSR NIDI, Royal Society of Chemistry, London, North Indian section, and specifically Dr. Shankar Chakraborty for giving me this opportunity to share my viewpoint in this regard. Now, I'm extremely sorry for this technical glitch, what has happened. I like to, since we are running a little late, so we just to, quickly summarize my presentation. And here, my presentation, the topic which I have selected for my presentation, that is recovery, recycle, reuse based green biorefinery strategies for a sustainable and secure future. So it is highly relevant to the conference theme, that is environment, water, agriculture, sustainability, and health. And here I am not going to represent any specific result, experimental result from here. I'm just going to represent kind of an overall strategy. Specifically, we know that greenhouse gas emission has been drastically increased presently, and one of the main reason for this greenhouse gas emission is the industrial sector. So how we can alternatively use some alternative approaches, biochemical approaches to reduce that greenhouse gas emission. So in that way, I just like to discuss here. And so first I like to highlight that what is the present condition. Now, human emission of carbon dioxide and other greenhouse gases are the primary driver of climate change. And we know that global carbon dioxide emission from the fossil fuels and industry has been drastically increased from 2000 onwards. And 2019 it reached around a record high of 36.7 billion metric tons. Although in 2020, due to the pandemic, there was a 5% of reduction from this original scale, but still it is going on, and the gases are mainly released during the combustion of fossil fuels, just like coal, oil, and natural gas, and it is during the production of electricity. So what are the basic emission sectors which are responsible for this emission? That is transportation, industry, agriculture, land use, and forestry. And among that, it was estimated like industrial sector actually contribute a large amount of greenhouse gas emission, and it was estimated around 22% of the greenhouse gas emission is being from the industrial sector. Industrial activities just like manufacturing, refining, food production, that are mainly responsible for this particular greenhouse gas emission. Now, if you are seeing the present condition, present emerging condition, just like India is going to see largest increase in the energy demand through 2030, and India's energy demand is annually increasing 3% from 2021 onwards. And India still managed to get importing more than 80% of the oil, 50% of the gas demand through inputs. And the current country is almost spending around $145 billion in 2021-22 for importing crude oil and refined products. Now, as a result, what will happen? Based on the Paris Agreement, keeping 1.5 degrees Celsius in the mind, still rich nations still they didn't agree to a particular conclusion that to shut down or phase down all the fossil fuel based productions. As a consequence, the production of fossil fuels has reached to a height of $64 billion in 2021. As a consequence, what we have observed that there is an increase in the temperature of 1.1 degrees Celsius above the pre-stated industrial level, and it comes up with increased death, heat-related deaths of 68% from 2017 to 2021 in this time period. So this is actually human-induced climate change, and it comes up with It largely affected the wildlife population, water resources, agriculture, natural ecosystem, and the health and food chain. Now, what is the solution of that particular problem? What is the solution? So already in COP27 meeting, our honorable Prime Minister already highlighted that this as a solution, that is inclusion of sustainable lifestyle. So what is that inclusion of sustainable lifestyle? There is a three mantras of that. One is sustainable pattern of consumption, sustainable pattern of production, and environment friendly lifestyle. So these are the things. And in this particular presentation, I am mainly going to highlight on the sustainable patterns of production, because sustainable pattern of consumption and environmental lifestyles are almost correlated with that particular point. Now, when you are talking about the sustainable pattern of production, there's a three main approaches are there. One is recovery, reuse, and recycle. So this, with the three specific strategies, we can we can substantially reduce the, and adopting biochemical engineering approaches, we can substantially reduce the greenhouse gas emission. Now, so basically there is two ways of solution. One is sustainable production, sustainable production through biochemical engineering strategies or biochemical production route, as well as sustainable production patterns of chemicals adapting green technology approaches. So it has been seen that there are several products which can comes out from the petrochemical based routes that can be produced through biochemical engineering, biochemical processing routes, and and it is highly advantageous in terms of environmental prospect. Like biochemical conversion routes mainly involves the application of the enzyme and microorganism. As a result, it normally carried out in a environmental friendly condition with a moderate temperature and pH, and the process is also considered clean. So although however there are some limitations are there, just like pre-processing strategies, that is pre-treatment, processing times, less selectivity, downstream processing is another major concern, but still biochemical engineering processes comes up with a better approaches. And another is sustainable production pattern while adopting the green technology approaches. Like I'm just like to give one example, just like a membrane technology, which has been rapidly used in the different industrial sector, just like a pharmacy, medicine, food, and all those areas. Now I just like to show you a that there are large array of the products, large array of the chemical products, just platform chemical, which can be produced from the fossil fuel based resources. That can be also produced from the biochemical production route while applying the different microorganisms. So there are a lot of here you can in terms of color coding, coded factor, you can see the C2, C3, C4. There is a different color, and you can also see the same color in the biochemical engineering route, where applying a suitable microorganism and suitable metabolic pathway, we can able to produce such type of products specifically here, with the help of microbial synthesis. Now, and I just like to highlight a little bit on this, biochemical pattern of production. So here, if we will see that, if we'll start from the point number 12, the basic pathway involves like, selection of the, microbial culture, which can be capable to produce that particular product, and then finally producing it in the batch level, in a lab level fermenter or bioreactor, and then finally before going to the industrial scale, it should be pre-validated in the pilot plant level, and then finally product recovery and packaging and all. And if the product is very specific, like we need to produce some kind of a animal protein or plant-based protein, or in some vaccines or something like that, we can able to isolate the gene for that particular product, and we can clone it in a suitable plasmid, and then recombinant plasmid can be developed, and it can be inserted to a microorganism, and then finally that microorganism can be used as the inoculum for that particular product. And if you are just giving an overview of this all this biochemical process for a production of a product, there are the few components are there, like inoculum development. So we need to, as I have already mentioned, we need to specifically identify the particular inoculum for that production, And if it is the before going to the large scale fermenter like 100 liter or 200 liter, we need to make the inoculum of 10% inoculum, that is almost 10 liter to 20 liter, like that. And for that, we also need to use some kind of a bioreactor fermenter. Similarly, we also need to, sterilize we need to develop the medium, we need to sterilize the medium. And, and finally, up to the production of that particular component, it will be considered on the upstream processing. Finally, it will comes up with the downstream processing where we are looking towards the separation of that particular product. So there are a lot of strategies can be used, just like, extraction, centrifugation, filtration, distillation, adsorption, large array of the different processes can be used. And if the product is intercellular, then we can need to separate the biomass, and we can we can digest the biomass for the further separation. So now I have my title suggests the recovery, reuse, and recycle. So we can use all those strategies in all those unit operations, and to make the process much more eco-friendly, as well as much more efficient, to reduce the cost. We can apply all those strategies. Now I just give you just a brief idea. Just like suppose for inoculum development, we can normally in batch process, in the inoculum, after inoculum development, it can be used. But inoculum development is also a time consuming process, as well as cost. It's involve a lot of cost. So what you can do is we can perform in a repeated batch condition, where inoculum will be generated in the first condition, and that can be transferred to a next batch or fed batch way. And there is another strategy that is like continuous mode, like chemostat system. So when the inoculum will be produced, it will be continuously provided to another bioreactor, and it can be in a multiple bioreactor system, where the inoculum which is initially produced, it will be further transported to another reactor and further goes on. So like that it can be done. Now next example I just like to give you, just like recovery and reutilization of the chemical reagent or solvent during pre-treatment of biomass. So normally I'm just like to give you a specific example, just like for this second generation biofuel production, we are normally using lignocellulosic material, and we are using conventional acid alkaline treatment, which comes up with a lot of heat generation. So instead of that, we can use some kind of other materials, just like we can use some kind of a primary alcohols with a low boiling point, and that particular alcohol, after using we can able to separate and reuse the material. Similarly, other solvents, just like acetic acid, formic acid, acetone, that can be with a even high, the solvent with a higher boiling point can be used for better separation of or segregation of this component, constitutional component, just like cellulose, lignin, and hemicellulose. Now there are other examples are also there, just like we can, if we want to separate the cell biomass, cells as a product, so we can able to use some kind of a membrane bioreactor for the cell enrichment technique. Normally for such kind of processes, we are normally going through batch wise for the production of the cells, and then making it in a concentrated level through different techniques. But here we can use a single bioreactor, that is membrane based bioreactor, and we can effectively separate the biomasses, and it can be recycled back, and the cell enrichment can be possible with the further addition of the media. Now there are a lot of applications are there, such kind of thing, when the cells can be used as directly as a product, just like lactobacillus strains, and similarly like we can say the yeast, baker's yeast. So such kind of production of such kind of materials, we can use such kind of systems. Similarly, intercellular products, suppose the product is intercellular, just like PHB or PHA kind of thing, we can use such kind of systems. Similarly, if we are dealing with the production of some kind of a component which is self-inhibitory for its production, just like carbolic acid, like lactic acid or citric acid or acetic acid through biochemical route, we can in the permeate side, we can effectively separate such kind of components as it is produced to reduce its inhibitory effect, and the cell microorganism can be recycled back, and with the addition of the media, the cells can be enriched. So another example is like membrane-based technology for the green recovery of the substrates. Normally in the fermentation process, what we are doing, we are using a substrate in a batch process. At the end, there may be some amount of substrate will be remain. So we are not considering about that particular remaining substrate, and that also contributes in the cost. So if we adopt some kind of membrane-based technology, where here you can see, I just like to give you some few details about it. It is a two-stage membrane technology where microfiltration followed by the nanofiltration. The microfiltration, we are basically separating the cells, and in the permeate we are getting the product, and as well as the unused substrate, and which it is when it is coming to the nanofiltration, we can able to separate the product in the permeate side, whereas unused sugars can be recycled back to the fermenter for the, again, it can contribute in the production. So such kind of a strategy actually are required presently, to make the process much more efficient, and in order to cope up with the economical standpoint. Now, another strategy is a very important strategy, that is membrane-based strategy for the recovery, recirculation, and reutilization of the enzyme. Now, it's having a higher application in the second generation biofuel production, where we are using the enzyme for specific production of the sugars, which will be further fermented to another product. Now, again, in that particular process, the simple sugars which will be comes out from the enzymatic process, it is having a kind of inhibitory effect. So if we continuously separate that particular product, or that is hydrolyzed with the sugar solution with the membrane processes, so we can effectively overcome the product inhibition effect, and we can constantly recirculate the enzyme over there for the further production, because enzyme is nothing but the biocatalyst, and we can able to reuse the enzyme much more time. So there is alternative approaches are also there, just like immobilizing the enzyme, but definitely we can reuse the enzyme multiple cycles or multiple times. But obviously, after as the process will be goes on with the cycles, the enzyme activity will be further reduced, but it will not going to happen for such kind of membrane-based reactor system. Now, another application is just like developing carbohydrate-based oligomers, which is having a huge importance. So recovery of the carbohydrate-based oligomers, like we are using a lot of lignocellulosic material for the different applications, but finally we are comes out with simple sugars or reducing sugars with the enzyme application. But if we are specifically apply the enzymatic system with a specific enzyme configuration, then we can able to get such kind of a lignocellulosic oligomers, specifically cellular oligomers, xylo oligomers. Even if we can apply such kind of techniques, for such kind of citrus food products to separate the pectin-based oligomers. So, and such kind of oligomers having a huge importance in terms of prebiotic. So it will largely effective for the growth of the probiotic bacteria. So it is largely useful for the human beings as well as other animals. Now, another strategy is like there are a lot of since we are discussing about the recovery of the important materials. So there are a lot of food production company and there are waste streams having a lot of value-added products, just like it is enriched with waste streams are normally enriched with carbohydrate materials, polyphenols, and we can effectively separate, and you can effectively recover such kind of a valuable materials, carbohydrate material. Just like I'm just want to give you an example, just like in initial days there was a process called rating process, in that coconut husk is normally been deposited in the ponds for around 1 year of time and 6 to 1 years of time, and then that process, after that what is normally done is like this process is useful for the separation of the coconut pith as well as the coconut fiber. Normally it has been done in the Kerala region like that, but it is in the 90s. But after that the process and during that process what it was noticed, like water has been highly enriched with the carbohydrate like pectin and polyphenols, and we can use such kind of membrane-based technology for the separation of the such important polyphenols as well as pectin materials. But now the process has been changed since it is having an environmental impact. So this rating process has been changed and modified. Further, like we can also separate the nanocellulose, which is of a higher importance nowadays. Nanocellulose is being used for different applications, specifically separation of the different recovery of the different materials, separation of the different microorganisms, viruses, toxic materials, as well as the carbon dioxide capturing also. So this nanocellulose is the integral part of the cellulose actually, and we can apply different strategies like pre-treatment production strategies, we need to optimize the process in order to reduce the production cost, and we can able to get the such kind of nanocellulose for the different applications. And finally, different types of biopolymers, integral biopolymer, just like, plant-based biopolymers, just like, cellulose, hemicellulose, even plant starch. So all those materials is a polymer, and it is a polymeric material. So it is having a repetition of single unit, and all those materials can be used nowadays for formation of different, having a different applications, specifically in the packaging industry, biodegradable packaging, container, developing different containers. So we need to, make a different types of, biocomposite materials with one another, and we need to check that what is its property. And even the intercellular biopolymers also, just like a PHB. So we can produce it with the microorganism, and we can able to check its property level with the combination with the other biopolymers, how much it is effective for the development of such kind of biodegradable films, or specifically applicable for the packaging industry and all. So this is all of my presentation, and just like to conclude, like again the same mantra what our Honorable Prime Minister has already discussed, that is inclusion towards a sustainable lifestyle, that is sustainable pattern of consumption, sustainable pattern of production, and environment friendly lifestyle. So if you will adapt in that particular way, specifically, as I have already mentioned, that industrial processes are contributing 22% in US. You are seeing is a large amount of it is contributing. So if we if we adopt such kind of a biochemical based green processes, so they definitely while avoiding such kind of a petrochemical based process or green, so we can definitely able to reduce the amount of greenhouse gas emission, as well as the sustainability of the process can be also enhanced. So thank you very much. This is all about my presentation.
Thank you, Professor Pinaki, for such a wonderful presentation. And now platform is open for questionnaire session.
Hi,
Hello.
yes, Dr. Jaitanayak here. And
yes,
can I ask you one question? Like, I want to know Dr. Pinaki's point of view regarding the plastic usage, because nowadays the plastic usage that is having a lot of impact, and like though we have to use it for some kind of non-biodegradable things are also like important in our lifestyle. So what could be your view? I just want to know your view regarding that biodegradability is also required, and non-biodegradable, some of the products are also required in our lifestyle. So how you see this?
Yes. It is a very important question, and I like to answer this question in a very broad way. Like if we see the existing material, polymeric material which are existing for developing such kind of a polythene, and just like, so what are their properties specifically? So first of all, we need to know the properties. So there are few properties, just like water vapor transmission rate, as well as oxygen transmission rate. So those are the properties actually there, and they are actually we are lacking. Means all those biodegradable polymers who are what we are looking. So that's why Brazil is still that thing is not comes to the market. You can see. So, but although the lot of researches is going on throughout the world. So what specifically the research is going on, like first of all, we are developing such kind of biodegradable polymer from the microbial route, just like a PHA or PHB even, what we are doing is finally its property also doesn't match with the required property. So what we are doing is like we are blending it with other type of polymers, just like as I have already shown in the last slide, just like a nanocellulose or other material, just like even lignin or lignin dots or lignin based carbon dots, or some other material just like starch. So it will get a particular property, that particular property. So the lot of research is going on in that particular direction, so that we can get that particular property of the requirement. And definitely then it will definitely come to the picture actually.
Yeah, sure. Thank you. Thanks a lot.
Thank you, Professor Pinaki.
Thank you. Thank you so much for giving me this
We
opportunity.
sincerely appreciate your presentation. And our next speaker is Dr. Aarti Bhatt. Over to Dr. Aarti Bhatt. Let me introduce her. Her basic is B Chemical Engineering from Assam Engineering College, Guwahati. She did her ME in same discipline from University of Turkey and PhD also from same discipline from IIT Delhi. At present, she is working as scientist F in the same organization, and her expertise domains are solidification stabilization of metal waste, environment impact assessment, risk and hazard assessment for chemical process industries. So, welcome, Dr. Aarti Bhatt.
Yeah, thank you. Thank you so much, sir. Very good afternoon to all of you.
Thank you. Thank you so much.
So I think, should I straight away start, sir?
Yes, sure, sure.
Yeah. Is my presentation visible?
No, Dr. Aarti.
Okay, just give me a moment.
Yes, sure.
Is it visible, sir?
Yes, yes.
Okay, okay. I'll put it on the slideshow mode. Yeah.
Yeah.
Okay, so in fact, when Dr. Shipra Mishra approached me for giving a presentation in EWASH, strategizing a greener future, I didn't know exactly how I would fit the bill. So, but then she said that you look at your domain expertise and see how you can contribute and talk on a topic which is related to this. So after a lot of thinking, this is what I thought about, how that green hydrogen, safety management of green hydrogen, because chemical process safety is my domain area. And there's a lot of talk about going for renewable sources of energy. And hydrogen is taken as one of the most prominent sources of energy, fuel in the future. Now, taking hydrogen in a greener route is what will make this planet more greener. So I thought I'll focus not about on generation of green hydrogen, but on safety management, because it is equally important to for a safety management to be in place if we were to go in a sustainable manner, if we were to use go into a green hydrogen economy. So with this background, I would like to share with you all, this is how I outline my presentations. I'll give a brief role and function about my organization, so that kind of, I think the participants can relate to what we do here. I'll just give a brief of the National Green Hydrogen Strategy, so that that background is built. Different types of hydrogen, why specifically green, the relevant hydrogen safety and safety standards in place internationally and in the country, the potential hazards which are associated with hydrogen, what are the failure mechanisms involved, and the risk management, and I'll end up with a summary. So I'll not take too long to share all this with you. I'll be very quick, I know, because I'm coming this lunchtime also. So coming to the
Sorry
first
to
slide.
interrupt, Aarti. Your slides are not changing.
They're not changing, ma'am?
Yeah.
Yeah, this is the first one I've changed. Is it visible? Role and function of CIFIs?
No. I can't see.
Okay.
At least I don't know about others.
I can see. I can see.
Oh, is it? Okay.
Yeah. Okay. So shall I continue?
Yeah.
Yeah, sure.
Yeah. Okay, so coming briefly to the role and function of Center for Fire, Explosive and Environment Safety. This is a DRDO lab from where I'm speaking. So it has three major roles. One is the regulatory role, in which we are involved in formulation and updating of safety regulations with respect to fire, explosives and environment. We also do implementation and compliance monitoring with these all these three verticals, in especially catering to the Ministry of Defense establishments, which involve all defense forces, ordnance factories and defense PSUs, etc. We also undertake a lot of advisory and do a lot of R&D in these areas which contribute to safety. So basically we develop a lot of safety systems which are required to be fitted in our defense for defense applications.
Dr. Aarti, please allow me to interrupt a little bit. We can see only your first slide, ma'am. I think you have moved on, but only the first slide is visible to us.
Okay.
Please put on to the sliding, the full view mode, and then please move on to this slide.
Yeah, I'm already on full view mode.
Ma'am, click on the second slide. Click on the slide number 2.
Are you able to read my slide now? National Hydrogen Strategy?
No,
ma'am.
we can
No,
see
no,
only
no, it's
green
not
hydrogen
changing.
safety management. Your first slide is visible to us.
Yes.
Now we can see your fourth slide, National Hydrogen Strategy.
Okay, I'm putting it in slideshow mode now.
Yes, do that, please. Yeah.
I think
So
slide
it is-
also needs lunchtime.
It's slowing down. Now, is it visible?
Yes.
Okay. So just to set the background about what the National Hydrogen Strategy is given, it is designed to construct a high-tech and low-carbon Indian brand. Of course, this is already there in the vision document, which has been given by Niti Aayog. And the targets are, of course, to have the world's largest electrolysis green hydrogen generation with over 60 gigawatt of 5 million tons by 2030. So we are not very far off unless and until we work on mission mode in certain technology areas. I think a breakthrough needs to be achieved in this area. But nevertheless, I think there are already policy interventions in place. And with proactive collaborations from the innovators, entrepreneurs, and the government and R&D and academic structures, I think we can largely help in developing green hydrogen so that we can reduce the CO2 emissions and reduce our carbon footprint. So this green hydrogen policy was given as latest in 23rd of February 2022 only and issued by the Government of India, Ministry of Power. And it has been very clearly brought out in the document Harnessing Green Hydrogen. It is available in the net. And for all those of you who are interested in this, you can go through it. There is also a National Green Hydrogen portal on the internet. And you can look at various areas in which researchers in the country are working in the area of green hydrogen. I think there are a lot of R&D people in this part as participants here. You can have a look at it. So we'll next move one step ahead where this green hydrogen roadmap is leading to. So this is a roadmap which has been laid out right from 2025 till 2050. So there are certain sectoral areas which have been targeted by the government, which they see has the maximum potential for absorbing green hydrogen, like ammonia, fertilizer industry. Then we have the refinery sector, and then city gas distribution sector, and so on and so forth. So till 2030, we see this is what is expected to be achieved. Then, of course, going to the industrial sector, green steel, domestic green steel, and heavy duty trucking, etc. So the point here in showing you all this is to show that how green hydrogen is going to be used on a very large scale and in as diverse as areas as gas distribution network to refineries and fertilizer industry. So you can imagine with the kind of hazards which are associated with hydrogen, safety management would be very prudent to have safety management practices in place so that all these sectors function with minimum number of accidents and minimum number of injuries. And just to brief people who are not exactly aware about what green hydrogen is, let me go back and tell you about other colors of hydrogen also. So it is often associated with gray or brown hydrogen. It is typically produced from natural gas in a process called methane reformation. This is a very well-known process. And brown hydrogen is produced from coal gasification. Again, a very, very old process. Now, these are mostly dominant. These are the most dominant and the cheap methods of hydrogen production. We all know, but they account for the highest CO2 emissions. Hence, thought about changing the method of product. There's a lot of R&D which has gone into changing the way in which hydrogen is being produced. Now, there's another color which is
Sorry,
associated.
Aarti, again,
Yes.
I think I'm sorry to interrupt, but
Oh, I
आपकी
think.
slide change नहीं हो रही हैं.
So should I then continue in this mode only?
हाँ, हमको तो इसी मोड में देख रहे हैं हम तो.
I think
हम
if
तो
you are
slide show
on,
में देख ही नहीं पा रहे हैं.
should I continue in this mode instead of slide
that
show
is
mode?
better.
Okay. So then I'll continue this way only. So after the blue, gray and brown hydrogen, the next is the blue hydrogen. This is slightly better because the CO2 footprints are a little bit reduced on the environment. It is produced by fossil fuels only, but only thing is it has got a carbon capture sequestration in place in order to reduce the global warming on the earth. And it can be a stepping stone towards a green hydrogen economy, because abruptly we cannot be changing everything in one step. So this is touted as the intermediate step. And the next comes the green hydrogen, where it is produced from a renewable source. It could be solar, wind, by electrolysis of water. The process is clean but currently expensive because of the technology still not meeting the breakthrough point. And the cost is majorly due to the electrolyzers and the cost of the energy source. So we see here that blue hydrogen and green hydrogen, there's only a little bit of difference, but ultimately they all merge together. That is what I want to conclude from this slide. Now, coming to the sources of hydrogen production, this is the data of 2018. So look at here, we see what is the potential. We have blue hydrogen 48% and green hydrogen 48%. And the green hydrogen, which is generated by water electrolysis, is just 4%. Now, this is being majorly targeted at. So there are other methods of production of green hydrogen. It could be using direct solar, biomass, solar, photovoltaic cells, wind, geothermal energy. You can use direct water splitting, but using all these renewable sources of energy, and then store hydrogen as per the requirement and go ahead with your distribution network, whichever sector it has to go. So this is all the background about the hydrogen. Let's come to the safety part of it, which is what I thought I should focus on. So coming to hydrogen safety, why hydrogen safety is important, right? If we are going to use hydrogen on such large scale, what is it that makes hydrogen to be so concerned about? First, let's do a fact check about hydrogen. Hydrogen is characterized by low molar mass, which indicates the low gas density. It doesn't have any odor and color. You can neither see it nor you can smell it like many other gases. There is a possibility of flaming because it has got a very wide flammability range, ranging from 4% to 75%. So the minimum ignition energy, ignition limit range, and energy density indicate that hydrogen can be easily ignited, and it will also release a large amount of thermal energy. Compared to natural gas, hydrogen is characterized by low ignition temperature and faster burning velocities, which indicate that spontaneous ignition will occur if hydrogen encounters sparks or hot surfaces. Therefore, ensuring hydrogen safety becomes the highest priority when you are looking for hydrogen utilization. And this is applicable for all sectors. Going a little more detail into the physical properties of hydrogen, because if we have to manage hydrogen safety properly, we need to know about the properties also, because the hazards are an intrinsic, reflect the intrinsic properties of the material as well. So you can look at this is showing a comparison with natural gas, which is also a very widely used fuel. So look at the density values, the minimum ignition energy values, see how easily hydrogen can be ignited, such low values. And look at the flammability limit range. It's very wide, 4 to 75%, whereas natural gas, it is only 5 to 15%. So the vulnerability of hydrogen in case it leaks and gets entrapped in an enclosed environment is much higher compared to natural gas. So this is what I just wanted to show this slide for, to highlight the properties of hydrogen. So, and now just coming next in progression, what are the codes and standards in place for regulating hydrogen? So if you look at this particular slide, we don't see any Indian regulation here. But yes, there are a lot of regulations which are there internationally, which are set up by national fire protection agencies. So NFPA 2 talks about hydrogen technologies code. NFPA 30A talks about rules for designing of refueling stations. That is, if we are going to go on a hydrogen gas-based economy, then there are certain set of rules which will have to be followed, keeping in view the very high risk associated with handling of hydrogen. And then there are, of course, other rules also in place. 40 CFR talks about chemical accident prevention provisions, and 49 CFR talks about regulations on shipping and handling hydrogen gas and cryogenic hydrogen. So these rules and regulations will have to be put into place whenever we are designing our facilities at appropriate levels. So coming specifically to a few hazards of hydrogen, I think just 1 minute. Yeah. So one of the I'll take up a few hazards which are associated with hydrogen. The first one is the combustion hazard. We are very well aware about flammability of hydrogen. So here we have methane, here we have propane, very well known fuels, and here you have hydrogen. So this is the wide range flammability range. So you can see, based on this itself, you can look at how high the combustion hazard of hydrogen is. So formation of combustible mixture, leakages, ignition sources, all these are going to compound the combustion hazard which is associated with hydrogen. Now, leakages are something which are very common in industries, and hydrogen being the lightest known gas is prone to leakages. It easily leaks into the environment. So as a safety expert, whenever we do an analysis, we look at the probable scenarios in which a particular event can occur. So if based on literature, we have, there are statistics that hydrogen leakages have led to the maximum number of accidents. So the probable ways in which a hydrogen can leak is, if it leaks, it can go into an open space or it can go into a confined space. If it goes into an open space, there are chances of rapid ignition in case a source is present, or it can lead to a simple diffusion, and there will be no, in case there is no ignition source, it will just diffuse out into the environment. But in case there is a source, then it can lead to a jet fire. In case of a confined space, again, there is a probability that this leakage could be either continuous leak or it could be an instantaneous leak. In case of a continuous leak, in case a rapid ignition source is it can lead to rapid ignition if a source is present, and again it will culminate in a jet fire. It may also lead to a cloud, I mean, depending upon the pressure at which it is released, or it can also have a scenario of delayed ignition. And this delayed ignition again can have a different ways in which the physical effects can manifest. It can be an ignition kernel, it can be an explosion, or it can be a fireball. So you see, a simple leakage can lead to different physical scenarios, and depending upon these physical effects, the damages will vary. For example, if it is just if it is a fireball only, there will be effects due to thermal radiation. It can lead to first, second, third degree burn depending upon the energy which is released. But if it is an explosion, then there are going to be physical effects and damages which are associated with blast and fragmentation also. So this is how a simple hydrogen leakage can culminate in, depending upon the inventory and the scenario in which it is being released and the duration at which it is being detected. So the earlier we are able to detect, the lesser will be the damage. Let's now talk about pressure hazard. This is another very common and very hazard which is of concern to all the industries. Now, pressure hazards arise from the need to concentrate hydrogen, right? We need to have hydrogen in a very portable scenario. So we have to concentrate it by putting it under very high pressures. So a concentrated form of hydrogen is nothing but it is a significant stored energy source. It can be at cryogenic conditions or it can be under very high pressures. This will lead, in case something wrong happens, it will lead to overpressure, shock waves, fragmentation if suddenly released. Now, the possible causes in which these can occur will be due to liquid to gas phase change. It could be due to overfilling. It could be due to pressurization system failure. It could be due to relief system failure or inadequate venting. Fire or overheating from an external source will also increase the pressure in which the hydrogen is contained and ultimately lead to these particular scenarios. So it is very important to keep these things in check whenever we are having pressurized hydrogen facilities in our surroundings. Now, hydrogen is also known to be associated with low temperature hazard. Now, low temperature embrittlement of containment materials and nearby materials. This is a very common cause of hydrogen failure, and it's very important to look at our design codes and choose a very appropriate material when we are thinking about handling hydrogen, and also looking at the conditions under which we are handling hydrogen, whether it is at low temperature or whether it is at high pressure, and so on and so forth. Because hydrogen embrittlement is something which is a cause of concern for most of the safety experts. The contact with the cold surfaces can also result in cryogenic burns, which is also commonly known as frostbite. So it's important to insulate the cold surfaces and use appropriate personal protective equipment also. Now, the low temperature hazards, the other things which are associated with low temperature are dimensional changes because of contraction. If just for the sake of example, if we look at stainless steel, it contracts by 0.3%, and PTFE will contract around, roughly it is reported at 2.1%. So you can look at there's a lot of dimensional change associated with low temperature hazard, then the chances of leaking and other issues will automatically creep into the system. Many plastics even become extremely brittle and cracking when cooled to 20 Kelvin. Now, such materials cannot be used for seals, valve seats, etc. So this is what I want to bring out when I'm sharing about low temperature hazards. The choice of material is to be done with extreme care, because the behavior of the material changes if you're handling hydrogen at such low temperatures. Now we come to the embrittlement hazard. This is, like I said, is a major concern in the hydrogen industry. What exactly happens is hydrogen dissociates and atomic hydrogen penetrates the metals, causing a decrease in the material strength. The mechanical properties are significantly reduced. When talk about tensile strength, ductility, fracture toughness, and crack behavior, these are some of the properties which have been measured under various conditions and during prolonged periods of exposure to hydrogen, and they have known to be deteriorate considerably. And the failures in the industries have also been recorded unexpectedly because of hydrogen embrittlement. This is commonly addressed by correct material selection, a conservative design stress, increased material thickness, choosing the correct welding technique when we are when we are getting things fabricated in the process industries for handling hydrogen, whether it is the pipelines, joints, etc., and also looking at the surface finishes. So the next hazard which I would like to highlight here is associated with health. Now, exposure to flame, hydrogen flame, especially because, like I said, you can neither see the hydrogen flame. So direct contact with hydrogen flame can lead to thermal burns. The thermal energy is radiated from the flash fires. And also it's comparable to extreme UV exposures also. And under extremely cold conditions, it can lead to cryogenic burns, which is equivalent to a 3rd degree damage to the skin. It can also lead to hypothermia and asphyxiation. And purging gas is generally recommended if there is excessive of hydrogen atmosphere in order to get rid of asphyxiation. And it is also known to cause overpressure hazards. That, of course, is already discussed. We have already talked about that. And the table on the right actually is trying to give the probit values which are associated, the damages to the personnel corresponding to the various overpressure values. These are, of course, taken from literature. And I was also trying to do a literature search in order to find out what is the probability of hydrogen failure and in which scenarios we have normally encountered hydrogen failure analysis. I could come across a very good review paper, and I'll share with you all the findings of this review paper. And this talks about the locations in which the hydrogen failures due to hydrogen have occurred. So there are different colors which have been indicated for different locations. See, their laboratory, commercial facility, delivery vehicles, storage facility, power plants, chemical plants, so on and so forth. So it's very astonishing to see that laboratories are associated with hydrogen maximum incidents of hydrogen failures. Probably it's in laboratories that a lot of R&D work occurs, and we are trying new and new things every time. So the failure probability or chances of overlooking or something going wrong increases. So we need to be very careful when we are handling hydrogen in our laboratory also, because this is very widely reported. Now the incident equipment, which equipment is responsible for maximum leakages? Here again, piping, fittings and walls have been known to be the major causes for hydrogen leakages. They amount to an alarming amount of 32%, and followed by other systems like vehicle and fueling stations, ventilation systems, and so on and so forth. Now, this slide had actually tried to analyze the probable causes which could have led to a hydrogen failure. Like we talked about, there are different ways in which the failures can occur. Now, some of the failure modes have been recorded here, including human error, design flaws, inadequate equipment, etc. So, equipment failure has been reported to contribute to the maximum number of failures. So now equipment failure could be probably because we are still trying to come up to levels with the design codes and materials which can handle hydrogen under very extreme conditions and for long durations. This could be one reason. Now, what kind of damages and injuries were associated? So 41% of the damages were in terms of property damage, and minor injuries corresponded to 10.2%. These were mostly related to burns because of the thermal radiation which was associated with hydrogen leakage. And most of them have not been recorded, but yes, property damage as of now has been reported. So looking at so many hazards and the risks which are associated with hydrogen, so you can well imagine if we are going to go in for a hydrogen economy, then what will be the implications, right? The vulnerability is going to increase manifold. The exposure in terms of people handling in hydrogen is also going to increase manifold. Hence, it is, as a safety expert, I recommend that we should have a safety, we should follow a safety system safety approach so that we can minimize the risk due to handling of this particular hydrogen. Now, the system safety approach talks about defining the objectives, defining the system. Now the system could be electrolyzer, which is generating hydrogen. It could just be a pressurized hydrogen tank, and the upstream and the downstream piping systems. So that is what when I mean system. Then the associated hazards which would be there in that particular system, and doing a risk analysis. Risk analysis is trying to find out what is the probability of things going wrong, and what are the likely exposure of people to be there around the facility. So this is a very detailed analysis which is generally done based on well-known tools and techniques. And then decision making has to be taken by the competent authorities who are responsible for that particular facility, whether the risk is acceptable or not. If the risk is too high, then we need to put in risk mitigation measures in place, and then relook at the risk values. So this is going to be an iterative process, and it has to be done on a periodic basis, so that the risk is managed well within the tolerable ranges. And this is what is the systems safety process. It talks about documenting the system safety approach, identifying and documenting the hazards, assessing and again documenting the risks. So identify and document the risk mitigation measures. So you see there's a lot of emphasis which is given on documentation, because often we try to do analysis and don't document them. So when some other person comes and takes over the facility, then they're not able to really make out as to what exercise has already been done and where to begin, or whether to go in for modifications and all. So well-documented approach, it emphasizes on a well-documented approach, and reduce the risk, verify, validate, and again document the risk reduction, accept the risk and document it also, and then manage the life cycle risk throughout the plant life cycle or the facility life cycle. So if I were to summarize this presentation, then I would emphasize that risks and hazards associated with hydrogen handling, storage, and transportation have to be assessed. I'll also emphasize that people who are working with hydrogen in laboratory need to take equal care. Just because the inventory is less doesn't mean the harm will be less. Depending upon the scenario, the harm could be much more. So do take care, all due precautions when handling hydrogen in laboratories. The risk mitigation measures have to be formulated and adhered to strictly. The hierarchy of controls in the order of preventive engineering controls, administrative controls, and protective measures should be in place in all hydrogen handling facilities. The personnel who are to work in hydrogen have to be trained appropriately, and the existing body of knowledge should be used in making the standard operating procedures for safe operation, and system safety approach should be adopted to minimize the hazards, manage risk, and use hydrogen for a greener future, I should say. So with this, I'll conclude my presentation. And if you all have any questions, I'll be very glad to answer them. Thank you.
Thank you, Dr. Atif, for such an informative information on renewable source of energy.
Yes.
Now, the platform is open for question.
Hi, Dr. Aarti. Am I audible to you?
Yes, yes, I can hear you.
Okay, okay. So I would like to know, like you have told about different types of leakages,
yes,
for which some kind of explosion may happen. So,
yes.
regarding your review, what do you feel that could be the best possible material of construction, MOC?
See, I would
hydrogen?
I would not be able to comment on that right away. Because like I said, depending upon the specific conditions in which you're going to handle the hydrogen, whether it's going to be under cryogenic conditions, or whether it's going to be under ambient conditions, under slightly pressurized, I think the MOC will have to be appropriately chosen. So right now, I'll not be able to give you a direct answer.
Okay, okay. No, I was just asking regarding not cryogenic, but liquid. gaseous hydrogen. So if we want to pass, because we have also worked on like some kind of hydrogen transfer. So like I would like to know like from you for the gaseous transfer, if we need to pass hydrogen through one reactor or through one channel. So what
I
could be
think
the
if you are doing at laboratory scale, maybe SS3 304 or 314 should be safe enough, according to me.
yes, that's what we have used actually.
Yes.
So that is good. Okay.
Yeah, I think in safety it is always better to err on the higher side.
Okay, nice.
Yeah.
Got it. Thank you.
Yeah, I have just one question.
Yes,
Whether
ma'am.
you are working involved in this kind of safety aspects?
Yes. Our lab is actually has the mandate. We do safety assessments for most of our ordnance factory, wherein we do a lot of chemical processing of high energy materials and all. So wherever hydrogen is required or wherever other chemicals are required, as a mandate under manufacture and storage on import of hazardous chemical rules, MSISC 1989. CIFIS has a mandate to do it for MOD establishments. So we do a lot of safety assessment.
For civil, I am just asking because it is equally important for civil users also, no?
Yes, ma'am. No, we are, I mean, we cater only to MOD establishments.
Yeah, but I think some way you should be helpful to, because now hydrogen they are going to use, in
Yes.
everywhere for civil purpose.
Yes,
And
yes.
my concern is also really like using it is so safe. If, all the safety measures what you have mentioned here, if they
Yes,
are
ma'am.
not followed properly, then
Yes, ma'am.
so
Yes.
that is the biggest risk in this. Although
Exactly.
yeah. Anyway, but it's a good, very good presentation.
Thank you, ma'am.
Thank you, Dr. Arti.
Thank you so much, sir. Thank you all.
Over to coordinator.
Okay. Now I request our chairpersons, Dr. Suraj Kumar Tripathi and Dr. Maulin P. Shah, to give their remarks on this session. Maulin sir and Suraj sir. Maulin sir, Suraj sir.
हेलो आवाज आ रही है मेरी?
Yes, sir, you are audible, sir.
Yeah. We are very much thankful to all the participants, including speakers, for today's session. And it was really a remarkable presentation from the speakers.
Thank you so much, sir. Thank you so much, Dr. Suraj Kumar Tripathi and Dr. Maulin P. Shah, for chairing this session so beautifully and nicely and for joining us today. Now we begin with the oral presentation. And the chairpersons for this session would be Dr. Rabia Parveen and Dr. Jayato Nayak. I now request Dr. Richa Tyagi to kindly introduce our chairperson, Dr. Rabia Parveen.
Thank you, Dr. Ragvi. Introducing Dr. Rabia Parveen. Dr. Rabia Parveen is currently working as an assistant professor in Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. She did her bachelor's from Jamia Hamdard and did her master's and PhD in pharmaceutics from Jamia Hamdard itself. She did her postdoc from Jamia Millia Islamia under UGC scheme. She also did 3-month research training from Princeton Institute of Medical Research on cell culture and anti-HIV screening of nano formulations. Her field of interest and research includes nano-drug delivery system for herbal drugs and chromatography methods for bioanalysis of herbal drugs. Dr. Parveen has published around 100 papers, including research articles, reviews, book, chapters, etc., and filed 1 patent also. She has been awarded several fellowships like DST Inspire, CSIR, and many more. She has also been selected as a subject matter expert for UGC e-Pathshala scheme. In addition to her academic activities, she's a reviewer of many good impact factor journals, member of different societies, and a member of different professional bodies, namely Student Life Member of Association of Pharmacy Teachers India, Life Member of Indian Pharmacy Graduates, and many more. We welcome you, ma'am, and thank you for joining us today. And I request Dr. Raghavi to introduce our next chairperson, Dr. Jyoto Nayak, once again.
Thank you, Richa ma'am. I take this opportunity to introduce our chairperson, Dr. Jyoto Nayak. Dr. Jyoto Nayak, he is currently working as an assistant professor in Center for Life Sciences, Mahendra University, Hyderabad. Dr. Jyoto Nayak is a prominent researcher with more than 10 years of experience in the field of novel design and applications, embracing the theme of process intensification. His core expertise includes process intensification, bioprocess engineering, water and wastewater treatment, graphene integrated photocatalyst development, and biosynthesis of value-added products. Currently, along with the national collaboration with IITs, NITs, and CSIR labs, he has extended research collaborations with research professionals in Poland, Brazil, Malaysia, Vietnam, etc. Till January 2023, he has published 25 international SCI Scopus indexed peer-reviewed journal papers, more than 20 book chapters, and filed 3 patents. As of now, with huge number of citations, he has achieved more than 10 H index. He is providing his expertise as resource person for several webinars and FDPs, and also as senior editor, reviewer editor, guest editor, editorial board member, and recognized reviewer of more than 15 international journals. He has received 2 Best Research Awards and 5 Distinguished Awards from various national and international organizations. We welcome you again, sir. Thank you for joining us.
Thanks for the nice introduction. So we are going to start the technical session now, right?
The oral presentation session. Yes, sir.
Okay,
yes,
fine,
sir.
fine.
Okay. So the oral presentation round has 10 enthusiastic participants who would be presenting their unique work. Following are the guidelines for oral presentation. Participants are requested to adhere to the guidelines. Presentations would be marked on the basis of number 1, originality and creativity of the work. Number 2, content and presentation skills. Number 3, impact of the demonstrated research on future prospects of humankind. Number 4, responses to the questions. And most importantly, the total time allotted for each presentation is 6 minutes. 5 minutes have been allotted for the presentation and 1 minute for the question answer round. And the results would be announced in the valedictory session today. Now over to you, Jayota sir and Rabia ma'am.
Raghavi, I think we can call the first presenters.
Okay, okay.
Yes, we can call the first presenters and we can start the presentation.
Okay, the first presenters are Priya Goyal and Reema Chhabra.
Yes, good afternoon, ma'am. I'm Priya Goyal. I will be presenting. Ma'am, can I?
We can hear you.
Yes, yes. Okay. Is my screen visible, ma'am?
Yes, you can start.
Okay. Thank you. I'm Dr. Priya Goel from Department of Zoology, Deen Dayal Upadhyay College of University of Delhi. And the topic is monitoring of pesticide pollution. What have we achieved and what lies ahead? So first of all, what are pesticides? The name is not new to us. Pesticide is a term which is widely used to denote any substance intended for repelling, mitigating, or killing any potential disease-causing organism. The commercial categories include organochlorines, organophosphates, carbamates, and pyrethroids. A pesticide has to be registered with CIBRC, Ministry of Agriculture, India, and regulation is done by Insecticides Act and Pesticide Management Bill, which has been introduced recently. Now, why are we concerned? Why are we concerned about pesticide pollution in the first place? Pesticides have been used since very long, but they have been used very injudiciously and indiscriminately, which has led to serious in human body, and this is dangerous for humans and livestock alike. They have been degrading soil quality and the surrounding environment. This is what we term pesticide pollution, for which the continuous monitoring is absolutely essential to preclude their entry into biological samples and human bodies. So this presentation will give a comprehensive overview of what on-site detection techniques of pesticides are possible. The first conventional techniques that have been used, which have been instrumental till now, include gas chromatography, HPLC, coupled with MS. However, they have their own limitations as well as advantages. So need of the hour is a rapid on-site and mass screening method for pesticide detection, for which immunoassays are considered gold standard, since they are rapid, cost-effective, and they allow large-scale quantitative screening of the samples. They have the potential to be adopted as routine tests. In market, a large number of rapid detection ELISA kits are available, which can use a large number of biological samples. The most common ELISA kits are based on dipstick assays. Now, dipstick assays utilize a paper mesh or a cardboard strip which is pre-coated with the reagents. As we can see in this diagram, here the antibodies which are specific to coated on the nitrocellulose membrane. Now, as they come in contact with the biological sample, which potentially contains a pesticide, the amount of color present in the dot gives us an idea how much of pesticide is present in the biological sample. They have their own advantages. However, though immunoassays, as I said, are considered gold standard, they have their own limitations. Since they are immunoassays, they are based on the interaction between antigen and antibody. They also show some false positives, cross-reactivity with similar epitopes. So further validation is required. Beside, a kit is generally used for a single analyte assay. So moving further, in scientific world, biosensors have dominated the diagnostic field since long. And in this pesticide detection also, biosensors have also come very handy. Now, the general scheme of a biosensor involves targeting a biological sample which contains a pesticide, and the pesticide, the analyte, will be bound to a bioreceptor, which in turn will generate a signal which will be transduced, amplified in the form of a signal. Now, which will be amplified in the form of a signal which is visible to the naked eye, or maybe some electronic equipments can be used. But this whole system can be made very portable, very compact in the form that they can be used easily for on-site detection, for field assays. Now here, when the transducer used, it generates an electric signal. We consider it an electrochemical biosensor. If it is heat signal, we consider it a thermometric signal. Then optical properties are changed, then we consider it an optoelectronic signal sensor. And when there is vibration frequency of a piezoelectric crystals changed with the increase in mass as the pesticide comes in contact with the bioreceptors, then it is considered a piezoelectric biosensor. So these are the biosensors which are based on the type of transducer or the detection signal which is used. Second are the biosensors. We can categorize them also on the basis of the kind of bioreceptor.
Dr. Priya, just 1 minute. Half of your time is over. So can you please wrap it up and just can you show the like what you are developing, or
In this presentation, I'm giving an
sure.
overview.
Sure,
I'm
sure.
not giving my research. So
Okay.
biosensors can be enzyme-based, they can be immunosensors, they can be cell-based, they can be DNA-based, APTA sensors, or even molecularly imprinted polymer-based sensors. So we know that certain pesticides, they inhibit the activity of certain enzymes. So what enzyme is involved, what specific pesticides are involved, and what is the level of inhibition? This can be made use of in finding out what is the amount of pesticide which is present in the sample. Now, based on the antigen-antibody system interaction, now immunoassays have been clubbed with biosensors to form a new category of sensors that are immunosensors. So the same phenomena of analyte antibody and interaction can give an alteration in physical properties, but still they also have their. Similarly, for binding to a solid support, instead of antibody, we can use whole cell microbes or we can use cell organelles. So the basic function is that when the pesticide will be degraded by the enzymes present in the cell, then those degradation products will result in the production of electroactive compounds, which will be detected by the biosensors and which can be visible. Similarly, the degradation products may be further oxidized by the bacteria to oxygen. Now, these kind of techniques, they are more stable and more cheaper alternative because there is no extraction and purification of enzymes involved. Next is the category of DNA-based
Ms. Priya, can we, I'm
Yes.
sorry to interrupt, but will you like to conclude because we're running short of time and it's already 5 minutes over from your side. If you can come to the conclusion side. Yeah, thank you so much.
Okay. So after DNA-based biosensors, we have APTA sensors. We have molecularly imprinted polymers. While biosensors have their own pros and cons. So future prospects involve the use of antibody fragments. It uses the miniaturization of the biosensing devices so that they can be used in field analysis. And we can also go to molecular modeling systems. So I conclude that despite making so many advances in the field of sensitive diagnostic, to also still need next generation devices for better on-site detection. Thank you so much.
Thanks, Dr. Priya, for your wonderful presentation, and you have given a quite a lot of overview regarding this topic for pesticide pollution. Can I ask you one question regarding like what you are thinking about the disposal of these strips, because it's handy regarding the pesticide pollution. If we want to monitor some kind of kit, that will be much more handy. But what happens that those kits, they further during the time of disposal, it is creating the most of the pollution. So what's your point of view on that?
That's right. Disposal definitely requires, I mean, for that purpose, we can use some kind of materials which are degradable. For example, some biopolymers can be used, or just imprints on which the antibodies, or we can say the enzymes, whatever we want to use, they can be placed. So if they are degradable, biodegradable, then it may be a better alternative.
Yes, thanks. But there also comes another perspective that the cost will be higher. So anyway, we need to optimize the cost also.
Yes,
so
that
for
is
that,
true. Like recently,
because it will be
starch
used
has been
mostly by the
used.
farmer. Yeah, because it will be
Yes,
mostly
that is
used by the farmers. So anyway, thank
Yes.
you. You carry on your research in this field, and let's see. Thank you.
The next presenters are Ms. Pooja Rana and Professor R.K. Sharma. In the meantime, there's an important announcement. Student activities to take place after the poster presentation of next technical session. I repeat, important announcement, student activities to take place after the poster presentation of next technical session. Thank you.
Shall I begin, ma'am?
Yes.
Good afternoon, everyone. I am Pooja Rana from Department of Chemistry, Delhi University. And the topic of my presentation is design and synthesis of two-dimensional boronitride nanosheet-based nanocomposites for diverse organic transformations. So let's begin with a brief introduction about BN nanosheets. So basically in BN nanosheets, boron and nitrogen atoms are placed alternatively via strong sp2 hybridized covalent bonds, forming a hexagonal ring-like structure. These hexagonal rings fuse together to form a layer structures where layers are held together by weak van der Waals forces of attraction in AB-AB symmetry. Due to the higher electronegativity of nitrogen atoms, BN sigma bonds are more confined towards nitrogen atoms, due to which boron atoms become electrophilic centers and are more susceptible towards nucleophilic attack. Owing to their exceptional structural features such as large surface area to volume ratio, high thermal and chemical stability, excellent mechanical strength, resistance towards oxidation, high durability and nanometer size, we have chosen BN nanosheets as a solid support and further utilize them in catalytic applications. In view of the importance of hybrid nanomaterials, a plethora of solid supported catalysts have been fabricated till date, amongst which two-dimensional nanomaterials, especially boronitride nanosheets, have proven their efficacy as a solid support, which offers a large number of active sites for introducing organic functionalities or metal complexes. Further, to enhance the susceptibility of BN nanosheets from the reaction media, ferrite nanoparticles are integrated with these nanosheets, which has emerged a greener alternative route to energy consumption techniques and the saving time, energy, as well as catalyst flows. So discussing my research work, which is magnetic boronitride nanosheets decorated with cobalt nanoparticles as catalyst for the synthesis of 3,4-dihydropyrimidones and thiones. This paper has been published in ACS Applied Nanomaterials. So here we have synthesized magnetically responsive boronitride nanosheets supported cobalt catalyst. So initially, we have synthesized BN nanosheets by subjecting a homogeneous mixture of sodium hydroxide, potassium hydroxide, and pristine boronitride powder to heating condition at 180 degrees Celsius using a muffle furnace. Thereafter, ferrite nanoparticles were integrated on BN nanosheets via solvothermal approach. For that, BN nanosheets were dispersed in ethylene glycol under sonication, followed by addition of ferric chloride, hexahydrate, sodium acetate, and polyethylene glycol to form a homogeneous mixture, which was then transferred to the autoclave and heated at 200 degrees Celsius for 16 hours. And after that, cobalt chloride hexahydrate was added to the ethanolic solution of magnetic BN nanosheets, followed by a dropwise addition of hydrogen hydrate to form the cobalt nanoparticles onto the surface of magnetic BN nanosheets. To confirm the synthesis of the catalyst, various physicochemical techniques were employed, where SEM and TEM depict shape, size, and morphology of the synthesized nanocomposites. As can be seen from figure A, BN nanosheet shows lamellar-like structure with smooth edges and flat surface. Besides, the same micrographs of magnetic BN nanosheets and the catalyst shows the uniform distribution of spherical ferrite and cobalt-decorated ferrite nanoparticles onto the surface of the BN nanosheets. The TEM image of BN nanosheet shows 4 to 5 stacked sheets with average thickness lying between 3 to 7 nanometer. TEM image of the magnetic BN nanosheet shows spherical ferrite nanoparticles of size around 200 nanometer. In addition, the inverse cubic spinal structure of ferrite nanoparticle was confirmed through a SAD pattern, which shows wide spotted diffraction rings. Moving to the TEM image of the catalyst, TEM image shows that cobalt nanoparticles are slightly agglomerated with no obvious change in the morphology of the nanostructures. Further, the elemental composition of the catalyst was confirmed through EDS analysis, and the presence of iron and cobalt in the catalyst was confirmed through EDXRF spectroscopy. XRD, FTIR, and BET analysis were also conducted, where XRD depicts phase and crystallinity of the synthesized nanocomposites. FTIR was conducted.
Pooja, 5 minutes
Yes.
are over. Please kindly conclude.
Okay, ma'am. FTIR was conducted to authenticate the stepwise synthesis of the catalyst by identifying various functional groups, and the BET was carried out to determine surface area of the catalyst. Further, VSM was also carried out to determine magnetic properties of the catalyst, and the TGA was carried out to determine thermal stability of the catalyst. Further, to determine the surface area, surface electronic state of the catalyst, XPS was employed, where the core spectrum of cobalt shows that cobalt is present in zero oxidation state. Thereafter, the catalytic efficacy was investigated in the synthesis of 3,4-dihydropyridones, which exhibits several biological activities such as antiviral, anti-inflammatory, anti-cancer, etc. So, to surmount the problems which we have been facing for so many years, such as harsh reaction condition, use of toxic solvents, low productivity, and prolonged reaction time. So we focus towards combating these intrinsic drawbacks by rationally designing of magnetically retrievable boronitride nanosheet supported cobalt catalyst to access the high product yields in short reaction time under solvent-free approach. Thereafter, we carried out the optimization studies and we found best results when 15 mg of the catalyst was utilized under solvent-free approach at a temperature of 60 degrees Celsius for 40 minutes. Further, we explored the substrate scope and we found that both the aromatic aldehydes bearing electron withdrawing as well as electron donating substituents resulted in good to excellent yield of the desired products. We have also compared our
kindly conclude, please. 6 minutes.
With the literature precedents, and we found the protocol is the best protocol in terms of reaction conditions, reusability, and yield of the desired product. Also, we have carried out heterogeneity tests, which revealed that no metal has been leached out from the catalytic surface, and also the catalyst could be efficiently reused for 5 consecutive runs. So in conclusion, we have synthesized magnetically responsive two-dimensional boronitride nanosheets, which offer several advantages such as magnetically separable, recyclable up to 5 runs, high productivity, ambient interaction condition under solvent-free approach. These are my other publications: scientific reports, reaction chemistry and engineering, material chemistry frontiers, standard transactions. Thank you.
Yes, Dr. Pooja, good publications and good presentation and nice work.
Thank you, sir.
Just I want to know that if this catalyst, what you have prepared, if I want to use it at a high temperature, suppose 1000 degrees centigrade, suppose 1000 degrees centigrade, so can I use this catalyst or it will get degraded?
No, sir. Actually, it is stable up to 900 degrees Celsius or more than that, because
How much stability you are getting based on the mass loss in TGA? Up to 900 degrees.
No, sir. Actually, if we talk about BN nanosheets, actually BN nanosheets are highly thermally stable. But we are integrating BN nanosheets with the ferrite nanoparticles. So ferrite nanoparticles are not very much stable up to a very high temperature. So it will degrade up to around 350 or 400 degrees Celsius. Sir.
So you can use the catalyst up to 300 or to 400 degrees.
Yes, sir. Yes, sir. Yeah.
Okay, got it. Thank you.
Thank you, sir.
The next presenter is Ms. Diksha Luhan, Professor Reena Jain, Professor Anju Srivastav, and Dr. Shuparna Datta. Diksha, please. Diksha, we're not able to hear you.
Can we start the presentation? Diksha?
Am I audible now?
Yes,
you're audible.
Okay. I'm grateful to the organizers for giving me this opportunity to deliver my research presentation. I am currently working under Professor Reena Jain and Professor Anju Srivastav at Hindu College in Department of Chemistry. Today, I'm here presenting pieces of my research work, which basically focuses on novel design and synthesis of novel functionalized magnetic biochar-based materials for selective removal of heavy metals from different various wastewater streams. Just giving brief introduction behind my work. As we all know, water is a precious resource that have been utilized for drinking, agricultural activities, various household activities, industrial work. And all this work is done only with the 1% of the Earth's fresh water. But unfortunately, this 1% is being contaminated severely with various pollutants. In fact, 1.8 billion people are still drinking contaminated water, which causes around 5 lakh diarrheal deaths every year. About these various pollutants, there are many categories of water pollutant which include bacteria, protozoa, heavy metals, nitrate, gasoline, detergent, and many more. Our research is focusing on removing toxic carcinogenic heavy metals from wastewater. Although this slide just give brief introduction about heavy metals. I think we all know that. We can skip this. Now, when these heavy metals accumulate in our body beyond certain limits, they can cause severe harmful effects, which include different type of cancer, neurological disorder, skeletal defects, etc., and many more. So therefore, the implication is very clear. We really need to make effort to make this contaminated water clean. There are many methods to combat heavy metal pollution, out of which adsorption has emerged one of the superior choice for eliminating heavy metals from wastewater. And the key benefits of the adsorption technique is its cost effectiveness. It shows high removal efficiency for heavy metals, and it shows rapid kinetics, less requirement of control system, etc. Coming to the choice of adsorbents, although activated carbon is one of the extensively employed adsorbents, but over the years, a number of low-cost adsorbents have been designed from agricultural waste material and subsequently applied for the removal of heavy metal from metal-contaminated water. Every year, tons of agricultural waste, rice, wheat, stubble is burned open in air, which causes harm to our environment. So we are converting this biomass burning into some useful adsorbent to use that adsorbent for heavy metal adsorption from wastewater. The conversion of biomass into biochar is produced under limited supply of oxygen, and this biochar material has many useful characteristics, including high surface area, high porosity, lots and lots of functional group, high absorption capability, and it has lots of nutrient content, both micro and macro nutrients. Over the years, biochar has been extensively used in previous years to remove heavy metals from wastewater, but the poor subsequent separation of this powdered biochar limits its large-scale application to the wastewater treatment. So to end this problem, magnetization have recently been introduced into biochar matrix to form magnetic biochar. As a result of these magnetic properties, the magnetic biochar can easily be attracted to an applied magnetic field and separated easily. Even though magnetic biochar have been utilized in most of the research articles for pollutant removal, however, there is not much focus on the effectiveness and selectivity of these materials. So in our laboratory, we are synthesizing those materials by incorporating an appropriate functionalizing agent to engineer the surface of magnetic biochar with suitable linkers and grafting chelating ligand that could show some good selectivity and effectivity for particular specific metal ion. The surface of magnetic biochar can be tailored with either doping off another metal onto the surface of magnetic biochar and functionalizing it with some organic molecules, nitrogen, oxygen-based molecules. For example, chitosan modification of magnetic biochar has increased its effective adsorption capacity from 30 to 120 units. In our laboratory,
you have 1 minute. You have 1 minute.
We are synthesizing such effective and selective adsorbents for wastewater treatment, and this is the step-by-step synthetic strategy I have used. We are using in our laboratory to synthesize those surface engineered magnetic biochar. And further, in order to acquire structural insights into the material developed, we have adopted a set of spectroscopic techniques, that is XRD, FTIR, SEM images. We can see in the XRD graph, the diffraction peaks appeared at 2 theta value of 41 degrees, 53, 65, and 74 degrees, confirm the incorporation of magnetic particles on biochar matrix. Further, in FTIR, after modification of surface of magnetic biochar, the appearance of various functional group peaks confirmed the successful grafting of chelating ligand onto the magnetic biochar surface. Finally, the SEM images of biochar and modified magnetic biochar are clearly showing the incorporation or embedding of the magnetized particles onto the surface of biochar. In order to optimize our material check effectiveness and selectivity of the adsorbent, we are in the process of doing these following studies I have shown in this slide. Some of the work is presented here. As we can, if we look at the pH versus percent removal efficiency graph, we can clearly see the modified magnetic biochar has shown very high adsorption capacity for chromium removal as compared to the pristine biochar. In future, we are extending to we are hoping to extend our work to the real samples. Like we have already collected a few samples from near Delhi sites like Yamuna River site, and there is Bhalsa site, which is heavily contaminated with the heavy metals. Also, we are intending to use these heavy metal adsorbents as catalysts to further degrade pesticides and harmful dyes, etc. These are my highlights of my protocol. We are developing and developed a few adsorbent which show increased adsorption capacity and selectivity for particular specific heavy metals. They are easy to separate from the treated solution. They have remarkable reusability and which make them cost efficient. Thank you for your kind attention. Thank you very much.
Hello, Diksha. Good work and good presentation. So I would like to know two things from you. What was the property of this adsorbent? It was hydrophilic or hydrophobic that you have obtained from this XRD?
So both, actually. Biochar is itself in carbonaceous material, and there are lots of functional group, OH, COH, carbonyl. There are many functional groups which are very much high, which are modifiable actually, and we are working on the modification of the structure. So we can say hydrophilic.
So what you have prepared, what material you have prepared, that is hydrophilic or hydrophobic?
Hydrophilic, sir.
Hydrophilic. Okay. So my next question is, you have shown one same image, right? So what was the scale bar there? How much scale bar you have used for that same analysis?
So 3 kx actually. 3? I have skipped the details. Details are not giving me the picture.
Okay, anyway, you have a thought on that.
I'll share you.
Yeah, sure. Two things you need to see regarding this same image. One is scale bar, another is magnification ratio.
Yes, sir. Yes,
okay.
Yes.
So two things are different. So have a check in that. Thank you.
Thank you so much for your reviews.
Next oral presenter would be Mukaddam Khan Nurmotova and Dilfuza Zaborova. Do we have them today?
They are not present.
They are not present. Okay. The next presenter is going to be Zanigor Abdul Malikova and Dilfuza Zaborova.
Absent.
Absent. Okay. We move to the 16th oral presenter, Mary Abraham.
Absent.
Absent. Okay. Then the next presenter, oral presenter, number 17 is Ujjwal, Dr. Devanshi, Professor Anju, Professor Reena, Dr. Dinesh, and Dr. Shriparna Datta.
Good afternoon, ma'am. Am I audible?
Yeah, you're audible.
Okay. Ma'am, is my screen visible?
Yes, it is very much visible. You can start.
Okay. So, good afternoon, everyone present. I, Ujjwal Kumar Tripathi, a third year undergraduate student from Physical Sciences with Chemistry, Hindu College, University of Delhi, is here to present my research work entitled Environmentally Benign Synthesis of Xanthine Derivative and Their Application in the Detection of Heavy Metal Ions using Fluorescent Enhancement Strategy. Yeah, so in this slide, I was going to tell about the hazard due to heavy metal pollution, but we have already seen in Diksha ma'am's slide about it. So the main manifestations are carcinogenesis, deformity, and mutation. Therefore, the monitoring of heavy metals is a prerequisite key to prevent and control heavy metal pollution. So how do we do that? So here's a proposed sensor, xanthine derivatives, which are the fused benzenoid systems having heteroatom oxygen present in their carbon skeleton. They have significant pharmacological properties such as antibacterial, anti-inflammatory, and antiviral, but they have luminescent properties too. It is found that they provide high quantum yield, great photophysical properties, and low photo-bleaching effect. There were several reported methods for the synthesis of xanthine derivatives. However, they were associated with several problems like use of halogenated hydrocarbons, volatile solvent, low yield, and no reuse of catalysts. So we have tried to synthesize xanthine derivative by greener approach using greener solvent and mild reaction conditions, thus following the principle of green chemistry and hence contributing to sustainable chemistry in our own way. Here the scheme 1 shows 1,3-diketone fused benzenoid phenolic derivative and aldehyde present in acidic condition under proper heating forming xanthine. We have standardized the reaction by 2 methods, one using organic acid PTSA and other using mineral acid concentrated sulfuric acid. We have used a combination of water ethanol cell system, which is a benign solvent and able to achieve good yield of desired xanthine derivative in short reaction time. The next step involves the protection of free hydroxyl group of xanthine by protecting reagent. I'm happy to share that after persistent efforts, we were able to achieve the desired product xanthine derivative within 5 minutes by using solvent-free grinding technique in presence of an environmental friendly catalyst like potassium carbonate. In this stage, we can also see we have used another base, triethylamine, and the yield was also good in that method. But our scheme was designed in such a way to follow the principles of green approach. The target molecule was synthesized involving greener solvent or even solvent-free conditions. One pot multi-component synthesis was also used to reduce the waste and low reaction time, thus following the principles of green chemistry. Synthesis of the target molecule was analyzed by TLC and characterized by different spectroscopic techniques like 1-NMR, 13-NMR, UV absorption, IR spectra. All the above mentioned techniques confirm the synthesis of xanthine derivative along with the production of heteroatom in the same. This slide shows the TLC that we have received in our work. This slide shows the 1H NMR of the target compound, and we can see a typical peak around 5.5, which indicates the formation of xanthine derivative. Here we can see 13-NMR, UV absorption, and IR spectra. Here in IR spectra, a peak occurred at 1731 centimeter inverse, indicate the synthesis of a target that is protection of xanthine moiety. Why I'm telling you this because our approach of designing the sensor is based on the deprotection of xanthine derivative, which may occur in the presence of heavy metal ions like mercury to give fluorescence. Our work is still in progress, but our next step will be the deprotection of oxygen atom on reacting heavy metal ion to give fluorescent xanthine with free hydroxyl group. Thus, measuring the intensity of the fluorescence, the contamination of metal ions can be calculated. These are the few references that we have used in our research work. And with this, I would like to conclude. Thank you, Professor Anju, Professor Reena ma'am, Dr. Devanshi ma'am, Dr. Dinesh and Dr. Shipanna ma'am for guiding me in my work. Thank you to the organizing team of EWASH for providing me the platform at undergraduate level to show my work. Thank you, everyone.
So thank you, Ujjwal. You are doing UG project right now?
Yes, sir.
Yes. So I would like to know 2 things from you. That is, first one is what is the motivation behind the study? And the second one is what is the future prospect of the study?
The motivation behind studies, as we already know, the heavy metal pollution is like a very increasing and alarming rate by industrial waste, water effluents, the agriculture runoffs, vehicular emission. Every day the pollution levels is increasing. So at somehow we are desiring to develop a small, not a small, but the greener method to detect the level of contamination for the
Fine.
same. And the future perspective that it first of all it is the environmental clean synthesis, so the use of boxes reagents are ignored. And secondly, sir, the probe that we are using is the organic moiety, and which can be used with great sensitivity and with very easily equipation, because the previous techniques that we are using like till now, like absorption spectroscopy or emission spectroscopy, the equipment cost is high, multi elements cannot be detected in a single runtime. So we are developing a technique which can solve those flaws from those techniques.
Thank you. Okay, got it.
The next
Thank you,
oral
sir.
present, the next oral presenter is Miss Soni Rani, Dr. Devanshi, Dr. Soma, Professor Anju Srivastav, and Professor Reena Jain.
Good afternoon, everyone.
Good afternoon, Soni. We can't hear you, Soni.
Soni Dubey, are you present there?
Yes, sir. I'm just sharing my presentation.
Sure. Soni Dubey or Soni Rani? As per the.
That's Soni Rani.
Okay, okay, okay.
Yeah. I think. Sorry, sir. Like some problem is there.
So in the meantime, we can take the next presenter and then we can come back to her later.
Okay, ma'am, no problem.
In the meantime, you can just check your technique.
You try sharing your slides. You can share your slide.
Okay, try. Yes, Soni, it is visible now. You can start.
Okay. Good afternoon, everyone. My name is Soni Rani, pursuing my PhD from the Department of Zoology, Hindu College, University of Delhi, under the guidance of Dr. Soma Murai and Dr. Devanshi Maggu. Today, I'm presenting a small portion of my research work, and my topic is in silico targeting of gamma aminobutyric acid receptors by the green synthesized spirobarbiturate as a potential drug for behavioral disorder. This is the content which is covered in this presentation. The first question is, what is the need to synthesize the novel spirobarbiturate? Actually, the spirobarbiturate is a derivative of the barbiturate, and barbiturate is a drug that causes anesthetic effect positively modulate allosterically GABA receptor. And its uses has been discontinued due to their heavy abuse. Hence, there is a growing interest to develop a derivative of the spirobarbiturate that have both therapeutic benefit and less undesirable side effects. So, one such class of barbiturate that is synthetically formed by a closed ring at the fifth carbon position is called a spirobarbiturate, and have been shown to possess various biological functions as anticonvulsant, hypnotic, anesthetic, and anti-cancer activities. In this study, we synthesized the spirobarbiturate. The green approach studied their druggability quiescent. Now the next slide is synthesis of the spirobarbiturate. Thus,
Tony, your voice is breaking a lot. We can't hear you properly.
Now,
same. We can't hear you. She's there or she has left?
So we're just taking out.
She is in the meeting.
Is there?
Yeah, she is in the meeting, but not sharing the slide, I guess.
So what do you suggest?
Yeah, we can move on to the next one.
Next one. Okay, sir. Next oral presenter is Miss Anita Dubey.
Yes,
Mr. Shailendra Kumar, Professor Reena Jain,
Actually,
and Dr.
ma'am, this is Sony. The network issue has been resolved. Can I start now, please?
Okay, fine. So Anita, you just wait for some time. Let Sony complete and then you will resume.
Okay, sir.
You're there, right? Okay.
yeah. Okay. The synthesis is pyruvates. Step 1 is the condensation. Condensation of the substituted benzaldehyde condensed with the 1,3-dimethyl butyric acid, and it forms the alkene in the presence of ethanol and L-proline at 50 degrees Celsius. Ethanol acts as a solvent and L-proline acts as a catalyst. Now the second step of the reaction was carried out using the produced alkene and triphenylphosphine and diethylene acetylene dicarboxylate, and it produced this pyruvates in the presence of CPME at room temperature. Now these are the different conditions for the optimization of the reaction, like using the glycerol at the room temperature or PEG 400 at 50 degrees Celsius and CPME used at a room temperature. Our work has been focused on developing a robust, efficient, and sustainable protocol of the functionalized pyruvates. After the compound synthesis, the synthesized compound was subjected to in silico study to the molecular docking tools and molecular dynamics stimulation. As we know that molecular docking is used for the prediction of the binding affinity and the identification of the ligand correct binding geometry in the binding site. So we did the molecular docking of 4 pyruvates, different neurotransmitter receptor like glutamate receptor, GABA receptor, NMDA receptor was performed to predict their binding affinity and detailed interaction. The docking was performed using the InstaDock, and binding affinities between the ligand and protein were calculated using the quick. This docking this is a dock is a all pyruvates compound and present binding exhibit these compounds and help.
I think she has left.
Yeah.
So shall we wait or we'll call the next one? Anita.
Next, we have Miss Anita Dubey,
yeah.
Mr. Shailendra Kumar, Professor Reena Jain, and Dr. Chirashri Ghosh. Miss Anita Dubey.
Yes, ma'am. I present. Ma'am, my screen is visible?
Yes, it is visible. You can start.
Okay, ma'am.
Yeah, you start, please.
Yes, ma'am. Yes, ma'am. Good afternoon, everyone. Thanks for giving me opportunity to present here. I am Anita Dubey, Department of Environmental Study, University of Delhi. I am going to present my research work entitled Nanoparticle Synthesis from Leaf Litter Extract for Dye Degradation.
Yes, Anita, you can move to next slide, please. Yeah.
Yes, ma'am. In 1968, Champion and Seth reported that in India, many types of vegetation are found. There are 5 most important vegetation. These are tropical evergreen forest, tropical deciduous forest, tropical thorn forest and shrub, mountain forest and mangrove forest. In this work, we have chosen North Ridge of India because of Delhi has a arid and semi-arid zones have a thorny shrub vegetation. The North Campus of Delhi, having different colleges, is blessed with rich vegetation and many departments possess a compulsory green belt. Due to a rich vegetation, high litter fall is observed. Dry leaves generated a considerable amount of solid waste every day. Organic material continued to be the largest component of municipal solid waste. As per record, 15% of municipal solid waste is garden waste. Leaf litter is major component of garden waste. In this time, disposing of leaf litter is major concern. Conventional method for disposing leaf litter include burning and landfilling. These processes give up many toxic or hazardous gases like PM10, PM2.5, carbon monoxide, carbon hydrocarbon dioxide, and methane carbon dioxide and volatile organic substance that have harmful effect on human as well as environment. What is nanoparticles?
Anita,
Yes,
Anita,
yes,
what I suggest you that all these things are pretty known to everyone. These are the general things. Okay, you just switch over to your own topic. That what you have done. That will be better.
Okay, sir. That is why nanoparticles are used for the removal of heavy metals and dye degradation. And plant extract has been demonstrated to provide some of the best capping and stabilizing property for the synthesis of nanoparticles, which helps us. And its advantage is eco-friendly, cheaper process, and varied shape and size, and abundant production. All the primary pollutant environment has been severely affected due to the increased influence
Anita,
of
you can switch over to your own work, what you have done.
Yes, sir. This is the layout. And the framework of study, survey of sampling sites, nanoparticle synthesis and characterization of nanoparticles and degradation of dyes. And in the current study, we focused on a few colleges along Delhi North Ridge as the site of the leaf samples. The most abundant leaf litter was collected, separated, and wetted and washed thoroughly and dried into the oven to remove all moisture of leaf. Dried leaf where the crust and powder to make the extract, and the aqueous leaf extract was added into the ferrous chloride solution, and after will a color of solution changed from pale yellow to intense black, which indicate formation of iron nanoparticle, and this solution dry to hot plate convert into black precipitation nanoparticle crust and increase in the fine powder. And selection of dye is EBT because of problematic dye is wastewater and anionic and water soluble, indicate in complexometry titration for dyeing nylon, silk and wool fiber. Procedure is 50 ml of 35 ppm dye solution, add 40 mg of nanoparticles, second and stirrer, and taking absorbance and different wavelength. And lambda max is 543 and degrading iron nanoparticle concentration of EVT is 4 mg per liter. Result and discussion. FTIR shows in nanoparticle peaks absorbed at 416 to 424 centimeter inverse vibrational transition of ferric oxide. Certain peak at 1030 to 1040 centimeter inverse carbon oxygen and carbon stretching, and peak at 1370 to 1400 centimeter inverse plane blending of hydroxyl ion. Some indices confirmed the amorphous nature of nanoparticles, and diffraction, X-ray powder diffraction was shows peak at 2 theta at 33.09 to 43.48, depicting formation of iron oxide. Silent features of this study: in color of almost transparent indicated decolorization of sample, degrading of dye attribute to iron oxide presence in the nanoparticles, and ferric which form phantom catalysts oxidize and degrading the organic dye. Size of nanoparticles play a great role in degrading the dye sample. Pogamia pinnata best performance due to small size, higher secondary metabolism, and Kingelia africana had size around 70 nanometers, probable reason for this performance. The research concludes that sufficient amount of waste is generated that create hawk in environment as it one leading to increased air pollution. The study conclude that leaf litter waste for synthesis of nanoparticles is approaches for sustainable development. Different nanoparticle synthesizers have different size and efficiency in degrading commercial dyes. So we can deal with air and water pollution. References of the study and acknowledgement of Environmental Pollution Lab and Reena Jain, Hindu College, University of Delhi, IOE, and Department of Environmental Study for providing financial support for the study. For advanced instrument analysis, author gratitude Mr. Harsh, Dr. Manvi, and Dr. Sonalika for their help. Thank you so much.
Okay, Anita, I would like to know that the study what you have shown, what is your future prospect in that? Because this kind of study has mostly been completed.
Sir,
So what is your
I
future
have
aspiration?
read, sir, I have many read articles. We can use fresh leaf earth using synthesis of iron nanoparticles, but very few articles used for leaf litter for the synthesis of nanoparticles. We can choose leaf litter as a waste, convert to the iron nanoparticle.
For iron nanoparticle, you are
Yes,
concentrating.
sir. Yes, sir.
Okay. Okay, fine.
Okay.
But anyway, I'll suggest you to have a better, like preparation in your future. So I hope in which year you are in PhD?
Sir, environmental studies.
No, in which year?
First year, sir. First
Year.
time.
Okay, okay. So anyway, no problem. You're having a bit less confidence, but anyway, you will improve it. Okay. Anyway, thank you.
Okay.
The next presenter would be Ms. Sudha Acharya and Ms. Vaishali Mishra. And in the meantime, Ms. Soni she would like to complete her presentation, so she can wait for another 5 minutes.
Sudha is not there.
She's not there? Okay, then Ms. Soni, please kindly come and complete your presentation.
Soni, are you there? Soni Rani, Miss Soni Rani?
No, she is not there, sir.
Okay.
I think she has left.
I think Vaishali will be there. Vaishali, I think, will be there. No?
Who?
I think she's joining. Soni is joining.
So we can proceed to the poster presentation. Then after she will speak.
If
it's okay.
Yeah, Miss Vaishali, ma'am, Shipra ma'am. So we can just wait for a minute and then we can just.
Sure, not a problem.
Yeah. Miss Vishali, Miss Soni.
No, Vishali is also not there. Vishali and Sudha are not present.
You can make the Sony as a co-host. She is there. I think she already joined.
Manoj, just make Sony as host again, co-host again.
Done, ma'am, done.
Hello, Sony this side, sir. Am I audible now?
Yes, no
You're
sign.
audible, Sony.
Okay.
Okay, thank you again for giving me this opportunity to finish my presentation.
Yes, you can start from there. Docking.
Now we discuss about the molecular docking. Molecular docking is used for the prediction of the binding affinity and identification of the ligand correct binding geometry in the binding site. Now we did the molecular docking in the 4 spirobarbiturate compounds with the neurotransmitter receptors like glutamate receptors, calcium channel complex, GABA receptors was performed to predict their binding affinity and detailed interaction. The docking was performed using InstaDock, and the binding affinity between ligand and protein were calculated using the Quick WinRW. Now this is the docking result of all 4 spirobarbiturate compounds and presenting a binding energy with the GABA receptor. Here we can see that para-methoxy spirobarbiturate exhibited the highest binding affinity, that is minus 8.8 kilocalorie per mole with the GABA receptors, and it shows hydrogen bonds, and these are the amino acid involved in the hydrogen bond. Similarly, these 4 compounds docked with the NMDA receptor, and here the parafluorospirobarbiturate has the maximum binding affinity with NMDA receptor. Now this 4 compound docked with the glutamate receptor, and here orthochloro-sp and metachloro-sp showed the good binding B-score with the glutamate receptor. Similarly, this compound docked with the calcium channel beta 2 subunit. Here, orthochloro-sp and metachloro-sp showed a good binding B-score with the calcium channel, and at last, these 4 compounds docked with the proton-dependent potassium channel. Here, parafluorospirobarbiturate exhibit the highest binding affinity, that is minus 8.5 kilocalorie per mole. So, to summarize the molecular docking results, clearly indicate that parafluoro-sp showed the highest affinity with NMDA receptor, and methoxy-sp has high binding affinity for the GABA receptors. Further, to check stability, we went to the MD stimulation. So what is the molecular dynamics? Molecular dynamics is a computer simulation method for the analyzing the movements of the atom and the molecules. The molecular dynamics simulation of a spirobarbiturate with the GABA receptor was run to find the action and the binding site of the GABA receptors. Now this is the RMSD plot. The result analysis revealed that the system is quite stable until 50 nanoseconds for the orthochloro and methoxy spirobarbiturate. Observed a significant variation in RMSD with a reduction of structural stability for the rest of the stimulation, whereas metachloro-sp gained stability after 30 nanoseconds, while parafluoro-sp showed almost a stable configuration till 100 nanosecond with the GABA receptor. According to the RG plot, GABA receptor maintained stable RG in complex with the parafluoro-sp than the other 3 spirobarbiturate. The other 3 spirobarbiturate show a slight expansion in the GABA receptor. As we can see in this plot, Yellow color shows the parafluoro-SV with the GABA receptor. Now, the major stability interaction of SV with the GABA receptor were hydrophobic in nature, and the interaction can be grouped into 4 types. Here, the green color shows the green color shows the hydrogen bonding, gray color shows the hydrophobic interaction, and magenta color show ionic interaction, and blue color shows the water bridges. The subsequent diagram of the figure illustrates a timeline description of the main interaction. A dark hue of gray indicates that some residue may make many distinct contacts. At last, to summarize the MD stimulation, parafluoro-SV shows the highest binding stability with the GABA receptor. GABA receptor so that definitely spirobarbiturate act as a drug patient on CNS as a depressant. Now in future, we intend to take our in silico study to the laboratory, and we will continue this topic to synthesize potent heterocyclic drug and their derivatization to illustrate their bioefficacy as antimicrobial, and further studies to check antidepressive effect on neuronal cell lines. Comparative study between the above 2 aspects in their future studies. These are the references which is used to prepare this presentation. Thank you.
Okay, Soni, you have told that you have done some kind of molecular simulation, right?
Yes, sir.
So any kind of molecular modeling you have done?
Molecular modeling, no, sir.
So without modeling, how can you do simulation? That's why I did not see any kind of mathematical equation there, because after the mathematical modeling, the simulation will come. Some mathematical model you should have, right? Based on that, you can do the simulation.
Sir, actually, we synthesized some compound in the lab, and from the derivative of the carbohydrate, and just
then how can you go for simulation without modeling? That's what, there is a gap. So that's what I'm telling. So I mean, as per you, what is called simulation? Simulation is basically solving the mathematical equations in computer. That is called simulation.
Yes, sir.
So the thing is there any mathematical model you have used, or you have taken it from some literature that you have made validated in this purpose? Because I did not see any mathematical model here. Anyway, you do your research on that, and I hope that afterwards you will become more confident in that. You see that what specific mathematical model they are using. If it is in computer also, there should be some kind of mathematical model based on what the simulation result is coming. Okay.
Okay, sir. Thank you.
Okay, thank you.
Thank you very much, sir. Actually, I got a little scared, but yeah, I'll look into your reviews. Thank you.
Sure.
Okay, thank you, participants. I would like to thank our judges, Dr. Jyoto Nayak and
Yes,
Dr. Rabia
please.
Praveen, for making this oral presentation session highly informative and meaningful. I request our chairperson to give their remarks on this session.
Yes, the other chairperson is present, or?
Yeah, she's there. She's very much here.
Yes.
Yes.
Hi, Dr. Rabia. Yes, madam,
Yeah.
please
I
you
would like
start,
to
then I'll proceed after you.
Yeah. First of all, I would like to congratulate all the presenters who are present here and presented their presentation, their work, and few are very nice. We have given the marks according to their
Stuck.
Yeah, Rabia ma'am, you're not audible.
Dr. Rabia?
I think there is some, she has come. She's unmuted. She will have to unmute, no? Okay.
Sorry, there's a network problem here. Sorry. Am I audible?
Yes, ma'am.
Yeah. So
Ma'am, you're not audible again.
I think there is some problem with the net.
So all
Yes,
you
ma'am.
have presented well. Sorry. Okay. Thank you, everyone. Now, sir, please over to you.
Okay, fine. Not a problem. Anyway, some good works we have seen today during this technical session. And particularly for me, that scholar's presentation, Pooja Rana, that was the best one for this session at least. But all the presentations were good. And but what I
Sorry,
have
Jeto,
seen,
you are not supposed to tell which is best.
no, it's okay. We have given the marks, but
Yeah.
her presentation was the best one.
Yeah.
I feel. So anyway, all good luck to all the participants here, and also I'm much more thankful to Dr. Shipra Mishra, Dr. Richa Tyagi madam, and Raghavi Kattar madam. So thanks for a beautiful introduction of mine, and Dr. Shankar Chakraborty and Dr. Shipra Mishra for giving me this opportunity to session chair. Okay, thanks a lot. Thanks to you, everyone.
Thank you, ma'am and sir. Now I request Dr. Neha Chaudhary to present the vote of thanks to our chairpersons, invited speakers, and participants. Over to you, Neha.
Thank you, Richa ma'am. A very good afternoon to all. I, Dr. Neha Chaudhary from Hindu College. First of all, I would like to propose a vote of thanks to our Honorable Chairperson, Dr. Suraj Kumar Tripathi and Dr. Maulin Pisha for gracing such an incredible and enlightening session. I would also like to thank our esteemed speaker, Dr. Pinaki De, Dr. Jayato Nayak, Dr. Aarti Bhatt for making excellent presentation and making this session very stimulating, expressive and educative. We are very thankful to our chairperson, Dr. Rabia Praveen and Dr. Jayato Nayak for judging our oral presentation session and making it very meaningful. I would also like to thank all the participants for such a wonderful oral presentation and yes, audience. Last but not the least, a special thanks to organizing committee for their continuous support and coordination. Now I would like to invite Dr. Meenu Srivastav, Ms. Manisha Chawla and Dr. Priyanka to conduct next technical session. Ma'am, over to you, ma'am. Meenu, ma'am.
Thank you, Dr. Neha. Thank you, Dr. Raghavi, Dr. Richa. It's a great pleasure to coordinate this technical session of EWASH 22 along with my colleague Manisha Chawla. I'm extremely delighted to introduce the two chairpersons of this session, Professor Rajendra Singh Chauhan and Dr. Usha Panjwani. Professor Rajendra Singh Chauhan, I think he's there. Is he there?
Not yet, ma'am.
Not yet. And
Usha ma'am is here.
is she there?
Yes, ma'am.
Yes, I'm here. Okay, ma'am. Okay, ma'am. So I would start with
Dr. Meenu, can I interrupt?
Ji, ma'am.
Actually, Professor Rajendra Chauhan is not able to join. So I think Usha ji आपको ही करना पड़ेगा. So I think you will be able to manage. दो हैं हमारे. Sorry
Okay,
for
ma'am.
that. But last
No
minute he
problem.
had to.
No problem, ma'am.
Okay, thank you.
Okay, so I would like to first of all introduce our only chairperson for this session, Dr. Usha Panjwani, who is scientist G at Defense Institute of Physiology and Allied Sciences, DePass, DRDO. Dr. Usha has leading research in life sciences for defense applications. She has significantly contributed in the area of sleep and cognitive function at high altitude and its mechanisms, and her research has made a great impact for maintenance of cognitive during sleep deprivation in the armed forces. She is a recipient of many illustrious awards and honors for her contributions in the research for armed forces. Rear Admiral M.S. Malhotra Award for her best paper in neurosciences is one of them. She was also awarded as a team member for product development aimed at improving life of persons with disabilities from Ministry of Social Justice and Empowerment. Dr. Jain Moitra Memorial Oration and Dr. Sushila Thakur Prakriti Mandir Prize for best paper in the field of yoga by the Association of Physiologists and Pharmacologists of India are some of them. She has to her credit a wide number of papers in journals of high impact. In fact, we welcome you, ma'am, here in this session.
Thank you.
To act as chairperson. We have two invited talks in this session by two
Yes.
eminent speakers, Professor Shubhankar Suman and Dr. Girija Bharat. I request you, ma'am, Dr. Usha, ma'am, I request you to please introduce Professor Shubhankar Suman and invite him for the talk. Thank you.
Okay, thank you so much, organizers, especially Ma'am Shipra Mishra. So I think according to the schedule, the first speaker is Dr. Girija.
Okay, you can start with Dr. Girija, ma'am.
Okay, okay. So yeah, so welcome to this session, everyone. And I have a great pleasure in introducing our first speaker of the session, Dr. Girija K. Bharat. She is the founder director of Mu Gamma Consultants Private Limited, Gurgaon. She's an international expert with 30 years of experience in water quality, and she conducts research and capacity building activities in India on water resource management, water supply and sanitation, chemical pollution control, and environmental management. She started her career as a scientist in CSIR, and she was a presidential scholar at George Mason University, USA. She has worked with many institutions like TERI, USAID, World Bank, and she has several publications, 103 national, international, and it also includes the Global Sustainable Development Report 2015 and 16. She has co-authored National Policy Framework of India on Safe Reuse of Water. So she has all these achievements to her credit. She is the recipient of several awards like Glory of India 21, Women Transforming India Award by Niti Aayog, and several others. So we welcome Dr. Girija K. Bharat and request her to start her talk.
Thank you, Dr. Usha. Many thanks for the kind introduction. I hope I'm audible.
Yes, yes.
Yeah. Thanks to Shipra ma'am for this opportunity, and always a pleasure to be associated with Save the Environment. It has been a great platform, and everybody knows that. So with that, I would like to start discussions about what we have done in this national policy, which you were discussing about. As you all know, recently on 5th of January, the national policy of India was launched by the Honorable Prime Minister and Water Minister. It has many nuanced sections, and it was fleshed out in 2020 during the COVID and 2021. Several rounds of stakeholder consultations. So I thought I would better discuss about this SRTW framework. We call it SRTW, Safe Reuse of Treated Water. The basic intent of forming this framework was to have fresh water conserved, because if we are able to reuse wastewater, it gives three benefits. Number one is it augments fresh water, and water resource which is already very stressed, that is being taken care of. Water quality, which is basically affected due to wastewater getting released into the environment, I mean river, lakes, even ocean. So if you are reusing this, that quantum would get reduced, and the pollution load to the marine environment, to the riverine environment also gets reduced. Fresh water is actually, the cost is increasing, and besides the environment and health benefits, it also would give scope to recharge lakes and wetlands. It is a great economic opportunity where we don't call it wastewater anymore, we call it used water, because it's a resource. We have to see it as a resource rather than a waste. So in the light of circular economy, reuse also provides revenues for treatment. I will be giving one case study at the end of this presentation where I will be discussing about Surat as a exemplary case within India. This is also contributes to the recovery of nutrients, which could potentially reduce the import of fertilizer. I am thankful to GIZ, India EU Water Partnership, National Mission for Clean Ganga, for giving this opportunity to me. And it is a great opportunity for me. The guiding principles for SRTW policy and administering these things are basically adopt a participatory planning process and provide clarity on priorities for allocating treated used water on social, economic, and environmental benefits. It would encourage the uptake through provisions on mandatory reuse, and we need to have water quality standards established that are fit for purpose. This would also set a competitive price for treated used water that is attractive to users and available, and it is sufficient for sustainable O&M. It would also adopt a risk-based precautionary approach and adopt incremental implementation commensurate with time to build capacity. It would incentivize compliance, address any of the issues that is related to prior use of water, basically wastewater, to manage the transition and encourage long-term commitment and stability to the supplies. This is also to basically move India on a pathway of mainstreaming SRTW by 2022 by encouraging states to adopt the necessary enabling environment. This is also to view SRTW as part of the wider water cycle and Government of India's commitment to environmental stability and achieving Sustainable Development Goal 6.3, which is on water quality through increased recycling and safe reuse. We also have tried to have the roles and responsibilities assigned by various government entities. We have seen that most of the time, any of the policies would remain as a policy without getting implemented because the roles and responsibilities are not well defined. We also have tried to give business models and establish a new funding mechanism and support synergies with ongoing programs such as Amrit, NMCG, the Swachh Bharat Mission, Jal Jeevan Mission, etc. This is basically the river basin planning, which includes the potential of SRTW within the catchment of water cycle and clearly entitlements to water. This gives a broad picture of SRTW within a wider policy. The purple lines, as you see, is the treated used water, and the brown lines are the used water, brown arrows, and the blue ones are non-potable, and the light blue are the potable reuse. This policy has basically been involved in with and aligned with many of the policies, as you see, National Environment Policy, National Urban Sanitation Policy, National Water Policy. In fact, the new National Water Policy of 2022 is also coming up, and we have contributed a para on safe reuse of treated water in the National Water Policy. It also is aligned to the FSSM policy, that is fecal sludge and septic management, and Atal Bhujal Yojana, Amrut, Jal Jeevan Mission, as I mentioned, NMCG's ongoing efforts, NRDW, that is the Rural Drinking Water Program, Prime Minister Krishi Sanchay Yojana, and few others like Swachh Bharat Mission 1 and 2. The standards and compliances in India, you must be aware of that we are quite lax when it comes to standards and compliances. We are not in a position to give a lot of attention, but at the same time, we have to see that the norms are maintained, not to be a thorough perfection, because if we go for perfection, it will be a non-starter at all. We have to be mindful about National Green Tribunal's oversights, because they are the agencies which keep these compliances and implementation challenges in place and checks. The sources of water for reuse has been covered by SRTW policy framework, where it is only dealing with STPs, sewage treatment plants, not the industrial wastewater, because industrial wastewater is reuse policy is being separately developed by GIZ and Central Pollution Control Boards. The potential areas of reuse, as you see, are industry, agriculture, municipal users such as landscaping, dual plumbing, and not the potable ones. And environment, which includes discharge into surface water bodies and maintenance of wetlands. Aquifer recharge is a no at this moment, like the potable water. We are not considering at this stage of compliance and level of achievement of India's capability of aquifer recharge anytime soon. But as we improve our compliances, our systems, then we can, in future, we can consider aquifer recharge. Construction is also a great industry and which is coming up. In fact, the ADB report says that 50% of construction is not done yet. 50% of 2050's construction is still not done. That means construction is an area where we have a lot of scope of reusing this water. There is on-site use within STPs, including the landscaping and cleaning of desludging vehicles, etc., where this could be used. We are talking here in terms of secondary treated water, because if we do it to the tertiary level, the cost will be very, very high. Nevertheless, we have left it to industries who can buy this wastewater, and then at their own expense, at their own cost, they can do the tertiary treatment, which will be beneficial for the respective industries. Having said that, many of the industries actually don't need tertiary treated water. Mostly secondary level is good. For agriculture, I must say that we are using secondary level. To tell the truth, in fact, there are places where it is directly treated, without treatment, the sewage water is being used. So all states, that is 12 states, are already having their SRTW in place. Some are basically in draft stage, some are in final stages. But then all states should adopt after this SRTW national framework has been made. We have put a section where we have put a framework for the states. And it is in the interest of the state that all states adopt and start implementing the state SRTW policy by the end of 2022 was what we had suggested, but now it has got revised to end of this year, 2023. And the states with already existing policy should align their policies with this SRTW, because it's always good to have a level playing ground. Having said that, there are many states which have done quite well. Some states have not even begun well. So we need to have a level playing ground for different states, and the ones who are already having systems in place, we would suggest that the treatment and collection capacity be made very high, say 50% and then 100% for sure. Additional targets for individual STPs can be set so that there is 50% reuse for after establishing a STP and 100% reuse within 10 years. These are ambitious for sure, but then nevertheless, if you don't keep these ambitions, we will not be able to achieve even a fraction of it. So basically, this policy is expected to yield significant benefits in terms of water security, health benefits, environmental benefits, social benefits, economic benefits, innovation and efficient technologies, increased capacity in ULBs to implement SRTW projects, including cross-sector collaboration, and reduced climate emissions. Having said that, we have to be also mindful that we in India have a very high non-revenue water, because it's treated water which is getting leaked into the environment because our water use efficiency is bad, or if there is leakages in the system, and it's something around 42 to 50% of non-revenue water from different states. We have also identified, as I had mentioned earlier, the roles and responsibilities of different ministries. Jal Shakti for sure. Jal Shakti is the one which has had assigned us this job of doing it, National Mission for Clean Ganga. Ministry of Housing and Urban Affairs, Niti Aayog, National Council on State Committee on SRTW, Central Pollution Board, Pollution Control Boards, CPHO. They also have their roles to establish end-use water quality standards for different uses. In the state, we envisage the responsibility to formulate and implement state policy. ULBs and Panchayat Raj institutions responsible for on-ground implementation, and pollution control boards of the states would have regulatory role in monitoring and compliance. The industries are expected to have proactive engagement on planning of SRTW projects. Well, water quality is an extremely important aspect, and it has to be treated, as I mentioned, to secondary level. And we have to have considerable work required to establish full range of standards, and the standards would be fit for purpose. We need to follow a risk-based approach. That's what we have done in this, and dealing with emerging contaminants, because emerging contaminants like the endocrine disrupting chemicals, persistent organic pollutants, even micropollutants, microplastics, they are coming under the risk. They are huge health and environmental risk, which we often don't pay much attention to. There is strict discharge norms on BOD, COD, total suspended solids, and TDS, etc. So reuse in agriculture, we have to be very sure that it meets standards so that we don't end up with food which is substandard, food items which is substandard. The quality for reuse in industry is very site specific, and then we can think of good models where industries could take this, and the financial responsibility and financial feasibility when we sell it to industries is much better. That's what we have seen in Tamil Nadu, we have seen in Surat, that industry, wherever the uptake is done by industry, there is good recovery of O&M for sure, entire O&M, large extent the capital cost also. And we, considering that we also just got over, I can't say got over, it's still on the pandemics. So tomorrow maybe some other pandemics might emerge. So the risk with pathogen has to be very, very well defined. We have to prepare this action plan of SRTW, and we are in fact doing one implementation project in Panipat, and we have seen the challenges which the implementation of SRTW is taking. The challenges, one of them is basically overcoming public reluctance, communicating the right way, right stakeholders, and the right level of communication. It has a big chance of not able to steer past the financial aspects, because if the financial viability is not there, we cannot encourage alignment of interest. So we have to be sure that there is fund allocated for this, or at least earmarked, and then subsequently it can be increased. There should be stress on compliance and standards, and integration and coordination with multiple agencies and actors has to be very, very strong. We have seen this in the case of Tamil Nadu and Surat and other states where it is able to take a good transition. It is able to make this transition. We have basically brought out 10 business models in this. I'll not go into the details of that in the paucity of time, but then the business model provides a guidance on selection and how it can be, if it can be hybrid annuity model, HAM we call it, or DBOT, which is design, build, operate, and train. And in fact, training of farmers, FPOs, farmer producer organizations is also very, very important when it is dealing with agriculture. The state should mandatorily have a committee for sure and approve business and contract model. One size fits all doesn't work, because different ULBs within the state itself are quite varied, and we have to address this plurality, multi-plurality in this sector of reuse. So the public health and water quality considerations, when we are talking about it, we are talking about turbidity, color, the physical water quality considerations, the chemical water quality considerations such as chemical constituents, including solid metal, nitrogen, phosphorus, etc. Pathogens in the biological water quality, which includes bacteria, helminthes, virus, etc. And emerging water quality considerations, because we are seeing a lot of pharmaceutical residues in drinking water. We in India we are still struggling with these things because many of the personal care products have many emerging contaminants, and we are not paying much attention to this. In the industrial sector, basically the water can be used for paper and pulp industry, which is a very, very high guzzler of water. In fact, most of them take groundwater, and some places which is not allowed to go beyond a limit because the water table is declining. But still, industries have acknowledged during our visit that they do drill and extract groundwater, which is actually a strict no. So whenever possible, other end uses of treated effluents such as recycling, cooling, power generation should be considered. I will briefly talk about the Surat case study. This is an exemplary case.
Ma'am, sorry to interrupt you. Can we wind up the
Yes,
lecture?
I have 2 more slides if you would allow me to.
Yeah, sure.
Yeah, sorry for that.
No problem.
Yeah, I will not go into the details of this, but then with increasing water demand in places like Surat, they have been able to get revenue of 150 crore per annum, and it has set an exemplary template for the whole of the country. 11 STPs are there, and the water from the STPs also go to the industries because Gujarat is a very industrialized state, and treated water from CTPs, that is effluent treatment plant, also goes to this industries, and it is highly viable option. So the conditions are to reuse 70% of treated water by 2025 and 100% by 2023, and Surat is all ready to do that. Some of the challenges have been basically I will talk about my last slide. I'll not talk so much about the challenges, but the learnings have been that Surat SRTW can be adopted in other ULBs in Gujarat, and it has structured approach. It has with success has reduced the dependability on river Tapi water, and more fresh water is available for other consumption, higher hierarchy consumptions like drinking and other uses. And there is significant reduction in surface and groundwater abstraction. And most ULBs in India which struggle for fund, Surat has set a great example by getting close to 150 crore per annum, which helps in its expansion. With this, I conclude. Thank you so much for the patient hearing. Thanks.
So thank you so much for such a nice lucid talk on water policy and safe reuse of treated water. And thank you also for finishing in time. I think we're running a little behind schedule. So if you permit, I will request the question answers after both the speakers have finished their talks.
Sure.
So yeah. Sure. So thank you. And now I have great pleasure in inviting Dr. Shubhankar Suman. I hope he's here.
Yeah. Good afternoon, Dr.
Good afternoon, Dr. Suman. So Dr. Suman is an associate professor, Department of Oncology, Georgetown University Medical Center, Washington DC, USA, and he's done his MSc in biochemistry from Banaras Hindu University and PhD from our very own INMAS DRDO. So, and he is a recipient of several awards like Radiation Research Society Earlier Career Investigator Travel Award, then Certificate of Outstanding Contribution in Reviewing from International Journal of Radiation Oncology, Biology, Physics. So there are so many of his achievements which I have with me, and he is also, he's the NASA Studies Section panelist and grant reviewer. Presently, he's the editorial board member of International Journal of Radiology. So, and he has several publications in high impact journals. So a hearty welcome to you. And this morning, he's going to talk about this afternoon. He's going to talk about space radiation-induced accelerated aging, associated cancer risk, and potential mitigation strategies. So welcome. We can start, Dr. Suman.
Thank you, Dr. Rosa, for such a nice introduction. I would also like to thank Dr. K. Sipra Misra for inviting me. As the title of my talk suggests, I am going to talk about space radiation and space radiation associated health consequences such as accelerated aging, cancer risk, and how we can mitigate these risks. So first of all, why space exploration is so important? So space exploration is very important to ensure the survival of humans and other species, plants and animals on the Earth in the event of planetary scale disasters such as asteroid collision or any full-scale pandemic or a nuclear war. In addition to this very important cause, space exploration also will add to the additional resources on Earth in case of, if there is an scarcity or need of a particular metal or element type which is not available on Earth that can be fulfilled from a space. And that these studies are also important to understand origin of life and climate change on other planets. Studies on climate change on other planets can help to study climate change on Earth or predict what is going to be in future for Earth in terms of climate change. And during the space exploration, there are so much of technologies involved, such as nanotechnology, telemedicine, robotics, and to make a space mission successful, we need to develop technologies related to all these areas, and that further helps for sustainable development of human beings on Earth. So there is a famous quote from David Greenspoon, and he says that as long as we are a single planet species, we are vulnerable to extinction by planet-wide catastrophe, natural or self-induced. Once we become a multi-planet species, our chances to live long and prosper will take a huge leap skyward. So there is a need for colonization of space, and NASA is planning to do that. And as per NASA plan, they are going to have manned mission to the Mars by 2035. So before sending man or astronauts to deep space mission or any interplanetary mission such as mission to Mars, we need to know all the health risks associated with space exploration. So 5 key space exploration related health hazards has been noted or projected, and among them, space radiation is the first one, which is perceived to be the most hazardous component of a space flight. And during the deep space mission, part of a space which is outside Earth orbit or where the Earth's magnetosphere is not available. So when a space flight crosses through this deep space, there is high likelihood of exposure from a space radiation. And 2 major sources of this space radiation is galactic cosmic radiation and solar particle events. Particularly in case of galactic cosmic radiation, there is a component of heavy ion charged particle. They can pass through the spacecraft shielding, and exposure to this type of radiation is imminent during deep space missions. So we need to know the health risks associated with heavy ion particle radiation and how we can overcome these health risks. So in terms of ionizing radiation, what we experience on Earth, it is quite different from what we experience in space. Particularly, there are 4 types of ionizing radiation. First is photons, which is X-ray and gamma ray, which is commonly used on Earth for diagnostic purposes like X-ray imaging or CT imaging, and also for radiotherapy of cancer patients. But the other radiation types, such as proton and heavy ion and fast neutrons, which are particularly present in galactic cosmic radiation, are more damaging because of their physical characteristic. Especially in case of heavy ion, they contain energy, mass, and charge. But in case of X-ray or gamma ray, they only contain energy. So due to presence of mass in these heavy ions, they are considered more damaging. And here is an image comparing gamma rays with iron radiation, which is a heavy ion particle. You can see that when heavy ion particle passes through a cell nucleus, it causes dense ionizing effect inside the DNA and causes lot of double strand break compared to sporadic damage from X-ray or gamma ray. So, as we know that there has been no manned mission beyond moon, so we need to know the health effects associated with a space radiation exposure, but there is no first-hand human data. So we need to have a ground-based research facility to simulate a space radiation, and which facility can be generated using linear accelerators to accelerate like particular ion particles to simulate space beams. And this facility is available at NASA Space Radiation Laboratory at Brookhaven National Lab. And they have 3 different types of accelerator to generate various space radiation types in this ground-based facility. And in absence of human data on space exploration or space radiation exposed human being, we are applying ground-based animal model approach. Particularly, we know from atomic bomb survivors who are basically exposed with the ionizing radiation, and we know the health consequences and long-term effect study has been done in Japanese atomic bomb survivors. So we know health effects of low LET ionizing radiation such as gamma or X-ray, but we don't know the health effects of space radiation. So in our animal model approach, we are exposing mouse or animal models to simulated space beams to study dose response and mixed field effects. And then we are developing model in combination with what we know from Japanese atomic bomb survivors. So particularly, we are using different mouse models, genetic and wild type mouse models, expose them with space simulated beams, and then we look for different pathologies. We are particularly interested in gastrointestinal tumorigenesis. And then we combined all these data and compare with atomic bomb survivors to model risk during the space mission. Particularly, we are focused on 2 major variables. One is the physical variable, where we are looking for dose response and dose rate studies, and other is the biological response to look into age and genetic heterogeneity. And final goal of our study to have a risk estimation after space radiation exposure and to develop some biomarker of these health risks and develop countermeasure to protect from deleterious effect of space radiation. So here is one study we did with mouse exposed to gamma radiation and with the space radiation, and we found that when we expose mice with the whole body of gamma and space radiation, there is a accelerated aging phenotype that can be seen with the skin graying in these mice with increasing dose. At the same time, at a very low dose of 50 centigrade or 0.5 gray of space radiation, we see a significant reduction of lifespan in wild type mice. So that clearly indicates that accelerated aging after space radiation exposure is also associated with reduction in lifespan. At the same time, using a genetic model of gastrointestinal cancer or colorectal cancer, APC 1638 mice, we did tumorigenesis study using different dosage of space radiation as well as with gamma radiation. And With different heavy ions which are present in space, we saw increased GI tumorigenesis, such as after helium, oxygen, iron, and silicon radiation. There is an increase in GI tumor formation with increasing dose, and which was very high compared to gamma rays. At the same time, cancer progression rate also increases with the LET or linear energy transfer of particular space radiation types. So carcinoma or tumor progression risk after space radiation exposure is almost tenfold higher compared to gamma radiation. So that further indicates that space exploration or space radiation exposure during space exploration carries a higher risk of carcinoma development later in life. We also did a longitudinal study at 2 time points when mice were exposed, and we counted tumors at 60-day and 150-day post-exposure. And then we translated this data into tumorigenesis rate. We found that particularly with the space radiation or space silicon ion exposure, tumorigenesis rate was much higher compared to control and gamma ray exposed mice. That further indicates that exposure of a space radiation poses a persistent oncogenic pressure on these mouse models. In addition to increased tumor formation and cancer progression, we also found increased accumulation of different base damage that was done using mass spectrometry from a mouse model exposed to heavy ion and gamma rays, and we isolated DNA from intestine, and then we looked for these oxidized bases in the DNA, and we found that 8-hydroxyguanine and R-cyclo-2-deoxyguanosine was significantly increased after space radiation exposure. And this increase in oxidatively damaged bases also coincided with different enzymes involved in DNA base damage repair or nucleotide damage repair. So both NER and BER repair enzymes were downregulated after iron, which is a space radiation exposure, compared to gamma and control mice. That further indicated that a space radiation induced tumorigenesis is somehow associated with oxidative damage to the DNA bases and deficient NER-BER pathways. So deficiency in NER and BER pathway basically sometimes it results into double strand break formation because of misrepair. So we looked into persistent changes in double strand break accumulation using gamma H2AX staining at different time point post radiation exposure. And these are the green dots here, that they are the foci, which represents a double strand break in the DNA. And we found that after a space radiation exposure, there is a substantial persistent increase in double strand break in mouse intestinal cells. And also there was evidence of persistent DNA damage response, and marker for persistent DNA damage response we use is 53BP1, and which was substantially increased long-term after radiation exposure. So it indicates that there is damage to the DNA, and if the damage is misrepaired, there is a double strand break, and that double strand break basically induces DNA damage response in these cells, and that may lead to mutation and tumorigenesis later in life. Another aspect of DNA damage is senescence, which is very evident during aging. In normal aging, cell stops dividing and they sit there, don't divide, and they secrete so many different inflammatory factors after acquisition of senescence-associated secretory phenotype, which is called SASP. So we found that after space radiation exposure, there is a substantial increase in double-strand breaks, and that double-strand break is basically driving accumulation of senescent cells, and these senescent cells are overexpressing SASP phenotype and SASP phenotype related markers like IL-8 and IL-6 and other markers related to inflammation. So that indicates that when a mouse model is exposed to heavy ion radiation exposure, it undergoes senescence via increased DNA damage accumulation, and these senescent cells acquire a senescence-associated secretory phenotype that leads to chronic inflammation, and it's kind of a vicious cycle that operates after heavy ion radiation exposure, and that may be related to both accelerated aging and increased cancer incidence. In human studies, it is well known that with age, cancer incidence increases and stem cell division rate decreases. So our hypothesis in the regard of space radiation is space radiation is accelerating these effects. So aging is accelerated and cancer incidence is increased. And this inflection point is basically moved earlier in life. So accelerated aging and higher cancer risk can be seen early in life compared to what we see during normal aging. So there are various countermeasure strategies that can be tested using ground-based animal model approach. And two particular type of countermeasure strategy is physical approach and biological approach. In case of physical approach, we can test different type of shielding materials, and currently available or currently frequently used shielding material for a spacecraft is aluminum, 20 gram per centimeter square aluminum. And we tested this aluminum shield in this mouse model, and we also tested genetic mouse model approach and drug-based countermeasure approach to mitigate space radiation-induced tumorigenesis. So in case of shielding, we found some partial protection from space radiation-induced tumorigenesis, but that was not enough to bring it to the control level. So we need some kind of therapeutic countermeasure in addition to shielding, effective shielding. In this study, we used 20 gram per centimeter square of aluminum, and we exposed mice with this shield. And there was substantial reduction in dose accumulation in these mice, and that dose reduction basically correlated with reduction in tumorigenesis. So there was 30 to 40% of dose reduction and around 40 to 50% of tumor reduction. So the shielding seems to be the best way to reduce tumorigenesis, but more and more shielding technology needs to be tested to reach effective tumor control. We also tested a mouse model
So
where
Dr. Shubhankar, we can wind up.
Yeah, just two slides.
Okay.
So in the genetic model, this is a weapon knockout mice we use. So in this mouse, there is a proactive DNA damage response, and all the damaged cells undergo efficient DNA repair, and there is a reduced mutagenesis and reduced tumor frequency. And when we use these mice and exposed to space radiation, we found substantial reduction in tumor development after space radiation exposure. In another study, metformin, which is a well-known anti-diabetic, and now there are studies showing that metformin is also an anti-aging drug. When we administered metformin, started metformin before radiation exposure and continued till the end of our study. we found effective protection in tumor development, which is induced by space radiation. So in case of space radiation, there is substantial increase in GI tumorigenesis. And when we added metformin in the diet of these mice, we saw substantial reduction in space radiation induced tumor formation, both in intestine and colon, and also tumor size was reduced. So in summary, heavy ion space radiation exposure increases cancer risk. Higher persistent stress and differential DNA damage response could be linked to differential changes in cell fate decision and cellular homeostasis after heavy ion radiation exposure. And higher number of senescence-associated secretory phenotype could drive pro-inflammatory changes and carcinogenesis after space radiation exposure. And proactive DNA damage response after heavy ion exposure is crucial in reducing space radiation-induced cancer risk. And there are many anti-aging and senolytic drugs that are under investigation, and probably they are potential countermeasure against space radiation-induced cancer risk. And finally, I would like to acknowledge all the participants from Georgetown University and NASA Space Radiation Laboratory and NASA for funding. Thank you.
So thank you so much, Dr. Shubhankar. I think now the forum is open for any question answers from both our esteemed speakers. Any questions? Any questions from the audience?
Good evening, ma'am. Good evening, sir. Am I audible?
Yes, yes, please go ahead.
Yeah. Sir, I just want to know about the...
Can you identify yourself, please?
Yeah, I am Dr. Neha from Hindu College, Chemistry Department.
Okay.
Yeah. So
please go ahead.
Okay, ma'am. So my question is with Dr. Shubhankar. Sir, can you little bit explain about what is the source you are used for this space radiation?
Yeah, so there is a space radiation facility. Basically, it is for NASA user community, and they use ion beam accelerator. So they have 3 different sources of space radiation, and I have the slide for that one. So they have LINAC, that is linear accelerator. They have relative heavy ion collider and electron beam ion source. So these 3 sources, especially with the RIC, relativistic heavy ion collider, we can generate all these heavy ions. But with the EVs and LINAC, we can generate some of the ions, but not all of them. And this is kind of a standalone facility globally. So if someone has to work with this particular type of a space radiation simulated beam, they have to come to NASA Space Radiation Laboratory to use it.
Okay, sir. Okay, thank you so much.
Anyone else? If there is no other question, then can I ask Dr. Shubhankar, I wanted to ask you that when we send our astronauts into space and they return back, are there any follow-up studies which have actually documented the incidence of accelerated aging and cancers in these astronauts? And what is the magnitude of the exposure which is going to cause this?
Yeah, so currently astronauts frequently visit International Space Station, which is basically low Earth orbit. It is not a deep space.
Okay.
So in low Earth orbit, still there is magnetosphere active. So there is some repulsion of these heavy ions from space. But in deep space, there is no such protection. So we don't know about deep space. As far as follow-up goes, all these astronauts, they go to International Space Station maximum for 6 months at a time. And when they come back, there is some lifetime limit to radiation exposure. So they can either go once or twice in their lifetime. Other thing is, in terms of follow-up, there is a rigorous follow-up on these astronauts, and they do like all the blood work, samples, chromosomal aberration, and so many different studies. And they do see a lot of perturbations related to not particularly accelerated aging, because dosage are very low in low Earth orbit. But in terms of DNA damage, chromosomal aberrations, some mutations, and now some epigenome changes are also reported.
Okay. Then when we talk
But
about
the problem is that International Space Station, it's kind of a combined stress. Even for deep space, it will be microgravity and space radiation, two major space flight factor, apart from like confined space and other things. So we are yet to decipher like why these changes are particularly associated with radiation or microgravity. We don't know. Because
right.
these astronauts are exposed to both the conditions,
True,
microgravity, and so microgravity causes like a lot of neurobehavioral changes,
Surely.
good shift in the body. So they have like edema and water accumulation in the brain and things like that. So that is completely different from a space radiation exposure. And we are yet to do a combined study, like where we expose our mice to both microgravity and space radiation. Probably in future, we are going to do that.
Great, great, great. And then when we talk about countermeasures, we were talking about shielding and several others. So which is the most effective strategy? Is it to use more than one at a time or something like that? Which is the most effective strategy as a countermeasure?
Effective strategy in terms of physical shielding. Physical shielding is limited because of the lifting capacity of the rockets available. So you can use like 10 feet of concrete wall to shield these ions. That is physically possible, but you can't make a capsule with 10 feet of concrete. So that's kind of a logistical issue. So they have to use some kind of composite material which is lighter in weight, and NASA has like rockets built to like lift these materials. So I think it all goes with the material strength and its weight.
Okay.
So it is not only like they are focused only on shielding, but they also need to take care of logistical issues related with the space flight.
Sure, sure, sure. So any other comment from the audience? So with this, it's my great pleasure to extend big thank you to both the speakers, Dr. Shubhankar Suman and Dr. Girija Bharat. So thank you for such a nice presentation on behalf of the STE family. Thank you so much.
Thanks.
Thank you.
Thank you, Usha ma'am. I now invite Manisha to formally pose the vote of thanks to our chairperson and the speakers. Manisha?
Yes, ma'am. Hello.
Manisha, are you there?
Ma'am, she's not able to unmute herself.
Okay, okay.
Hey, Richa, she's already unmuted. I can see her unmuted.
I'm here, but there
It's
seems
okay.
to be some
I
problem.
express my heartfelt thanks to our chairperson, Dr. Usha Panjwani, who has chaired the session very effectively and has made this session very, very interesting. And of course, thanks are due to, and of course, deep appreciation to our speakers also, who have given us a very scintillating and motivating talk. Thank you very much for the session. I now request again Manisha to please carry forward this session to the poster presentation. I request Usha ma'am to be there for the judgment of the poster.
Thank you so much, ma'am.
Thank you.
Thank you.
Manisha, please check your audio settings or try rejoining because we can see you as co-host and unmuted. So it seems to be some technical issue at your end. Anupama, can you hear me? Dr. Anupama?
Manoj, can you see what is the problem with Manisha audio system?
Okay, ma'am, I'm coordinating with her.
Okay.
Manoj, do we already have the poster presenters with us here?
Just wait a minute, ma'am. I'm telling you. Yes, ma'am, Ankit is there. Vrinda is also there. So we can start with them.
Yes, I think we can start.
We can start with Ankit.
Can we have the first group? Usha ma'am, if you permit, shall we begin?
Usha is muted actually. I think she might have gone, but just should I call her?
हाँ मैम पूछ लेते हैं बिकॉज अगर उधर से प्रॉब्लम है नाम तो हमारे पास है तो हम टाइम नहीं वेस्ट करते शुरू कर देते हैं
उनको बोल दिया ना उनको
I've already called Manisha, वो कह रही है मैं कर रही हूँ ट्राई लेकिन ऑडियो नहीं लग पा रहा है उसका.
We can start with
Am I audible now?
Are you there?
Yes, Manisha, you are audible.
Yeah. Okay, ma'am. Okay. Sorry for the this inconvenience, ma'am. Actually, I don't know how it got disrupted. In the morning, I was able to speak very in a proper manner. Yeah, ma'am. So I was just thanking, I was just thanking Greja ma'am and Shubhankar sir for such an informative talk. So now we are going to begin with the next segment of the session, that is the poster presentation. And for that, I would like to request all the chairperson, so the chairperson and the invited speakers also to stay with us to judge the session. This is for the kind information for the judges that there are 3 participants for poster presentation, and the winners would be decided on the basis of a novelty of work, content, presentation, and response to questions. Let me read the guidelines for the presenters also. The presenters are required to present their paper live at the online platform, and a maximum of 2 presenters can work. Maximum of 2 presenters can present their work together. The time limit for each presentation is 6 minutes, 5 minutes for presentation and 1 minute for a question answer session. So I would request the presenters also to adhere with the guidelines, as we are already running late with the time. So before starting with the presentation, I request Usha ma'am to say a few words.
So now we are having the poster session. So I would just say that we are very keen to hear the young students and the young researchers, and they should stick to the time of 5 minutes and 1 minute question answer. That's all.
Yes, ma'am. So let me call the participants one by one. The very first entry for the session is Ankit, Shyala, Professor Reena Jain, Professor Anju Srivastav, Dr. Shriparna, Dr. Devanshi, and Dr. Aman Bhardwaj. Ankit, over to you.
Ma'am, is the screen visible?
Yeah, it is visible.
Okay, ma'am. Ma'am, may I start now?
Yeah, please start.
Thank you, ma'am. Waste, which has often been treated as undesirable and unwanted as it leads to uneconomical use of resources and capital, is now being considered as a precious resource. Green chemistry has shown the phenomenal capability of solving the twin
Can you be a
problems of
little
resource.
loud, please? Speak a
Okay,
little
ma'am.
loudly.
Green chemistry has shown the phenomenal capability of solving the twin problems of resource and waste by making the latter a solution to the former. Henceforth, the linear economic model is slowly being replaced by the circular economy model, where an increasing emphasis is placed on converting different types of waste materials into value-added resources. Good evening, everyone. I, Ankit, a second-year student pursuing majors in chemistry from Department of Chemistry, Hindu College, along with Shayala, second-year student of BSc Honors Chemistry in Hindu College, is going to share our work on green perspective of waste of spent lithium-ion batteries. With the booming of consumer electronics and electric vehicle, a large number of spent lithium-ion batteries have been generated worldwide. More than 50 million metric tons of e-waste is generated globally every year, averaging some 7 kilograms of e-waste per capita. Resource depletion and environmental concern driven from the sustainable industry of CE and EV have motivated the scientific community towards focusing on the recovery of the spent lithium-ion batteries urgently. Here, under this work, specific emphasis has been directed towards recovery of lithium cobalt oxide from the spent batteries of mobile phones, which have been chemically converted into cobalt oxide nanoparticles using a greener pathway. Since the conventional process combined with leaching, precipitating, and filtering was found to involve the use of toxic precursors and quite energy intensive. Typically, through this work, we have developed a novel alternative sustainable recovery process using non-toxic greener reagents. When the optimal parameters for leaching process was controlled at 150-minute retention time, 95 degrees Celsius heating temperature, 15 gram per liter solid liquid ratio, and 400 RPM rotation rate, the recovery rate of lithium and cobalt from spent lithium-ion batteries was found out to be 98% and 97% respectively. The obtained oxide nanoparticles displayed promising efficiency in the photocatalytic degradation of the methylene blue dye, on which we have done work over the photocatalytic degradation. Additionally, the work also tentatively discovered the leaching mechanism of lithium cobalt oxide using the oxalic acid and the leaching order of sampling of spent lithium-ion batteries. Now, I would like to call Shayala to focus more around the characterization of the lithium cobalt oxide during the process. Over to you, Shayala.
Thank you so much, Ankit. Here I'm discussing the characterization of lithium cobalt oxide. First, XRD and SCM. Second is SCM. The XRD pattern recorded for lithium cobalt oxide synthesized at different temperature is appended in the given figure. The diffraction pattern indicate that crystal structure is identified to that of single phase structure of lithium cobalt oxide for temperature such as 600 and 700 degrees Celsius. On the other hand, lithium cobalt oxide synthesized at 800 degrees Celsius. This corresponds to a mixture of different structure of lithium cobalt oxide, which has not been investigated yet. So there is also the reduced intensity of doublets corresponding to the some ordering structure, and the lithium cobalt oxide, which is synthesized at 900 degrees Celsius, match exactly with the we can say the cubic or the rod-like structure of lithium cobalt oxides. It also form a striking similarity. So now I'll discuss the surface morphology. SCM studies. This scanning morphology is used to analyze the initial morphology of sample and morphological changes in the reaction product at different temperature. In the initial state, the sample has some irregular shape, but when we see the figure B and C, we can evaluate that there is a presence of formed rod-like perfect structure of lithium cobalt oxide, which is synthesized at 600 degrees Celsius, or in B at 800 and 900 degrees Celsius. There is also more specifically, there is also the coexistence of CO3O4 that is evident from the brightest secondary particles formed with the desired lithium cobalt oxide compound, which is synthesized at 900 degrees Celsius. Here is also the particle size analysis. The particle size analysis of lithium cobalt oxide synthesized at different temperature. This clearly evidences the presence of nanocrystalline, which is less than 100 nanometer size particle size at 600 degrees Celsius, and the higher temperature synthesized lithium cobalt oxides. It corresponds to large particle size.
So you can wind up now. The 5 minutes are over.
Shweta, please try to wind up. Okay. Now over to you, Ankit.
Thank you, Shaila. Concluding the work, there was we used the spent lithium-ion batteries and extracted the useful cobalt cathode material, lithium cobalt oxide, and on further leaching process, we designed the cobalt oxalate ion, on which mixing with the desired dye and on the photocatalytic degradation, we found out that we got positive results in respect to this, but the research methodology did not stop here. Accordingly, in the near future, we are also thinking to change the variation of the concentration of the oxalate ion with the dye to check the efficiency of the cathodic material, as well as on changing the different dyes. We will be taking into consideration the work in the very near future. Thank you so much for listening us.
Okay.
Thank you. Any questions on this?
I think, ma'am, we can call the next presenter.
Okay. Or you were talking about efficiency of green battery vis-a-vis the lithium-ion battery. So what is the status of, what is the current status?
In today's time, less than 5% of lithium-ion batteries are currently recycled, but it has been predicted between the 2017 and 30, around 11 million tons of lithium-ion batteries will be disposed of, which presents both a major need and opportunity for recycling program.
Okay.
Thank
you. So the next poster, we can start with the next poster.
Yes, ma'am. So the next presenter is Surya Pratap Singh. Surya, are you there? Surya Pratap Singh. Ma'am, shall we move to the next presenter? Meanwhile,
yes, we can go to the third one if the second one is not here.
So the next presenter is Vrinda, Professor Anju Srivastava, Professor Reena Jain, Dr. Shri Parna, Dr. Devanshi, and Dr. Aman. Vrinda, can we have a word from you? Can you start with the presentation?
Good afternoon.
Good afternoon. Start sharing your screen. Yeah.
I guess my screen is visible.
Yeah, very visible.
Okay. Education. Education is a beautiful gift of humans, passed and nurtured through generations. It is passed by the means of teaching. Each generation teaches the next one. It is nurtured by the means of research. Each generation takes what's already there, does their research work, and takes it ahead, thus developing education. Our teachers understand the importance of it and wholeheartedly give their contributions in developing and our interest towards research. Similarly, because of the contributions of Professor Anju Srivastava, Madam Principal, Professor Reena Jain, Madam Vice Principal, we students got the opportunity of value-added courses in which we not only learn by the means of books and teachers, but also by performing practical application projects which have real-life implementations, which in turn motivates us towards research. One of the value-added course of which I was blessed to be a part of was Insights of Hydrogels through Molecular Spectroscopy. Hydrogels can be defined as cross-linked hydrophilic polymers. The number one capability of hydrogels is to hold water molecules. It has the wonderful ability to hold 100 times of water than its weight and still maintaining their structures. In layman language, they are super absorbent sponges. Because of this capability, they find their applications not only in agrochemistry, drugs and medicinal chemistry, but also in day-to-day things like flexible eye contact lenses, sanitary pads, and many more. The present work, our work, aims at investigation of aggregation formation tendency of standard biomolecular probes in hydrogel aqueous-based systems at varying pH conditions. In the first phase of the project, we made hydrogels of 5, 10, 15, 20 monomer concentration. The monomer that we use in N is N-isopropyl acrylamide. In the next phase, we will be performing UV spectroscopy and time-resolved fluorescence spectroscopy to actually authenticate and investigate the formation of aggregates. In the last stage, we will be moving towards the application and future users portion into actually see where the project can be put use to. We will be studying catalysis, we will be studying dye degradation, and how they can be used to degrade various organic pollutants like dyes and pesticides. On this note, I'd like to express my gratitude to Madam Principal, Madam Vice Principal, and all the faculty members who were involved in the value-added course, because of whom we students got this wonderful opportunity, this wonderful exposure. On this note, thank you so much. With this, I remain.
Thank you, Reena. Any questions on this poster? So, Vrinda, if you had to summarize what you did in this, can you just summarize once again and tell us?
Ma'am, our major aim is to see at various hydrogel concentration, monomer concentration, how aggregation of dye is at varying pH, how the same dye, a porphyrin, is aggregating at various pH.
So what did you find? What is the conclusion?
Ma'am, this project is still going on. It will be completed in the current semester, but we have completed with the first phase. We have made hydrogels of concentration 5, 10, 15, 20 percentages, and we found that the 20% monomer concentration has the most water holding capacity.
Okay. So thank you, Vrinda. Any other question? So thank you. We have come. Is the second speaker, Surya Pratap Singh, poster 2, is he there?
He's in the meeting, but I don't know why he's not speaking. Surya Pratap, are you there?
Shall we wait for him or shall we wind up?
Can he be called? Just
Yes,
try.
ma'am, we can call him in case he is not able to unmute himself or not able to speak, just like me.
Manoj, he is an undergraduate physical sciences student. Do you have his number?
So now he has left the meeting. I have texted
has
on
he
the-
left?
Yeah.
Then I think we should move forward. If he replies, then maybe in between we can give him another chance if Usha ma'am permits.
Yeah, of course.
Manoj, you try to contact him in the meanwhile.
Okay, just give me 1 minute.
So I think we should move forward, and I would like to thank students for their work. They have presented their work in a very fine way, and they must be waiting for their results, but the results will be declared in the valedictory session. So before we wind up this informative session, I would like to request our chairperson, Dr. Usha Panchwani, to kindly embrace the session with your concluding remarks. Over to you, ma'am, please.
Okay, so this is the last session before the valedictory. So it has been really informative. We had 2 speakers. Dr. Girja, she gave us such a good insight into the policy, water policy and safe reuse of water. I must congratulate her and her team for the nice work which they are doing. So, and the second speaker, Dr. Subhashankar Suman, who is also an alumni of INMAS DRDO, is doing very nice work pertaining to the aging and tumor genesis in the effect of space radiation on these measures. So he is, and also looking at some of the countermeasures which can be used, biological, physical, different kind of countermeasures which can prevent accelerated aging and cancer progression in in this mouse model which they have made and which they are studying. So a lot of application of this work on the astronauts and who are and also to say that not just NASA but our own ISRO, Indian Space Research Organization, is also doing very path-breaking work and trying to send a mission to the Mars. So these kind of scientific studies are very useful. And following these 2, we had a very nice interaction with the youngsters. The first speaker was Ankit. They were Ankit and Shayala, then Vrinda. So thank you both, the poster speakers. And thank you all of you for the patient listening. Thank you.
Thank you, ma'am, for your words. So now we are heading towards the conclusion of the session. And to conclude, I would like to present the formal vote of thanks. So on behalf of the entire coordinating team, I take this opportunity to say thanks to all who have helped in making this session a wonderful one. First of all, I would like to express thanks to our chairperson, Dr. Usha Panchwani, for gracing and conducting the session in an articulate manner. Thank you, ma'am. I would like to express my sincere thanks to our distinguished speakers, Dr. Girija Bharat and Professor Shubhankar Suman, for giving such informative talks and making the session interesting and the enlightening one. Thank you, sir. Thank you, ma'am. I would also like to thank our participants for their exceptional participation as well as the wonderful audience for their august presence. At the end, I would like to express my deep sense of gratitude to the organizing team for their constant support and unmatchable coordination and making this event echoing with success. Thank you. Thank you, everyone. Now, for the students' activity, I hand over the platform to Mohit. Over to you, Mohit.
Thank you so much, ma'am. It's time for the student to be attentive because now we are heading toward the weighted student activity of day 2 conference. We are overwhelmed by the yesterday's participation and hope for the same in today's activity, which we are going to be much more interesting, and we are hoping the students will enjoy the activity. The basic rules and regulation will be given before the respective activities. Best of luck to all of the participants. Gauri, please carry on forward.
Thank you so much. I request Shara to share her screen. The moment you doubt whether you can fly, you cease forever to be able to. Good evening, everyone. Greetings, Hinduites. I, Kauri Vasist, a second-year student pursuing majors in chemistry from Hindu College, elated to bring you all to Quizmania from Tattva, the Chemical Society, Hindu College. For this, the teacher coordinators are Dr. Hemant Verma, Dr. Dinesh Kumar, and Dr. Aman Bharadwaj, along with student coordinators Ram Datan, Gauri Vasist, Shayala Kashyap, and Vansh Sharma from second year. The activity is divided into 3 segments, and each segment is completely different from the other. So let's gear up for the first segment, that is portrayal, in which you will be shown a picture and you have to simply name the scientist. Remember, each question carries 5 points and you will be given 30 seconds for answering. Also, answer should be typed only in chat box and the mics must be turned off. I request all the participants to keep their cameras on. I also request the volunteers to keep a track of scores. All the best to all of you, and here is your first question. The balloons all have same volume. This means they all contain the same number of molecules. This law was given by which scientist? Your time starts now. We have got the answer. And the law is Avogadro's law, and it was given by Avogadro. Moving forward.
Excuse me, Gauri. Wait a minute. Actually, ask all the students to come up with their original names.
Okay, wait. You all are requested to join with your original names. We have to point you according to your names. I request everyone to change their names as soon as possible.
I think you should complete this question because you have displayed this.
Yeah. If you have a cat and something like that could kill the cat, like a radioactive atom in a box, and sealed it, you would not know if the cat was dead or alive until you opened the box. So that until the box was open, the cat in sense both was dead and alive. It is a very famous cat story, and it was given to prove the misconceptions about quantum theory. Can you guess the name of the scientist who used that? And the scientist was Schrödinger. I saw a dream, a table where all the elements fell into a place as required.
Yes, Mendeleev was the scientist who saw a dream in which a table where all the elements fell into the place. In 1922, he published his work in molecular diffraction of light. Name the scientist who worked for the molecular diffraction of light. Chandrasekhar Venkataraman was the scientist who worked for the diffraction of light. The merchant death is dead. This quote blasted a French newspaper in April 1888. Clock is ticking, guys.
This is a tricky question, Gauri. Huh? No answers till now.
No, ma'am.
Good, good, good.
It was Alfred Nobel whose fake death news blasted French newspaper in 1888. With this, our first segment comes to an end. Don't be disheartened if you were not able to answer a few questions, because as Winston Churchill said, success is not final and failure is not fatal. It's the courage to continue that counts. So let's be courageous and let's dive into our second segment, that is pop quiz. You have to choose from your answer from the 4 options. And please note that you will have 20 questions and each carries 10 points. You have to write only A, B, C, or D and not the complete answer of the question within the time limit of 25 seconds. Let's begin with our first question by wishing you all a lot of luck. Which were the first element to form after the Big Bang started to cool? Your options are hydrogen and helium, uranium and plutonium, carbon and silicon, oxygen and nitrogen. Yes, hydrogen and helium were the first element to form after the Big Bang started to cool. Which among the following could cause bright red color due to the burning of crackers? I repeat, which among the following would cause bright red color due to the burning of crackers? Your options are A, sodium, B, sulfur, C, strontium, D, magnesium. Yes, your answer is strontium. The set of quantum number n, l, and m not allowed for hydrogen atom. Option A, n3, l equals to 1, m equals to -1. Or B, n equals to 3, l equals to 0, m equals to 0. C, n equals to 2, l equals to 1, m equals to 0. Or D, n equals to 2, l equals to 2, and m equals to -1.
Wait a second, Gauri. Gauri, in the
Yes,
second
ma'am.
activity, you just tell them the answer with the question number. Actually, second question answer and the third question answer matches in between. So just tell them the answer with the question number.
Okay. Please answer by writing a question number in front of them, like A3D. Let's move forward. Name the world's first artificial nuclear reactor. A. Experimental breeder. B. X10 graphite. C. Chicago Pile 1. Or D. Apsara. Yes, Chicago Pile 1 was the world's first artificial nuclear reactor. An ester which is used as medicine is ethyl acetate, methyl acetate, benzyl salicylate, or ethyl benzoate. And the correct option is C, benzyl salicylate. Which of the following pair do not show similar chemical properties? I repeat, which of the following pair do not show similar chemical properties? A, fluorine argon, B, beryllium aluminium, C, boron silicon, D, lithium magnesium. Yes, the correct option is A. Fluorine and argon do not show similar chemical properties. Which of the following metal is the main component of pewter alloy? Your options are A, lead, B, zinc, C, tin, or D, iron. Tin is in highest composition in pewter alloy. Artificial sweeteners is A, saccharin, B, sucralose, C, L-tame, or D, all of these. Yes, you all are absolutely right. All of these are artificial sweeteners. Barbituric acid is used as antipyretics, tranquilizer, analgesics, or antibiotic. Yes, barbituric acid is used as a tranquilizer. You all are performing very well. So let's keep this spirit high and move to our next question. Which of the following process of colloidal suspension is used to determine the nature of charge in the particles? A, dialysis, B, electrophoresis, C, sedimentation, or D, ultrafiltration. Option B is the correct answer. Which gas is used in atomic research and radiotherapy? Your options are A, xenon, B, radon, C, neon, or D, propane. Your correct option is A. The element present in largest number of rocks and minerals is A. Carbon, B. Silicon, C. Aluminium, or D. Hydrogen. And the correct answer is B. Silicon. An alloy used in making heating elements for electric heating device is A. Solder, B. Alloy steel, C. Nichrome, or D. German silver. The correct option is C. Which charcoal absorbs coloring matter and odoriferous gases? A. Activated charcoal, B. Animal charcoal, C. Wood charcoal, or D. All of these. The charcoal which absorbs these are animal. Which does not come under intellectual property right. Your options are industrial design, trade secret, trademark, or copyright. Yes, trade secret does not comes under intellectual property rights. Which of the following compound has highest surface tension? Propylene glycol, water, glycerol, or ethylene glycol? Option B is the right answer. Water has the highest surface tension. That's the spirit, my friend. Diane Johnson said, success isn't about greatness. It's about consistency. Consistent hard work leads to success. So keeping this exuberance consistency, let's move to our third and the last segment, that is Guess Who. Please note that you will be asked 16 questions and each question carries 15 points. Answer should be typed in chat box within 30 seconds and the first one to answer will only get the point. You are requested to keep your mics off and your cameras on. I request all the volunteers to keep a track of scores. I am the element which a dead chemist or grave diggers has to deal with. Who am I? I repeat, I am the element which a dead chemist or grave diggers has to deal with. And the answer is barium. This is an element that has been derived from the famous Norse god. What it is? Yes, the element is thorium. I come as a gas and I am in the line with neon. I am also the home of very Superman. Who am I? Yes, the answer is krypton. What is black when you buy it, red when you use it, and gray when you throw it away? And the right answer is charcoal. He is popular for Bose-Einstein condensate, the fifth state of matter studied, which was later predicted to be the fifth state of matter by Albert Einstein. Who is he? Yes, the correct answer is Satyendra Nath Bose. She is credited with the invention of early type of distillation apparatus. Without this useful chemical technique, we would not have petrol, plastics, or indeed vodka. Can you guess the name? Guys, we are asking the name of the scientist. And she is Cleopatra the alchemist. Shiny like a silver, denser than lead, pricier than gold, could stop a bullet if I wanted to. What am I? Yes, you all are very right. The correct answer is osmium. You can get me for free, but inside an atom. I add a lot of weight. What am I? And the answer is neutron. Keep working hard, guys, because the harder you work, the better you get. A very famous chemist was found murdered in his kitchen today. The police have narrowed it down to 6 suspects. They know it was two-man job. Their names Felice, Maxwell, Archibald, Nicholas, Jordan, and Xavier. A note was also found with the body, 26-358 and 28-27-57 and 16. Who was the killers? I request you all to read the question since the answer is in the question itself. Yes, the murderers are Felice and Nicholas. What is the major component of drying oil? Your next question is, what is the major component of drying oil? Unsaturated fatty acid is the correct answer. The Nobel Prize in Chemistry 2022 was jointly awarded to Carol R. Bertozzi, Morton Meldal, and K. Barry Sharpless for the development of what? Yes, they got the Nobel Prize for their work in click chemistry and orthogonal chemistry. Which compound does red chili consist of? Your next question is, which compound does red chili consist of? Yes, the answer is capsaicin. Who coined the term consilience? Okay, and the answer is William Whewell. Which type of hydrogen is produced by the process of splitting of water by electrolysis? It is green hydrogen. Which compound is used in RAST method because of its high value of K? I repeat, which compound is used in RAST method because of its high value of K? Yes, it is camphor. With this, we come to an end, and I hope you all enjoyed this activity and you get a little bit of knowledge out of it. The results will be announced later in the validatory session. Thank you all.
Thank you, Gauri. With this, our activity ends here. Amazing energy shown by all the participants. Hope you are enjoying. Now it's time for the final activity of the day. So, guys, buckle up and get ready for the next activity. Again, Gauri, heads the next activity, please.
I request Shayala to present the screen. The aim of art is to represent not just the outward appearance of things, but their inward significance. Good evening, everyone. Quick sketches, sharp minds. The Department of Chemistry, Hindu College, is elated to bring a Pictionary competition under EWASH 2022, mainly focusing on sustainability and green chemistry. Do you know what does sketching and creativity means? Okay, let me help you. Sketching is almost everything. It is painter's identity, his style, his conviction, and then color is just a gift to drawing. And creativity is allowing ourselves to make mistakes. Art is knowing which ones to keep. I thank teacher coordinator Dr. Vinita Narulla and Dr. Raghavi Khattar for their guidance and constant support. I also appreciate the student coordinators Harsh Yadav, Muskan Kaswan, Monisha, and Simran Nishchal in making this activity successful. The theme of the activities were renewable resources are better than non-renewable resources, and second, the nation that destroys its soil destroys itself. The guidelines for the activities were a picture should be depicting the theme. A4 or A3 sheet should be used. It can be made using pencil, crayon, watercolor, oil paints, etc. Photographs, wires, or other 3D objects, printouts are not allowed. It should be original and handmade. Word limit is 50 words. Submission in PDF format and file should be less than 10 MB. Keeping this in mind, we opened this activity to a variety of students, and we are delighted to say that we have received many entries showcasing the hidden artist of our chemists. Now I would like you to take to the entries that we have received depicting a need of the hour to shift to renewable resources. But first, the term alternative energy, clean energy, green energy, and sustainable energy are not uncommon to hear these days. You may even note a sense of urgency around them. But do you know why? These are all common phrases used in reference to renewable energy resources. Currently, the primary source of energy in United States are fossil fuels, such as coal, oil, and natural gas. Unfortunately, fossil fuels are non-renewable resources created over the course of millions of years underneath the Earth's surface. When these finite resources are gone, they are gone. But the rate at which we are using fossil fuel is too fast. If we don't make a drastic and immediate change in how we are using them, we could potentially be depleted within our children's lifetime. Using these natural resources allows us to conserve and extend the lifespan of our non-renewable resources. Switching to the renewable energy source can positively impact the environment by slowing climate change. This positive environmental impact is where the term green energy and clean energy comes from. This is an entry given by Gitanshu, a first-year student of BSc Honours Chemistry, Hindu College. I could tell that you have made just by looking at it. The second entry is of Muskan Sharma, a second-year student of Physical Science with Chemistry, Hindu College. The closer I look at this piece, the more I am mesmerized by it. Wonderful work, Muskan. This is the entry of Ruchika, a first-year student from BSc Chemistry Honours, Hindu College. An entry of Arun Lobre, a first-year student of BSc Chemistry Honours, Hindu College. All the hard work you put into the piece really shows. The work of Anshuka Khandari, a first-year student of BSc Honours Chemistry, Hindu College. This is the work of Abhay Pant, second-year student of BSc Chemistry Honours, Hindu College. The color in this painting make me feel at peace. It is the art of Komal Kumari, a first-year student of Physical Science with Chemistry from Hindu College. The work of Kwaljit, a first-year student of BSc Chemistry from Hindu College. The art of Jia Bharadwaj, a first-year student from BSc Honours Chemistry, Hindu College. It is the creativity of Anshika Mishra, first-year student from BSc Chemistry Honours from Hindu College. The art of Ankit, a second-year student of BSc Honours Chemistry, Hindu College. The color in this painting makes me feel at peace. The work of Supriya Jain, a first-year student of Physical Science with Chemistry from Hindu College. The second theme, the nation that destroys its soil, destroy itself. Given the current state of the world, it should not be surprising that air quality is improving, water pollution decreasing, and the earth is seemingly healing itself. Due to this reason, the people have been posting online claiming that the world would be better off without humans. While I understand the thought process, this just simply isn't true. The idea that humans should be erased to better the world is very irrational and does not take the entire picture into consideration. Humans have come up with many solutions to the issues we are having environmentally. However, at the same time, humans have taken little to no action and have seemingly looked the other way, rather than solving the number one threat to our future. One potential solution to the loss and degradation of soil is the creation of soil from waste material, where appropriate inert material generated as waste are carefully mixed together to create a substrate with the characteristics of healthy soil. Their uses include manufacture of topsoil for urban grasslands and materials for high-value markets such as horticulture and agriculture. Moving to the entries depicting soil preventions, it is a work of Bittu Sharma, a second-year student of BSc Honours Chemistry from Hindu College. All the hard work you put into this piece really shows. The work of Kriti, a first-year student of BSc Honours Chemistry from Hindu College. Your style provokes many emotions, Kriti. The art of Haridyanshi Roshan, a first-year student of Physical Science with Chemistry from Hindu College. The closer I look at the piece, the more I am mesmerized by it. The work of Kanupriya, a second-year student of BSc Honours Chemistry from Hindu College. I love how you have used all the colors in the painting. The art of Surya Pratap Singh, a second-year student of Physical Science with Chemistry from Hindu College. I could tell you have made it just by looking at it. That's all. These are all the entries, and I thank all the participants for their hard work and for creating an outlet for us to be challenged. The results will be announced later in the validatory session. Thank you, guys.
Vinita ma'am, are you here?
Yes, Ragvi. The students are over with their activities?
Yes, ma'am, they have just finished it.
Yes, ma'am. Dictionary is over.
Dictionary, yeah, I was going through, but any more activities left or you're through with all the activities?
No, ma'am. No, ma'am, only 2 activities today.
Only 2 activities. I must say, because we were the judges for the Pictionary and the number of entries that we received, it was very difficult for us to judge those entries. Me and Raghavi ma'am almost spent more than half an hour to judge कि किसको first और किसको second दिया जाए, because it was very difficult for us, and all the entries were very good. You must have seen, everyone must have seen here right now. So anyway, though we have extended our result, जो हमारी limitation दिया गया था, we were supposed to give only first, second, and third, but we extended the final list, and we found 2 students who have stood first. Then one has got the second, third. Even we have given names for the consolation prizes. So
if they agree, ma'am.
Yes, if the sponsors agree to this result, if they are,
yes, ma'am.
I think Ajay sir is there. He's sponsoring all these prizes, so definitely, sir will extend his. Sir, are you there, Ajay sir?
Ajay sir, unmute yourself, sir.
Yes, I am unmute.
Anjit sir.
Okay. Yes, ma'am.
सर आपने तो बच्चों की एंट्रीज अभी देखी ही हैं और काफी अच्छी परफॉर्मेंस रही है, बट इसमें काफी मुश्किल हो रहा था हमारे लिए जज करना, और अभी जो क्विज मैनिया चल रहा था उसमें भी स्टूडेंट्स का रिस्पांस देख करके तो ऐसे लगा था पता नहीं कहाँ से ये स्टूडेंट्स
They are so talented, man.
Really very brilliant students.
ये तो काफी अच्छी परफॉर्मेंस है बच्चों की, सो दिस इज हाईली एनकरेजिंग फॉर ईच वन ऑफ अस कि हमारे स्टूडेंट्स अच्छा।
हमने अजय सर को तैयार कर लिया है फॉर मेनी प्राइजेस, इवन कंसोलेशन प्राइजेस आल्सो. वी हैव टॉक्ड टू हिम एंड ही इज वेरी काइंड इनफ टू एक्सेप्ट दिस. हमें कई प्राइजेस,
मतलब ये तो ना बुक्स ना हमेशा के लिए रहेंगी बच्चों के लिए.
बस यही
ऑल द बेस्ट.
मेरे को जब बाद में पता चला कि बिकॉज जब हमें रिजल्ट दोबारा से रिवाइज करने के लिए कहा गया तो मुझे लगा कि बहुत ही मुश्किल है, किसका नाम आगे पीछे किया जाए, बहुत ही मुश्किल था.
बहुत मुश्किल था मैम.
मैंने नीचे लिख दिया उसमें कि कंसोलेशन प्राइजेस इफ पॉसिबल, एंड
हम कंसोलेशन भी दे देंगे, कोई दिक्कत नहीं.
नहीं मुझे एक्चुअली तब तक भी नहीं पता था कि आप स्पॉन्सर कर रहे हैं, तो फिर तो शायद मैं और नाम भी डाल सकती थी इसमें.
बिल्कुल मैम, अभी भी अगर हो सकता है.
आप पे डिपेंड करता है, आप जो चाहे कर सकते हैं. यू आर फ्री.
अब तो खैर रिजल्ट फाइनल हो गया और सबमिट हो चुका है.
ठीक है.
तो विनीता, कैन वी स्टार्ट नाउ दिस वैलिडिक्टरी, बिकॉज हमारे थोड़ा चीफ गेस्ट इज वेटिंग.
यस मैम, डेफिनेटली.
ऑलरेडी लेट हो गया थोड़ा.
Yes,
sorry to interrupt. I did not
no, it's
want
okay.
to. But
That was the casual talk that was.
So because he's waiting and he has to go somewhere. So
yeah, definitely, ma'am.
Thank you, ma'am.
Thank you so much, ma'am.
Okay, a very good evening to all. It gives me an immense pleasure to commence with the valedictory session of this enlightening conference, EWASH 2022. Yesterday was marked by the presence of our honorable chief guest, retired IS officer Sumedha Kataria ma'am. During the 2 days of conference, we all had the golden opportunity to listen to some of the eminent speakers, wonderful presentations, and events participated by the students. Today, amongst us, we have Dr. Kalpana Bhargav as the chairperson of this session. I would now like to invite the co-convener of EWASH, Dr. Devanshi ma'am, to introduce. Over to you, ma'am.
Thank you, Dr. Pragya. Kalpana ma'am, do we have you with us?
Yeah.
Thanks for joining, ma'am. It's a pleasure to have you in the valedictory session, and I have the pleasure to introduce you. So we have with us as our chairperson for this session, Dr. Kalpana Bhargav, Scientist AF and Additional Director, High Energy Material Research Lab, DRDO, Pune. Ma'am has completed her PhD in peptide chemistry from BHU in a joint collaboration with Indian Institute of Science, Bangalore in 1999. After her PhD, she pursued 10 years of multidisciplinary research in academics and R&D environment from USA. Dr. Bhargav joined DRDO in 2008, and she has almost 30 years of research experience in a wide range of areas ranging from antioxidant peptides, free radical biology, nanoconjugate chemistry, proteomics, ribosomal biology. This is only a few that I am speaking. She has an extensive experience. She has almost 95 international peer-reviewed journal publications, books, book chapters, patents, and several scientific awards and honors to her credit. Her research work includes proteomic evaluation of biomarkers for high altitude related maladies, use of nanoceria for high altitude application, and development and evaluation of propellants and cartridge cases technology for gun ammunition systems. We are privileged to have you with us, ma'am. And I would request you to please introduce our chief guest for the session.
Thank you so much, Devanshi. Thanks a lot for the introduction. First of all, I am very happy to be here at this platform, and my thanks goes to Dr. Shipra Mishra, President ST, as well as Hindu College. Everybody in Hindu College, like, they are doing great job, excellent job. So congratulations to everybody, and thank you very much for having me here. It's my honor to be here. So without much delay, I would like to introduce our chief guest, Dr. Manoj Umesh Sharma. Dr. Sharma is a scientist G in SSPL, Solid State Physics Laboratory. He pursued his PhD in electronics from Kurukshetra University in 2015. He joined DRDO in 1985 as student officer at IIT Pune, and then was successfully promoted as scientist B at SSPL, and since then he has been actively working in SSPL. Some of his achievements, like, I would like to mention that his first thing was to make SAW activity functional. He designed and developed the first band pass filter and pulse compression filter at SSPL for Project Akash. He has also taken the responsibility as head of mask fabrication group, resulting in fabrication of more than 500 masks for various ongoing activities within and outside DRDO. He has handled EBMF activity, which was on the verge of closure, and also developed for 0.25 micron T-gate technology, which was a stepping stone for development of 40 gauge technology in the country. Dr. Sharma has developed the first working system in ASICA of SSPL for the 3 services. He is also recipient of Critical Technology and Group Technology Award. Dr. Sharma has established costing department for arriving at the right cost of stock product, successfully developed and supplied large number of pressure transducer to various programs of DRDO, and also developed G-switch for the requirement from armament. So with this much credentials, and which is enormous, I would like to welcome Dr. M.U. Sharma, Dr. Manoj Umesh Sharma, to kindly grace the symposium, and please deliver your address. Thank you. Dr. Sharma, are you there, sir?
Yeah, I'm there. Am I audible?
Yes, sir. Yes, yes.
So thanks a lot, Dr. Kalpana, for that beautiful introduction. And it's a pleasure seeing you here after a long time.
I know, sir. I have moved to Pune actually. Now I
Yeah.
long back.
Yeah.
3 years back,
So,
I moved to Pune.
so wherever you are, you'll be doing extraordinary work. That I am very confident.
Thank you. Thank you so much. Thank you, sir.
So,
Hello.
anyways, I think today's conference topic or workshop is a beautiful topic, saving the environment. And yes, environment is a matter of concern. And definitely over the years, if I recollect, we have been continuously hearing about global warming and what is wrong with the environment and the temperature rising, whatnot. I think a lot has already been done theoretically, I should say, theoretically and through media to talk on these issues. But it's not sufficient. If we talk of environment, I think the first thing which is a matter of concern is that we don't get a good quality drinking water these days. I'm talking of natural water. We have to depend on the technologically available drinking water, be it in the form of AquaGuard or of RO water. And environment is not just, we always talk of the factors affecting human health as far as environment is concerned, but it's not just restricted to human health. If you talk of, I think I just saw a message, a country which is not protecting the soil is destroyed. So protection of soil is, again, it's a matter of concern. And the way the industrialization is blooming and growing, what we are doing as a result, we are damaging our soil, we are damaging our natural resources, we are damaging the groundwater. And then we talk of trees, and I think somebody had beautifully shown just now. I was going through the activities, and a nice pictorial representation of the trees, how the trees are getting damaged and how we have to protect the trees. And I think I joined some 10 minutes back, and actually I felt I should have joined long back, because the activities were really good. I felt somehow I missed a lot today, but somehow I was tied up, and I was, when I was about to join for, I got a message it's getting delayed. So I joined at 5. This particular conference, workshop, whatever you call it, actually it has done a very good work, roping in the youngsters, roping in different dignitaries, important personalities. And that's how we have to start somewhere. And topics were good. And I don't know what we can do. See, when we talk of DRDO scientists, frankly, we are all involved in all sorts of science and technology development, which is actually not protecting the environment, except for a few labs in DRDO, which are really into it, and they are working on the groundwater, they are working on the water resources, creating a filtration plant, creating good quality drinking water for the forces, for the services. So somewhere the academics has to jump in and come out with all good possibilities, good ideas, good thoughts, how we can really protect this beautiful environment where we are living, which we call the Mother Earth. Let me tell you, it's very easy to destroy it, very, very easy. But to protecting it and reviving it is a tough job. And that's where I should say the academics play a very important role, because we as DRDO scientists, we are more into the technology part, technology development, and we concentrate on the technology part. But the basic research, the basic idea from how we can do it, the way we can do it, comes through such conferences and deliberations that we have in such conferences. So it was really good. And I'm really fortunate to be a part of it, although at the fag end, and I enjoyed the activities. It was really good, nicely organized. The topics were good. And I congratulate you all for the grand success, how it has been managed. And let's hope this chain of people involved in this, it grows. Like specialists from different domains, they join hands with you all and move towards a sustainable root sustainable idea, how we can save the environment, how can we protect the environment. So I'm sorry I cannot talk much because I have to leave and I was waiting, and my sincere apologies for that. But I congratulate you all, and I thank Dr. Shipra for giving me this opportunity to interact with you all. Maybe sometime in future, I'll take more time and interact with you in a different forum. Thank you all. Thanks for the patient listening. That's all from me this time, and all the best. Thank you.
Thank you, sir, for accepting our request, and in such a short time you agreed to grace the occasions. I'm really thankful. Thank you.
It was a pleasure. Pleasure is all mine. Thank you.
Thank you so much, sir. Thanks a lot for your valuable words. We will remember and we will keep it.
Thank you.
Working on it. Thank you.
Thank you so much, sir. Thank you, ma'am. It's my honor to invite our chemistry department teacher in charge, Mr. Ajay Kumar, to address the audience, followed by the most exciting and awaited announcement of results. We are grateful again to sir for kindly providing us with books sponsored by Ayush Publications as rewards to the winners. Over to you, sir.
Pragya, please share the slides also from Ayush Publications.
Yes, ma'am. Ma'am, is it visible?
Yes, yes, it is visible, Pragya.
Okay, ma'am. So this is the first book, Advanced Inorganic Chemistry. The second book, Chemistry of the Main Group Elements. Third, Basic Inorganic Chemistry. Fourth book is Organometallic and Bioinorganic Chemistry. And Coordination Chemistry.
Pragya, the slides haven't moved.
You can click on the slide number 2.
Slideshow mode पे जाओ ना, या फिर slides पे click कर दो.
मैम वही किया है. अभी मैम first slide
doesn't matter. Pragya, doesn't matter. We all have taken look of it. आगे बढ़ो Devanshi and Pragya.
जी मैम.
Ajay sir, we request you to please continue with your address.
Let me start. Thank you so much, Dr. Pragya. I am Ajay Kumar, Associate Professor, Teacher in Charge, Department of Chemistry, Hindu College, and co-convener of this international conference. Thank you so much, sir, for gracing the valedictory session and addressing the gathering. Good evening, everyone. Honorable Chief Guest, Dr. Sumidha Kataria, Dr. M.U. Sharma, Dr. Kalpana Bhargav, Chairperson, Professor Anju Srivastava, Principal, Hindu College, who is not present in this conference because of some busy in some interviews, Professor Rina Jain, Vice Principal, Hindu College, and convener of this conference, Dr. Shipra Mishra, Director STE and convener of this conference, my esteemed colleagues, participants, and dear students. It has been an honor to be a part of this international conference, and I feel privileged to get the opportunity to share my views. I am sure that you all have learned a lot and enriched your knowledge from this international conference. I would like to appreciate the quality and caliber of the eminent speakers. The talks delivered by them were really outstanding. The topic selected for this international conference is really one of the most important because this is related to our daily life as it affects our health as well as wealth. I would like to appreciate the organizing team of EWASH to select such an interesting and necessary topic for discussion. Now, I would like to share my view in few words that agriculture produces safe and nutritious food and generate jobs and income. But simultaneously, agriculture is both a cause and victim of environmental pollution. Agriculture is a major source of water pollution because of a huge amount of huge amount of chemical fertilizers, insecticides, pesticides, herbicides, fungicides, bactericides, and other contaminants by the farmers. This polluted water is really very dangerous for health directly or indirectly. The farmers are advised to minimize these chemicals. The purpose of such a conference is not only to deliver and to listen the lectures, but we have to learn that how can we all contribute to minimize the pollution. It is a duty of all to save the lives of each other by minimize the environmental pollution. Now I would like to express my gratitude towards you all, my dear colleagues and students, to make this event of great success. This is possible. This is the passion of my dear colleagues and dear students to make this event of great success. Finally, my best wishes to the organized team, participants, and the students. Thanks.
Thank you, sir. I would now like to invite Dr. Meenu ma'am to announce the results for the student activity. Over to you, ma'am.
Thank you, Pragya. Mohit, can you display the slide, please?
Yeah, sure, ma'am. Just a second.
I would like to thank you very much for asking me to announce the much awaited results of the students' activities organized during EWASH 22. Mohit will display these slides of the results on my behalf.
The slide is visible.
So this is Mohit. This is the in which order? It is from day 1.
Yes, ma'am. It is starting from the reel making, ma'am.
Okay, so the day 1 we had 2 activities: the reel making activity and the screening.
Yes, ma'am.
Results for reel making contest from bottom to top. We have the second runner-up here, Danin K. Shaji. Next. The runner-up is Vidyanshi. Both are from BSc Honors Chemistry of Hindu College. And the winner is Prince Kumar from second year BSc Physical Science. Can we have the round of applause?
I would like to add here, ma'am, that Prince is the same boy, I guess, who has made the video for the organizing partners. So I think he's done a fabulous job there also.
Yes. Okay, Mohit, can we move on to the prizes of the second activity, which was screening quiz. Again, from bottom to top, we have the second runner-up, Akshay Kaushik from second year BSc Honors Chemistry. The runner-up is Gitanshu from first year BSc Honors Chemistry. And the winner is Gauri Vashisht from second year BSc Honors Chemistry. So can we have the clap for all the 3? Here we have the, is it consolation prize also? No. Okay, so today we had 2 competitions, 2 activities. It was Pictionary and the quiz. So for Pictionary, we have the prizes for Consolation Prize, Hidyanshi from Physical Science first year. Consolation Prize for Muskan Sharma, Physical Science second year. And the second runner-up is Ankit Sharma, second year BSc Honors Chemistry. Runner-up is Gitanshu again, first year BSc Honors Chemistry. And Kanupriya is the winner in this activity, second year BSc Honors Chemistry. She is from BSc Honors Chemistry second year class. So round of applause for all the three.
We have
Okay,
two
we
winners
have another winner. So there's a tie. We have two winners here, Abhay Pant and Kanupriya, both from BSc Honors Chemistry second year. Okay, so we move on to the next activity, the last one. It is Quiz Mania. And for Quiz Mania, there was tremendous participation. And we have the second runner-up, Ruchika, from first year BSc Honors Chemistry. The runner-up is Siddharth from BSc Honors Chemistry first year. Winner is Ankit Sharma, second year BSc Honors Chemistry. Do we have a tie here also?
Yes, ma'am.
Okay.
No, ma'am. Oh, no. In that, we have no tie. Only first winner.
So we have the sole winner, Ankit Sharma from BSc Honors Chemistry. So let's have the round of applause for all the winners of various activities. Thank you, Mohit.
Yes, ma'am.
Over to
I would
you,
like.
Mohit.
Yes, ma'am. Just a second. Ma'am, just a second. There is some technical issue that my laptop is hanged.
Host, I think you can disable the screen sharing, right? So probably here.
Ma'am, it's okay. It's now okay. Like I would like to. Okay. Finally, we have reached the end of this wonderful two-day conference. I want to thank you, the teacher convener of the society, Dr. Meenu Srivastava ma'am, for their unconditional support and the guidance at each level over any type of parameter, for which I am very much thankful. So we have such a teacher convener, we have such teacher in charge who support unconditionally in whole event. And I would like to thank you, council member, and all teacher coordinator and student coordinators who worked untidily for the successful conduction of these activities. I also thank you all the council members and all the students who are participated in these activities. Thank you so much, ma'am.
Okay, thank you, Mohit. It's my honor to now invite Professor Reena Jain, Vice Principal of Hindu College. Ma'am, please kindly announce the results for the presentation.
Pragya, I'm so sorry. I don't have any result with me.
रीना जी हमने भेजा है आपको, आप देखिए अपना व्हाट्सएप.
अच्छा मैम, मैं देखती हूँ. But before
इसीलिए हम
that,
कॉल कर रही थी मैं आपको.
A very good afternoon to all of you and a very happy Lodi to all. And I can see Kalpana ji, Sharma ji as a part of the valedictory session with us. We are extremely grateful that they could take out time from their busy schedule to be here with us. All the faculty members, Shipra ma'am, आपको मैं अपने साथ ही गिन रही हूँ, इसीलिए अलग से
मुझे थैंक्स देने की जरूरत नहीं है, ये तो हम सबका फंक्शन है.
And Shipra ma'am and all the guests who are still with us at almost 6 o'clock in this conference. And of course, dear students who have been sitting patiently participating in all this in the 2 days of conference. Yesterday, we started with a technical snag initially, but finally the issue was sorted out overnight, and hence we were able to accommodate all of you in the direct one-to-one mode. Otherwise, we were made to, watch everything via the YouTube link. So, but still, it was a wonderful, event. The workshop was a big success. All these speakers, they were able to come down to the level of the students and make the topic understand very well to the students. And then the student also very enthusiastically participate in various activities that has been organized. So a big congratulations to all the prize winners. Okay, so now we start with the prize, ceremony, which I would like to announce. I can't see anything now.
WhatsApp में?
नहीं मैम, मेरे पास है, हाँ. But I don't know the screen, how the screen is moving.
आप बोल दीजिए.
Okay, so we'll start with the presentation.
Yeah.
A presentation in the UG category. The best presentation in UG category, it goes to Ujjwal. I think it's the same person who has just won the another prize.
No, ma'am. This time, ma'am.
ये दूसरा है?
Yes, ma'am.
So sorry. Huh. And then in the researcher categories, the best presentation award goes to Pooja Rana. And the second researchers category, it goes to Komal Verma. A big congratulations to Pooja and Komal both.
And one more is there, Rina. Diksha Lohan also.
Acha, Diksha Lohan also in the research category.
They are all equal actually. So no
Okay,
first,
so
second.
3 prizes for research category: Pooja Rana, Komal Verma, and Diksha Lohan.
Yes.
And for UG, we have a separate category, which I have already announced. That goes to Ujjwal, because you can't compare UG student with the researchers. That is why UG category has been kept separately. We also have a faculty category. The best presentation in the faculty category goes to Kornika Banot. Kanika या Kornika? Kanika ही होगी ना मैम ये?
Kornika लिखा हुआ है, Banot.
Kornika है मैम,
Kornika Banot.
Kornika Banot. Are these prize winners with us right now?
लग नहीं रहा कोई भी है.
I can see Diksha, I can see Ujjwal in the participants list.
A big congratulations to all the prize winners. And the posters, since there were only 2 entries and both the posters were very good, so both the posters have been awarded the best poster award. So congratulations to all the award winners of the posters as well as paper presentation.
Thank you, ma'am.
Thank you, ma'am.
Thank you so much.
Thank you, everybody. Actually, on behalf of ST, I also would like to thank each and everyone. And special thanks to, of course, Professor Anju, Professor Reena, Professor Sharma, and Dr. Goyal for collaborating with us and giving us chance to collaborate with them. It has been really 4 years, and looks very nice to see that we have been continuing doing this collaboration, and hopefully we'll continue to do this in future also. Thank you, everybody. Special thanks to Devanshi also and other teachers, faculty, students who have participated and taken really pains to make this successful. Thank you. Special thanks to
You are the one who brings us all together.
No, it is all mutual actually. One person cannot do everything.
And congratulations, madam, for a grand success of the conference. And I'm sorry I could not attend for.
No, I understand, and at least there are few people whom I can depend at any time. Last moment they come to the rescue. So
I
you are
came
one
running
of
from
them.
the office actually.
Yeah, I know.
So,
It's difficult, but
but
then
thank you very much. And I'm glad that this conference was a grand success.
yeah, this time I'm also really satisfied because actually the talks were really good and interesting. रीना जी आपने थोड़ा मिस किया इस बार, but
थोड़ा नहीं, मैंने काफी मिस किया मैम,
हाँ,
काफी मिस
बहुत
किया
अच्छी
मैंने.
talks थी इस बार, तो
हाँ.
I can really say that, and it was not overwhelming. मतलब काफी आराम से अच्छे से हुआ.
हाँ, मतलब बच्चों के लेवल तक आके अगर हम लोग स्पीकर्स इस तरह से बच्चों के लेवल पर पहुंच के अगर समझा पाए, so nothing like that. I've been
And they are
getting a very
general
positive
topics,
feedback.
mostly
हाँ.
general, not highly technical, so that everybody
They can
understands.
understand. हाँ. I'm getting a very good feedback also from the students. They really
Chalo,
enjoy
अच्छी
being
बात.
a part of it.
Then I am really happy. If the students are happy, then we are happy. Actually, our objective
Yes,
is
that's
to
very
make
true,
them
ma'am.
happy and learn.
That's true, ma'am. So we come to a closure of this 2-day gala event.
We have a formal vote of thanks, ma'am.
Oh, acha. So sorry.
Okay.
Pragya, please announce.
Okay, thank you, ma'am. Thank you, everyone, for the kind words. Now, I would like to invite respected Dr. Charu Kumar, ma'am, the senior most faculty of chemistry department, for the concluding remarks and vote of thanks. Ma'am, please kindly take the charge.
Yeah. Thank you, Pragya. Am I audible? Is it okay?
Yes, ma'am.
Okay. Good evening and a very warm greeting to one and all present. I was looking for my turn to thank everybody formally. Gratitude is a powerful emotion that can be expressed in many ways. Sometimes a sincere thank you can do the trick. With this note, I, Dr. Charu Kumar, Associate Professor, Department of Chemistry, Hindu College, on behalf of the whole organizing team, feel incredibly honored and privileged to have been given the opportunity to formally propose the vote of thanks on this auspicious occasion. I sincerely thank our chief guest, Honorable Ms. Sumedha Kataria, ma'am, former IAS officer, retired Chief Administrator, Haryana and State Agricultural Marketing Board, Panchkula, for kindly accepting our invitation and gracing the function. Thank you, ma'am, for your very innovative address, and I am sure that all the participants have taken a note of your suggestions. I also extend my deep regards and hearty thanks to the Chief Patron, Professor Anubha Majumdar, Emeritus Professor, Jadavpur University, Kolkata, and former Director, AIIPH, Kolkata, co-patrons Professor R.K. Sharma, Honorary Secretary, Royal Society of Chemistry, London. Sir, we really admire your valuable guidance and the opportunities you provide to us. I extend my deep gratitude to Dr. S.K. Goyal, Chief Scientist and Head, CSIR, National Environmental Engineering Research Institute, for taking out time and grace this occasion. I would also like to extend my deep gratitude to Dr. Manoj Umesh Sharma, Scientist G, DRDO, who is the chief guest for this valedictory function. My thanks are also due to our principal, Professor Anju Srivastav, who leaves no stone unturned to provide best of opportunities and platform to our faculty and students. Words are not enough to thank our cherished conveners, Dr. Shipra Mishra, President, Save the Environment, and former Additional Director, DIPAS, DRDO Delhi, and Professor Reena Jain, Vice Principal, Hindu College, for their constant motivation, guidance, and support at all stages in organizing this conference. They have been the backbone and the pillars, and their organizational skills are unmatched. I also extend my deep appreciation and thanks to the co-conveners, Mr. Ajay Kumar, TIC, Associate Professor, Hindu College, Dr. Devanshi Maggu, Assistant Professor, Hindu College, and Mrs. Chanda Basu, General Secretary, STE, for their tireless efforts, support, and coordination in all possible ways in organizing this successful conference. I also mention my deep sense of appreciation to our organizing secretaries, Dr. Meenu Srivastav, Associate Professor, Hindu College, and convener of Tattva, the Chemistry Society, and other staff advisors of Tattva for their efforts and mentoring students in organization of all student activities. I also thank the organizing secretary, Dr. Sankha Chakrabarti, Assistant Professor, Kalinga School of Biotechnology, Chemical Technology, KIIT, deemed to be University, Bhubaneswar, and co-organizing secretaries, Dr. Jigni Mishra, Project Associate IARI, and EC member STE for their endless efforts and guidance. My cheers and thanks to the President of Tatva, Mohit Agarwal, and the student coordinators, Vansh and Aditi. Further, I would like to thank our keynote speakers, Professor Pankaj Kumar Roy, Professor and Director, School of Water Resources Engineering, Jadavpur University, Dr. Nipur Bahadur, Senior Fellow and Head, NMCG Terry Center of Excellence on Water Reuse, Dr. Neeraj Tandon, former Scientist G and Head, Medicinal Plant Division, ICMR New Delhi, for giving excellent and valuable presentations by highlighting the theme of the conference. I also propose hearty thanks to our invited speakers, Dr. Ramesh Kumar, Postdoc Researcher, Department of Earth Resource and Environmental Engineering, Hanyang University, Republic of Korea, Dr. Suhail Parvez, Professor and Head, Department of Toxicology, School of Chemical and Life Sciences, Jamia Hamdard, Dr. Scientist, CSIR, National Environmental Engineering Research Institute, Dr. R.K. Gupta, Associate Professor, Department of Chemical Engineering, IIT Kanpur, Dr. Pinaki Dey, Senior Scientist, CSIR, National Institute for Interdisciplinary Sciences and Technology, Kerala, Dr. Jayato Nayak, Assistant Professor, Center for Life Sciences, Mahindra University, Hyderabad, Dr. Aarti Bhatt, Scientist F, Center for Fire Explosives and Environmental Safety, DRDO, Dr. Girija Bharat, Director, Mu Gamma Consultants Private Limited, Gurgaon, Professor Shubhankar Suman, Associate Professor, SOM Oncology Academic Department, Georgetown University, for sparing their valuable time and making the conference interesting and meaningful through their stimulating work and an attempt to achieve sustainable development goals, which really is commendable and a source of inspiration. Further, I wish to express my deep gratitude to the chairpersons of the keynote and technical sessions, Dr. Lakshman Prasad, Group Advisor, RKJIT Group Ghaziabad, Dr. Patri, former Outstanding Scientist, Director, NMRL DRDO, Professor Parimal, HAG Professor, Department of Chemical Engineering, NIIT Durgapur, West Bengal, Professor Sunil Dey, Professor and Head, Department of Geography, Northeastern Hill University, Shillong, Dr. Amrita Mishra, Associate Professor, KIT School of Biotechnology, Bhubaneswar, Dr. Suraj K. Tripathi, Associate Professor and Associate Dean, KIT School of Chemical Technology, Bhubaneswar, Dr. Maulin P. Shah, Chief Scientist and Head at Enviro Technology Limited, Ankleshwar, Gujarat, Dr. Rajinder Singh Chauhan, Dean, Center for Life Sciences, Mahindra University, Hyderabad, Dr. Usha Panjwani, Scientist G, DPAS DRDO, Dr. Kalpana Bhargav, Additional Director, Scientist F, High Energy Material Research Science Lab, DRDO, Professor Shachi Shah, Director, SOITS, and Professor in Environment and Studies in IGNU, Dr. Sushil Kumar Singh, Additional Director, DRDO, and Dr. Rabia from Jamia Hamdard. We are grateful and thankful to all of you for the smooth running of the technical and keynote sessions. I would also like to extend my good wishes and congratulate all the STE awardees for their exemplary work and achievement. I also congratulate all the student winners who have actively participated in various competitions organized in the conference. I sincerely thank all the members of the Save the Environment and all faculty members of the Department of Chemistry, Hindu College, University of Delhi for their sincere efforts, guidance, and perseverance in making this conference a sparkling gala event. I would also like to thank the technical team for the uninterrupted running of the conference. We have been fortunate enough to be backed by a team of very motivated, enthusiastic, and dedicated students and volunteers from the Department of Chemistry, Hindu College. Your efforts are commendable, and in fact, the students are the motivation and reason behind all these efforts. My special thanks are due to our sponsors, Gaur Surgicals and Ayush Publication, for rendering and supporting our mission in making this conference a successful one. And I'm sure that the conferences organized by STE and other associates will continue to get a generous support in future also. I cannot thank everyone enough for the involvement they have shown and the willingness they have expressed to take on the completion of the task beyond their comfort zone. And pardon me, please, if I have missed out any name. In the end, I would like to thank everyone and wish that Hindu College will get more such opportunities, and it will be our pleasure to organize such events in future also. Looking forward to a more sustainable development and a better future ahead. Thank you all and namaskar.
Thank you so much, ma'am, for your kind words. And with this, we come to the end of a successful conference, EWASH 2022. I request the audience to please rise up for the national anthem. Thank you.
Okay, then. Thank you, everybody, once again, each and everyone, all the students, faculty. Special thanks to Professor Anju, Reena ji, Devanshi, Meenu, Charu ji, all, Ajay ji, Dinesh. सब लोगों का नाम लेना मुश्किल है, सब लोगों के. So I'm really happy, and
we really wish that this becomes an annual event and we get the privilege to.
Correct. We'd like to continue like this. And special thanks to Kalpana and of course MU Sharma ji. बहुत short notice में इन लोगों ने हमारी request accept की, और मतलब in spite of having office till 5 o'clock, Kalpana made it possible. So that's a great thing.
Thank you, ma'am. Thank you so much. When we are together for so long, it becomes difficult to break and snap in a moment. But all good things do come to an end. So now we will have to say goodbye and be ready to meet the next time sometime soon.
Yes.
Thank you. Thank you so much,
Thank
ma'am.
you, ma'am.
A big thanks to everybody, including the technical team. Thank you so much. Thank you again. Bye-bye and happy Lodi.
Happy Lodi.
Happy Lodi.
Happy Lodi.
Happy Lodi. Yes. So we have finished well in time, actually. Everyone can enjoy Lodi now. Bye.
Okay, ma'am. Bye. Thank you.
Manoj.